Perfect, good stuff. Okay, so I'm gonna be doing the timing for the talk. What examples do you want me to do? The aside or like a two good?
So I have 2 minutes, right? When I get any questions. So I think I'll be talking for about 15 minutes. So I would say at 13, to make it very obvious.
Sure, okay, sounds good.
And then I will wrap it up and hopefully have time for a question or two.
Sounds good. Yeah, and then one thing we're doing is we're having the owl centered on the stage. So if someone asks a question from the audience, they just like repeat that question, go and answer it for people on the live stream and hear the question. Is that okay?
Yep.
And we've got a clicker for you. Yeah, that's all my things.
Okay, so we've been.
Thank you. Yeah, after I people like we're starting. And then like
13, give me a signal at 14, make it look happen. I do think we'll go with 13.
All right. Good morning, everyone. Welcome to our speed talks. We're going to be starting with our very first speed talk at 10:05, and then we'll be kind of running pretty continuously for the rest of the day. We'll have a little break at noon for some trivia in here, which will be very exciting. That will be led by me. And yeah, so if you just want to sit tight for the next couple minutes, our first speed talk is not far away. Yes, I just got to go out.
Are you first?
Yeah.
You're gonna be around, so everybody can play basketball. Who's next?
That's what I mean. I'm doing that. Yeah. That's right.
Yeah.
Okay, for those of you joining us on live stream, hi, we'll be starting in 2 minutes.
You don't remember that? I will lend you the email. Can't remember. Yeah.
They're right up there. It's the time. Hi, everyone. Thank you so much for joining us today at the Moss Landing Green Labs Open House. We're going to get started with our speed talks. Sorry if you heard this before, if you've been in here, but if you just came in, our speed talks are going to be running basically all day with a little brief interlude for trivia at lunch. And most of our talks are going to have a time for questions at the end of them. So if you've got questions, hold on to them. And yeah, all of our speakers are super excited to chat with you. All right, take it away, Guido.
Well, thank you for coming to hear my talk on penguins today. I'm always excited to talk a little bit about the research that students and I are doing in my lab. And in particular, one of my favorite organisms is the emperor penguin. So emperor penguins are one of 18 penguin species that are all found in the southern hemisphere. They range in size from the small 3-pound little blue penguin up to the large over 80-pound emperor penguin that I'll be talking about today. Emperor penguins have a circumpolar distribution, and with the use of satellite technology, so pictures taken from space, they have now identified 66 different colonies that range in size from less than a couple hundred birds to over 20,000 pairs. Before I talk about the research expedition that we took to Antarctica last year, I want to do a brief introduction to their annual cycle so you'll better understand when we were conducting our research. So emperor penguins return to their breeding colonies kind of in their fall. So, mid-April, mid to late April. They then, for 6 weeks, fast, stay out on the ice in the breeding colonies, have their courtship behavior, and then the female lays the egg after about 6 weeks, transfers this to the male, and then she goes out to sea for 2 months and feeds. So this blue area is when they're out at sea feeding. She comes back after about 2 months while the male has been fasting this entire time while incubating the egg. And then he goes out, it's his turn to go out and forage. And then once he returns, they start to alternate. When the chicks are little, there's always one adult that stays with the chick. And as they get older, as in this picture here, the birds are big enough that both adults can be out feeding at the same time. So the adults will go out and forage for about a week, come back, feed their chicks, and then go right back out again. So feed their chick for about a day. So that's the chick rearing. At the end of chick rearing, this period right here, the adults just stop coming back to the colony and they start their pre-molt migration. And we put some tags out. So there was a team of us that went down to Cape Crozier in 2022. We put some tags out on birds. We got several foraging trips, but then the bird took off on its pre-molt migration. And one of the reasons we left the tags on is we were going out later that year to try to catch birds after they molted, and these tags were going to tell us where they were going. I have to admit, I got a little bit scared. All the literature says that they should pretty much molt in that square box, and our birds were going much further. The year 2022 to 23, that summer, Antarctic summer, had the lowest ice on record by about 6 standard deviations. So very little ice. So there's usually a lot of really good ice in this box, but it wasn't there. So they kept going. So that we'll come back to that when we talk about that. So that's that pre-molt. But once they get to their molting grounds, they molt. So any tags we put on them at the breeding colony, they fall off. And it's hard to get to these remote areas of Antarctica. So we don't know very much about what they do after they molt. And so that's the project that I'm going to talk to you about today. So this is part of a bigger project where we were studying the foraging ecology and habitat use of Ross Sea emperor penguins. So basically we were putting data loggers on them that measure their movement and diving behavior, trying to identify if there's individual strategies. Are these strategies associated with colony, sex, mass, or condition? And then we're integrating that with environmental data to understand what features of the environment are important for them. And the reason that this is important, this is a little bit of a discouraging image, is that if we don't change our behavior and change the rate at which the global temperature is increasing, 90% of emperor penguin colonies are predicted to be extinct, basically in 75 years. So all of these red dots are penguin colonies that will not exist in about 75 to 80 years, based on current modeling projections. So these models are based primarily on what are the sea ice conditions during the breeding season. So we want to learn more about the rest of the year so we can improve the accuracy of the models. But I do want to leave on a slightly positive. If we do change our behavior and we can restrict temperature rise to about 2 degrees Celsius, a lot more of the colonies will be okay. Some of the more southern ones, and there's others that are just threatened. So it is promising if we keep moving in the right direction. The reason we are focusing on the Ross Sea area is if you look at this picture, you can see this is one of those areas that's predicted to be a little bit of a refuge where they might survive with the increased temperatures. And it's also where 25% of the penguins reside. So it's an important, a large number of penguins live in this Ross Sea area, and it's also a potential refuge. So our study actually took place, and we had 2 different study periods. One was in Lake Chickreary, but what I'm going to focus on today was our trip to the Eastern Ross Sea to study that post-molt migration. And today's talk is going to be more about the expedition. We're still working on the data, but I'll give you a glimpse of what we're seeing. So how do we do this? Well, this took us a long time to actually get there. So first, we put together a nice group of scientists, a colleague at USGS, a graduate student, a postdoc, and me. So solid team, went down, traveled to New Zealand during COVID time. So you see we're wearing masks a lot of the time. If we got COVID before we got to the Antarctic, our project was off. Once we got to New Zealand, we had to try on all of our gear. You have to have a lot of warm gear, you have jackets, long underwear, and boots, try it all on, make sure it works. Then it's a lot of waiting, and we had to wait in our hotel rooms or outside because if we got COVID again, project canceled. So here we checked into our flight, we're having breakfast. This is actually from a different trip, but getting to the Antarctic, weather can get in the way. So a lot of times there's more waiting. This is the day where we actually boarded the plane, they found a mechanical issue, and then we went back and just, you can see postdoc sleeping under the table here. So lots of waiting. We finally are on our way to McMurdo, weather's good. You know, we get these lovely flights where we feel like cargo, but we're traveling down in these planes that have skis so they can land on the ice shelf. And then there's 4 very happy people to have finally gotten down there after about 2 weeks of isolating at home, 1 week of isolating in New Zealand. Now we're finally in the Antarctic. And then this was our bus into town. So I always joked if I wasn't going to be a scientist, I'd write kids' books about all the crazy vehicles in the Antarctic. So while we were in McMurdo, it's not a very nice, attractive town. We were actually housed in a little building over here where we were isolated from the rest of the community again. So we didn't get COVID, but we were allowed to get out and get hiking. And when we were on our hike, we actually saw our new home arriving into port. So we spent about a week here in isolation, taking numerous COVID tests. And once we passed all our COVID tests, we were allowed to board our ship, and we were very excited to get on board and finally be able to take our masks off. We are sharing this vessel with people. So we actually still had another 4 weeks before we started looking for penguins, but I'm going to skip those 4 weeks because we were with a geology cruise that just went back and forth over a grid for 21 days. But finally we got the ship. We went looking for penguins. We headed over to that Eastern Ross Sea. We saw lots of birds, the wrong species. We saw pieces of ice that would be too dangerous for us to work on. And then finally we started to see what we wanted. So here you can see some emperor penguins on a nice big ice floe that we could safely catch on. So then it was time for us to get off the big boat into our little boats. And then we got to work in, we often take photos on the really beautiful days. So beautiful sunny day, reflection, we're trying to cut off the birds. And what we'll usually do is we'll just kind of surround a small group of these. And I have a short video of how we do this. So this is one of the less nice days. And so it's a very slow, we try to let the birds we're not interested in get by. And then this is actually just a gift, or it will stop kind of right as we get to our bird. But we're letting, we're not interested in these. So yeah, so that's sort of how it is. And then we hook them with those hooks and we can catch them. Once we catch them, we put a hood on them, and then they tend to be relatively calm. This is a bird that's just kind of leaning on the leg, and this allows us to tape the instruments onto the bird. So here you can see this, we put a little bit of tape, and then we use super glue to secure the tag onto the bird. This does very little damage to their feathers, so when they molt off, there's very little remnants of what we have done. This is an example of us working on an ice floe. You can see the Zodiac that took us there and our kind of home base in the background. Once we caught our birds, we would measure their flipper, we'd weigh them, we'd attach our instruments, and then we'd let them go. This is the second day we actually saw one of our birds with a tag here. So this is now out collecting data. It's transmitting where that bird is, what its diving behavior is. And so the penguins are off collecting data, and then we started our journey home. So this is our crew grew a little bit. We adopted some people on the boat, made it a little bit easier to have some extra bodies on the capture. So this is after our last capture, took a group photo, had a great team, and then we started home. Our trip home sucked. I'm not gonna lie. There was like 3 storms in a way, kind of in a row. Our captain managed to keep like threading the needle between 2 of them. So this is kind of an image of the seas. We had about 9 or 10 days of this. And this is not as bad as it got. This was a day where we actually felt good enough to get up and, take photos. So that was horrible. So we were very excited when we started to see land in New Zealand here. And then once we got back there, we were excited to go hiking. And here's our little boat again. So very different from the first time we saw it in McMurdo. Now it's summer in New Zealand. So now just a little bit of the data. So while we were traveling home, these birds were out foraging, moving, transmitting their data. And this is showing data from 16 of the 30 birds that we instrumented. So this is where we tagged them, kind of in these little star areas. 8 birds returned to Western penguin colonies in order to breed, and 8 remained in the Eastern colonies. And so I was a little bit surprised, like, okay, these birds are traveling a lot less than these, and you can see that here. So birds that returned to the Western colony, which has the largest colony, had to travel 2 to 3 times further than the birds that stayed on the east in the Eastern colonies. This meant that they actually had to travel a lot faster. So they were traveling about 40 kilometers a day versus about 25 kilometers a day. So in order to get to their breeding colonies, they had to move a lot faster. They also, even with moving faster, they arrived to their colonies 2 weeks later. So we always thought that the birds arrived in April. Previous studies have shown that. In this year where we had very low ice, and we think that the birds might have had to travel further east than normal, they arrived at the colony much later than we thought they would. And then despite having that difference in how fast they were moving, their diving behavior was very similar. So they're probably feeding on similar things. They did about 80 dives per day. Average dive depth, or kind of average maximum dive depth, was between 50 and 100 meters. Maximum dive duration, or maximum dive depth, they can dive to 300 to 400 meters, so over 1,000 feet. Average dives are 3 to 4 minutes, but they can dive for up to 20 minutes. So dive behavior was similar, but they had to travel a lot further. And what we think this means is that at least in years where there's extremely low ice, like there was in January, February 2023, the Western birds had to travel much further than normal. The birds that ended up going back to Western colonies tended to weigh a little bit less. So it's possible that this longer journey made them kind of a lower body condition. If these types of low ice conditions persist, or we continue to have more events of these extreme conditions, it's going to put these Western birds, where more of the birds are actually historically have been breeding, it's going to increase their energetic requirements, potentially put them at greater risk, having to travel much further to find that stable sea ice. And so why it's so important to have that stable sea ice is they molt, they lose all of their feathers, and they can't go into the water when that's happening. So they need to find ice that's going to be stable for that entire period. And then there's also, if the birds continue to arrive about 2 weeks late, then there might end up being a mismatch. So a lot of penguin colony failures in the past has been because the ice breaks out too early and the chicks kind of go out before they're ready. If they're starting to breed later, then that's going to potentially, even if the sea ice goes out at the normal time, the chicks may be too young. So Not looking good. I want to get down there and do this in some more normal years and kind of see what's going on. And this is just, like I said, the start. We have a postdoc that's now trying to link some of their behavior to environmental variables. And with that, I don't know if I have time for questions.
We do.
Okay. Mhm.
Are there behavioral changes that the penguins might do that would help them adapt to the changing conditions?
Well, that's what we're trying to find. Like, so they went further. So that is a change. They swam much further than we've seen in the past to get to that stable ice. There's not much else they can do other than that. Like, they have to have good ice in order to breed that they can get or to molt. It could be that maybe more birds start to breed in those eastern colonies, but that could have impacts. That's one of the reasons we want to know more about their foraging behavior is there may not be enough food during the breeding season to support more birds breeding over there. So there's like a chain reaction. So it'll be a little bit interesting to see kind of how their dynamics change. And we are trying to learn more about what they're eating during the breeding season, how it's changing. So.
What is their percentage of weight droppage when they're in fasting?
So they can get down, so at the end of the molt, they're the lightest. We weighed a bird that was about 17 kilos, so 35 pounds.
So that's like,
So at breeding, when we're catching them during the breeding season, they're about 25 kilos. They probably fatten up a little bit on that molt migration, but we aren't weighing them right when they get to the molting grounds. So I know at the breeding season, they're about 25, 23. And then when we're catching them during the molt, they were about 17 to 18. But then the trip that we're studying that we want to learn more about, they need to go from about 20 kilos up to about 40 kilos for breeding.
yeah.
So that's one of the reasons we really want to learn more about this trip, because that is an important one for them to really fatten up before the breeding season. And the males then have to fast for 4 months. So,
Awesome.
Yeah.
All right.
okay, I gotta go.
That's all the time we have. Thank you so much, Gita. Our next presenter. So yeah, thank you, Gita. And our next presenter is ready to take it away.
Okay, so my name is Katie. I'm a student in the psychology lab, but this talk is just going to be on wetlands and local conservation going on with them. Very broad overview. So let's get started with what is a wetland? Pretty much exactly what it sounds like. It's wetland. The EPA defines wetlands as, quote, wetlands are areas where water covers the soil or is present either at or near the surface of the soil all of the year or for varying periods of year or of time during the year, including during the growing season. Very vague. So that can be just about anything. They can be coastal or inland. We're going to talk about our coastal wetlands today. They're highly productive, generally ecologically important. Our local wetlands support a variety of migratory birds, juvenile fish coming in from the ocean, and also marine mammals. We have harbor seals giving birth to pups right now in our wetlands, and also many sea otters. They also support a variety of other animals like that Hymenopteron sponge in that picture right there. And this productivity is underlined by very complex hydrology and geochemical processes. If this works, we're just gonna have to do a manual transition. I'm just gonna speed walk through this. This is just for one nutrient. This is nitrogen in the sediment. Basically, the plants change the oxygen condition in the sediment, changes what type of nitrogen cycling you have going on in the soil via the microbes, which changes the availability of the nitrogen, the type of nitrogen available. It's all very complex, and that's just for one nutrient. So we're going to breeze on past. It gets even more complex when you consider the variety of wetland habitats that we have. Mangroves are considered wetlands, tidal marshes, mud flats, the list goes on. These are also both salt marsh, the one on the left. Sorry about that. This one on the left right here is actually just off of Martin's Point of View. That's salt marsh and mud flats at low tide. The one on the left, right over here is salt marsh at high tide. So these are highly dynamic environments with different types of plants, even within the same definition. Again, it's very, very complex. So you have your biogeochemistry affecting your ecology, affecting your habitat type, and it kind of mimetically fractals out from there. And in any conservation talk, there does have to be a bit of doom and gloom. It's estimated that 21% of wetlands have been lost globally, up to 50% in regional hotspots. This really started during the Industrial Revolution. You can see around 1700, there's this sharp decrease. And then this heat map on the right over here is, if it's red, that means there's over 75% loss. You can see North America and along the coast of California is considered in that, is in that category. The primary drivers of this degradation are urban and agricultural land use. This is something that also impacts our local wetlands. I'm going to breeze through this so y'all don't have to read it. Basically, historic records like maps and letters indicate that significant loss has gone on, but we don't know how much. But we know that tidal marsh, mudflats, willow stands, all of those have been lost in some shape or form. Local wetlands local waterways are heavily impacted by eutrophication and poor water quality. Eutrophication is just basically where you have too many nutrients in the water and you get this massive algal bloom that when it dies, it creates an anoxic zone from all of the bacteria eating it. Luckily, water quality has improved recently. In 2010, water surveys by the Elkhorn Slough National Estuarine Research Reserve revealed that most of the sites they monitored were either moderately to hyper-eutrophied, which is just a fancy way of saying very, very, very eutrophied. And then 2019, there was an overall increase in water quality. You can see that in this figure over here. This is 2001. You can see it's more yellow, especially in the harbor mouth and the body of Elkhorn Slough right there. And this increase in water quality is associated with conservation efforts and also increased tidal flow. So local conservation is actually really interesting. It's a highly collaborative thing. We have two main conservation groups in the area. We have the Elkhorn Slough National Estuarine Research Reserve, or ESNR for short, and the Central Coast Wetlands Group. They partner with researchers, students in this lab, the public, landowners, businesses, and volunteers to restore wetlands. And I'm just going to go over a short list of local restoration focuses that these two groups have undergone. We're going to break them down into conservation, research, and monitoring. For ESNR, some of their conservation projects include Hester Marsh, where they're restoring grassland and the wetland itself. And the grassland is actually really cool. So they're replanting native grass and managing it with cattle grazing. So it's, again, an agricultural partnership right there. Research, there's research into our locally endangered Olympia oyster population. Some students have helped with that. You might see their posters along the hallway about oyster genetics. Also research into introduced species like the European green crab and that Hymenyacidon sponge that was in the first picture. And they have that continuous monitoring. Since 1986, there's been monitoring throughout the year where they look at water quality, weather, biological surveys. And if you go to their website, you can actually check that out and see how the water quality has changed through the year. And then CCWG is the other one that's in the area. They mainly focus on Morro Coho. Their main goals are reducing agricultural runoff in the Morro Coho Slough. And some of their research that I'm interested in, I'm working on, is the effectiveness of bioreactors in wetlands and the effectiveness of treatment wetlands. They also monitor the water quality of these treatment wetlands to see how well they're removing nutrients from agricultural runoff. So what does that look like all together? Come on. So on the conservation side of things, this is an aerial view of the Castroville Slough treatment wetland. You probably passed by it if you were driving from on the northbound. It is 12 acres of wetland that was built to reduce the amount of nutrients in agricultural runoff and to prevent eutrophication further downstream. So these white arrows are the flow of water from this Castroville drainage ditch. And as water flows through, there's a very significant reduction in nutrients. Nitrogen is reduced by 95 to 99%, and phosphate is reduced by up to 80%. And this little circle right here is the site of where an experimental bioreactor was. And a bioreactor, in this case, is just a pile of wood chips. It creates an anoxic environment that those bacteria like. And basically you get nitrate in and nitrogen gas out. So you're taking nitrogen out of the water and shunting it into the atmosphere. And this is some data that I've worked up from the bioreactor. The teal line on top is water that's flowing into that bioreactor. And then these two purple and pink lines on the bottom is the overall reduction as it left those wood chip bioreactors. And that's kind of on the research side of things. You can see it was highly successful. And then there is monitoring involved with this, and this is a partnership with ESNER and CCWG. So this is data from all of them. You can see that, and you have it broken down into exceeding specific concentrations. So you have 10, 8 milligrams and 1.7 milligrams of nitrogen per liter of water that is leaving Morrococo Slough, where this treatment wetland is. And you can see that from 1989 to 2006, you had it exceeding these thresholds quite a bit, 5% of the time and 12% of the time when they were monitored. Then 2006 to 2016, that dropped to about 0.3 and 4%. And then the Castroville Slough treatment wetland was operational in 2016 to 2019. Those exceedances dropped down to 0%. So kind of to wrap this up, these wetlands are highly successful at reducing nutrients. Conservation helps with mitigating that agricultural runoff. And that's, sorry, wow, my brain just blinked there for a second. Our wetlands are very important for our local ecology. Our sea otters, harbor seals are reliant on them. And they also have the capability to reduce eutrophication. And these partnerships with businesses, agriculture owners, research, and these conservation groups is really what drives kind of this fantastic restoration that we see going on here. And it's something that is, I think, very special and kind of unique to this area. Anyways, thank you for your time. I also included some QR codes up there to CCWG's and ESNER's websites if you want to check them out. ESNER has a really cool sub-tab where you can check out water quality throughout the years, and I would highly recommend taking a look at that because it's very fantastic and interesting. But thank you. Does anybody have any questions?
Can you go back to the table where you showed the progress? So, the number of samples collected between 2016 and 29 is quite a bit smaller. Is that because the period of time is less?
It's probably a period of time, and also this is a compilation of many different groups. So there's ESNER, CCWG, and other monitoring efforts that have all contributed to this. And ESNER was the main one on its own from 1998 to 2006. And then a couple of more groups hopped on from 2006 to 2016, but that 31 number is probably just because smaller time frame as well. Any other questions? Good. Okay.
Thank you so much, Katie. The next talk is going to be by our fearless leader, Petra, but we're going to start that at 10:35, just in case anyone is joining virtually and on that schedule that we published. So just sit tight for the next couple minutes.
We're gonna. Yeah, no, it's so silly, but.
No, it's not silly. That's
Exactly.
it's professional.
If you want to test out the clicker, maybe you said it's getting a person help you.
I think it's low. It's just like flashing. I don't know if that means that. Oh, there we go. But I think, can you click it again? I just feel like I am gonna,
Let me see if I can,
if you, oh, it will.
that did the animation.
Okay, great. Yeah, thanks. Okay, you can go back. Thanks.
Yeah, and I tested everybody's animation and stuff,
Oh,
and sound, all of it.
You want to tell me when to start?
Yes, I will.
Yeah, you got 1 minute, so.
Yeah.
Right? Yes.
I thought of you the other day because Scott Hamilton gave a talk about conservation, you know, and about fishing inside the reef areas and the outside. It's an amazing data set you contributed to. Yeah. Yeah. Let's chat. I want that. Yes.
Nice to see you.
Should I start?
Yes, all right, Petra is going to share some of her thoughts with us. Petra, take it away.
Okay, I'm gonna start a timer because my stopwatch, because I have a tendency to talk too much. Okay,
Are you an academic?
yes, it's a characteristic of us. Welcome, everyone. I'm Petra. I'm the executive director here at Moss Landing Marine Labs. I started here in September and it's been just an amazing 8 months. And so what I wanted to do a little bit today is just talk broadly about how climate and our oceans are changing and how the research that's done here at Moss Landing is so critical to our understanding of how the ocean is changing. And just a little bit about what makes this place unique. Okay, so that's my plan. So I'm going to start. I'm supposed to use the clicker, they said. There we go. Okay, so I'm going to start with this animation. So what you're seeing here is there are the months of the year are going around, and this is long-term global temperature data. So as it's going around, it's recording January, February, March, and then you can see the years in the center. And it's all being done relative to a mean. So the further out it goes, the warmer it's getting, right? And so I'm using this animation as a way to demonstrate the global change in temperature. So let's see, we're in the 80s. You can see it's starting to, you know, the colors are also representing the temperatures. You can see it's going out, and then look at that dramatic change since 2000. Yeah, so climate is changing. When I first started school, so you can see when they turn it off up the head, so temperatures, global temperatures have already warmed by 2 degrees Fahrenheit. When I started graduate school, climate change was something that was, you talked about as like, oh, it's happening, but like, as though we had all of this time to adapt. We don't, it's happening now. We're already there. Sea surface temperature is also changing. This does not have a nifty animation, but I'll just talk you through it. So this is similar in that this is the average monthly temperature. So from January through December, and each squiggly line represents a year. On this website, if you guys get curious, you can go to this website. You can actually click on each of the individual lines to see what year they are, but they do exactly what the previous plot did, right? So the earliest data in the 80s is down here, and as you move up, it's getting warmer and warmer, and this orange line is 2023, and this is 2024. So, the sea surface temperatures are going up. This is just a different way to show you that data rather than all the squiggly lines. If you average all of that, you can see this very, very clear trend. So this is from the 80s to 2025, and you can, 2023, I think this goes. This is the, we tend to look at anomalies or differences from the mean, and you can see there's a very clear trend towards warming, and in particular, there's this really big spike the last few years. So that's all global stuff. So I want to focus on us in a little bit and think about what is happening in California. So this is the change in temperature in degrees Fahrenheit in California. It very clearly shows you that the temperatures are going up. Droughts are becoming more frequent in California. The snowpack, the long-term trend in snowpack is that there's less, which is really critical for our water resources. That's where all our fresh water comes from. Sea level is rising. Wildfires are increasing. And so we think of these things as like really broad terms, like when you think about, okay, sea level is rising. And when you see the numbers, they don't look like big numbers. But when you look at the impacts of sea level rise, you're all from the area, you'll remember this happening a couple of years ago in Capitola. What it means when we have sea level rise is that it's not just the change in the average. It means that when the tides are high, they're higher. When a storm surge comes in, the surge is bigger. And that's where a lot of the damage comes from along the coastal areas. And there's lots of other changes happening in the ocean, right? This is a picture of what used to be a kelp forest and is now just covered by sea urchins. And the cause of this is a variety of things, but it definitely was triggered by an initial heat wave in the ocean. So where there should be lots and lots of kelp, there's very little kelp because these urchins are blooming because the sea stars are not around to eat the urchins. So there's a lack of a balance there. And so we have a dramatic reduction in the amount of kelp forest. This is a picture of harmful algal blooms. So this is, the research on this is really, really interesting in terms of what triggers it. It's a combination of temperature, but also nutrient concentrations. These are all things, by the way, that there will be talks on later today. So you'll hear more about each of these things. Okay, so what do we do at Moss Landing? What is the kind of research that people do? It's a pretty wide range of research. One of the things I love about this lab is it has a very long history. So nearly 60 years of having a really diverse set of research, right? Anywhere from physical oceanography to marine geology, to vertebrate ecology, invertebrate ecology. But when I think about what we've just talked about, the research here really helps us understand how the ocean is changing. It helps to find ways to minimize the impact of the changes. And it helps us build climate resilience. So I'm just going to give you a few examples of that. I'm not going to talk about these a lot because there are talks about these later today. So there's two on here. Actually, there's several of them here. So the CalHAB maps, there will be a student talking about this today. And the Central and Northern California Ocean Observation System and this California Collaborative Fisheries Research. These are long-term monitoring programs. So what that means is that somebody goes out at these sites very regularly and takes measurements. And these are the kinds of things that traditionally, what happens is they get funding from the National Science Foundation or something like that, but it can be really hard to keep up that funding because, the newest, most exciting science has come along. And you're like, well, you've been measuring the same thing for, 20 years, but that data is so critical for helping us to understand how climate has been changing, how the harmful algal blooms have changed, how the temperature has been changing. So these are really, really critical projects that a lot of people at Moss Landing Marine Labs are involved in, both in terms of researchers and students. And then this ocean iron fertilization, this is a really good example of the kind of research that people are doing to say, how can we mitigate climate change? Is there a way that we can reduce the impact? And the idea here is, and there will be a talk on this later as well, is that if you add iron to certain parts of the ocean, it can increase productivity and draw down carbon dioxide from the atmosphere. So there's a really wide range of work that's happening here. Okay, so why is Moss Landing so unique? What puts us in a position to be able to do this kind of work in this broad scale? So you're sitting right now in the main building, right? So you're sitting in this building. This was a beautiful day. This is just a lot of days look like this at Moss Landing. It's kind of lovely. I was just me on a walk taking a picture. But in addition to the main building, which has labs, so I hope that you take the time to go see some of the labs and some of the facilities that are here in the main labs. But in addition to the main building, we have a set of other properties that I want to tell you a little bit more about. We have an aquaculture center where people are doing all kinds of research on cultivating kelp. They're doing research on the effects of ocean acidification and low oxygen on different species of fish. They're doing research on kelp restoration. We also have an additional lab building up here, the Norte facility. And then we also have our marine operations, which I'll tell you a little bit about. But it's those resources, those physical resources that really allow us to do a wide range of work. So we have a boating program. So we have a set of boats. Some of them are small boats. What you're seeing here is pictures of students in these small boats. One of the things that is so amazing about this program is that not only do we have these resources, but we have the program that trains our students to use them, right? So students don't need to wait for faculty to take them out on boats. They get trained on how to use the boats safely. And they can go out and do their own work. So those marine operations are really, really important. We also have an amazing research diving program. So our students get certified if they want to, if their project is relevant to this, they get certified in scientific diving. So they take the boats out, they take their gear and they go out and they do their research and their diving. Is that my 5-minute warning?
That's you talked for 10 minutes, 5.
Okay, thank you. That's perfect. Okay, and then we have, like I said, in the main building itself, we have a lot of resources that are available to students. So I was talking to someone outside earlier. I feel like when you do climate change research and when you do ocean research, it can be kind of depressing. Like there's a lot of sad information there about how the climate is changing and how it's impacting the ocean. But the thing that I have found since I started this job is that I'm so much more optimistic. Like there's so much energy in this building from the faculty and the researchers, and most importantly from the students. And the work they're doing, I think, is where I find hope in that work. There's some pictures of students here. And then I'm just going to wrap up. I don't know if you all follow Moss Landing Marine Labs on social media, but if not, you should. They've done a really great job of like highlighting students. They're doing a wide range of research, and they are the reason why they're part of the reason why we're here. Training the next generation of marine scientists is really critical to what we do. And let's see, I wanted to end with just a thank you and to remind you that all of those facilities and the way that we run the program, it all is an expensive endeavor. And it's particularly expensive for the students, going to graduate school, they can sometimes get paid for their research, but oftentimes they're not. And so any support that you can give today will go directly to the students. I put the QR code up there, but it's also all around, and the purchases that you make just goes directly to the students because they are really the driving engine of Moss Landing Marine Labs. So I'm going to end there, and I'm happy to take any questions. Joe, go ahead.
The sea urchins, since they've eaten up all the algae there in the picture, will they produce a die-off of the urchins?
No, so there'll be a talk about urchins later, and my understanding is that urchins have the capacity to go a little like, what's the word I'm looking for? Sort of, what's that? Yeah, they go a little dormant and they shrink, and then they'll just wait for more food to show up, and then they go back.
And then how do you produce the iron to drop into the ocean? Are there pellets? Will it hurt the fish?
Will it hurt the fish? I'm gonna so Sarah Smith is giving a talk later, and I'm gonna let her answer that. I think that there are definitely with iron fertilization, there's definitely a lot of work in trying to understand what the ecological impacts would be, right? Because if you're doing that, you're definitely impacting the ecology. So I recently went to the Ocean Sciences meeting and I sat in on a bunch of these talks. I'm a paleoceanographer by training, so it's not what I focus on, but it was really interesting. That was definitely something that people are looking at. So not just, does it draw down CO2, but what are the subsequent consequences? So people are doing that in a cautious way. Yeah.
So how's the landscape on state, federal, and private funding for research? And then my follow-up question is, how's the impacted dissemination of the information from the scientists?
Yeah. This is a good question. The federal outlook on funding is not great. So the proposed budget that the president put out had some pretty massive cuts. There is an effort to try to not have that happen. But yeah, the outlook on the funding is not great. One of the things so the National Science Foundation has an ocean sciences directorate, and their budget has been cut. So it's not great. And this is one of the reasons why I highlight those really long-term monitoring programs, because those are the things that tend to get cut. And those things are so critical to helping understand change through time. There's just no other way to do some of this work. When we have satellite data, right? But you're just looking at the skin of the ocean when you're looking at satellite data. You really need these long-term monitoring projects to go out into the water, measure things at different depths. So the funding is a problem.
What about, I don't know if there's any other questions behind me. Oh, business partnerships?
But there is some of that. So I would encourage you to talk to the aquaculture people. There's definitely some of that as well. I saw one more question. Do I have time to take it, or no? I have no time to take it, but you can come and find me.
Okay.
Because I'm just going to be wandering about.
Okay, thank you.
Thank you.
All right, thank you so much, Petra. Sorry to cut off that last question, but we are moving right along. Kirsten is going to tell us a little bit about urchins and urchin ranching at Moss Landing.
All right, the question set me up really well. Right. Yeah, I can answer some of those urchin questions for sure. Everyone's moving around. How are you guys doing? I'm doing good. Happy to see you guys here. Thank you guys so much for coming to support us at Moss Landing Marine Laboratory Open House. We really appreciate it. My name is Kirsten. I'm a second-year master's student at Moss Landing Marine Lab, and I'm going to talk to you guys today a little bit about purple urchin ranching. So you guys are probably familiar with classic ranching, right? Where we've got our cowboys and our cowgirls on their horses. They're moving cattle around the open plains. And ranching is just one form of terrestrial livestock agriculture. And so they're raising animals like cows and pigs and chickens. And this is something that we can see on land, and it's something that we're very familiar with. And aquaculture really isn't so different. And so we're still raising animals for people to buy either as food or as pets or to put in their aquaria. But instead of terrestrial livestock, we're raising marine animals. And so there are a lot of similarities. And matter of fact, when I came to Moss Landing and they came to me with the project on purple urchin ranching, in my head, I had this picture on the left right here. I was like, absolutely, I'm definitely going to join this project. That sounds awesome. But as I continued working on it, it became very clear that it looked a lot more like the picture on the right, which is still really cool. And there's still a lot of similarities between agriculture and aquaculture. And especially for purple urchin ranching, we're still raising these animals typically for people to consume. And so let's get into the species a little bit because I haven't really introduced them yet. So what is a purple urchin? So a purple urchin is a species of marine invertebrate, and they're actually native to our California coast. And these guys are marine grazers. And so that means that their favorite thing to eat is kelp. And in a healthy ecosystem, typically that's not a problem. They'll hide away in their cracks and crevices in the rocks, and they'll wait for pieces of kelp to float by and they'll grab it. But in 2013 and 2014, our oceans actually experienced a marine heat wave, and this had a lot of negative effects on a lot of the marine species that we see on our own coast and in oceans around the world, right? And one of the species that was extremely affected was the sunflower star, and that's a really important major natural predator for purple urchins. And so the sunflower star populations, the oceans got warm, their populations declined very, very severely. We rarely see them in California anymore. And once that natural predator was gone, the urchins were like right, perfect. So their population just absolutely spiked, and we see so many purple urchins, and they're just very, very overabundant. And they are marine grazers, and with their numbers being so high, it led to overgrazing of our kelp forests and decimation. And we'll get more into what that looks like in a little bit. But I want to talk about their role in the culinary world too, because it's also really important. So in the culinary world, urchin uni is often considered a delicacy, and it's often put on things like sushi and other seafood dishes. And so purple urchins historically have not been used because they're a lot smaller than a lot of the other species that we have available, like our reds and our greens. And so they've been passed over. But right now we're in a situation where there are just so many urchins that it's creating an opportunity for fisheries and commercial divers and aquaculturists to work together to go out and collect these animals from these urchin barrens, which we'll talk about in a second. But they go out to the wild and collect these animals, and then we grow them in a captive setting. We feed them, we fatten them up over a series of maybe 2 or 3 months. And then we create our industry. And so it's got economic benefits, but it's also got environmental benefits. Oh, maybe I might play. Maybe. Oops. So sorry, guys. I think I messed that up. Anyways, you guys can see we have a nice photo of our kelp forest here. And like I said, kelp is one of the sea urchin's favorite food. And kelp is also a really, really important species of algae that grows our kelp forests along our Monterey Bay coast, right? And kelp forests are a really important habitat. They provide lots of food and shelter that creates area for lots of different marine species to thrive. So we've got lots of fish, we've got lots of invertebrates, we've got lots of algae. It's a very, very biodiverse habitat. And that not only affects the animals that are there, but it also draws humans there. It affects our human communities. People want to fish there and people want to dive there. And there's a lot of economic value that comes to people traveling to come see these kelp forests. With so many urchins, we're starting to see these change. The urchins there are a lot of them, and they're overgrazing, and they're turning our kelp forest into something that looks a lot more like. The suspense. Here's my clicker. There we go. It looks a lot more like these underwater deserts that we call urchin barrens. And so urchin barrens no longer have that natural resource of food and shelter for all these animals to live in. And so it becomes a lot less biodiverse. We lose a lot of the species, and really the major species that you're going to be seeing there are the urchins. And without all of the fish and all the algae and all the invertebrates, the fishermen don't want to fish there anymore. The divers don't want to dive there anymore. And so that's also affecting our human communities. And we're losing a lot of economic value that we had when we had the kelp forest there before. And so there are a few different projects at Moss Landing Marine Laboratories aquaculture facility that's focused on using urchin ranching as a solution to this problem of urchin barrens by going out and collecting urchins, growing them in captivity, creating industry to incentivize more divers to go out and collect more urchins until we can kind of start to reverse this purple urchin barren habitat back into those lush, beautiful, biodiverse kelp forests that we have seen before. So at Moss Landing Marine Laboratory, my focus on this, there's a lot of things that go into creating a plan for people to use to raise purple urchins. And one of the big questions is, well, what do we feed them? So they can grow efficiently and they can grow uni that is the largest and the highest quality and that people want to eat. And so typically urchins will eat kelp, but of course we don't want to go out and collect the kelp to feed them in captivity because we're trying to restore that kelp by growing them in a captive setting and taking them off of those barrens. And so one of the solutions that we had in mind was, well, let's feed them terrestrial food. What can we feed them that's going to be accessible, that's going to be practical, and that's going to be inexpensive for people to be able to do this on their own? And one of the things that we thought was, well, what about vegetable scraps? Monterey is a very, very agricultural community. There are lots of different farms. And so what I did for my project was I went and I connected with the different farmers from local farmers markets all over Monterey County and a little bit further past. And I was able to use their vegetables. So use local vegetables and local farmers to come up with a local solution to a local problem, which is a really cool kind of twist on the whole thing. And so for my study, I actually use carrots, I use artichokes, because artichoke capital of the world, right? I use spinach and I used romaine, and they ate all of it, right? Urchins will eat a lot, they'll eat anything, really, if it's soft enough. But what worked best was actually that spinach over there, and that it's created these uni that were just, the urchins were very full, and the uni was this beautiful orange color, very good texture. And the spinach was providing the amino acids and the proteins and the nutrients that were required for those urchins to grow well and efficiently. And it happened in a period of maybe 2 or 3 months, which is relatively short when you think of echinoderm or urchin aquaculture in general, right? That's very cool. We have a few other projects upcoming that we have a few other upcoming projects that are happening in the Moss Landing Marine Lab Aquaculture Lab. And so like, I'm still doing a series of feed studies that go beyond looking at vegetables. And so a more practical diet that we typically feed at a larger industry scale is going to be a pelleted diet, where we're putting in different ingredients and then feeding it as a pellet. And so I've just finished one study. I'm still looking at the data for that one. So I'm not sure what that all looks like, but I'll do that with different ingredients, with different protein levels or different starch levels or different fat levels to see what is the least expensive and the most practical, but also the most efficient diet to be able to grow these urchins in captivity and then sell them in the shortest period of time. We're also going to be looking at a finishing diet, and all that, what that looks like is it'll be your feed a pelleted diet for a certain period of time, and then you'll follow it with just a little short piece that's going to be something like kelp or something like fresh vegetables or something that's going to help to increase the quality of the uni that you might not get with the pelleted diet, right? So we're going to see how effective that is. There's also other aspects of urchin aquaculture that goes beyond what they eat, and that has to do with the amount of light that they get, because that can change their behavior, it can change their spawning activity, and that can also change the quality and the amount of the uni that you get after you ranch them for a certain period of time. And so these are all the projects that are going into urchin ranching to try to create a game plan for people who want to go out and collect urchins and have this available to them to do this. So we can get as many people as possible collecting urchins off of the barrens and trying to restore those kelp forests. So that's the future of what we have in Moss Landing. Thank you guys so much for coming to my talk. I hope that I was able to enlighten you with a little bit of urchin information, and I would love to hear questions from you guys if you guys have them. I have one in the back right here.
Thank you. So are you saying you want people to go outside and then bring them to some place to do aquaculture in a controlled environment since they're eating up all our kelp? And number two, when you're feeding the urchins, are you noticing any difference between feeding them organic scraps versus pesticide-laden items that we normally eat?
Yeah, that's a great question. So the question was, one, are we going out and collecting urchins to go take them somewhere for them to grow and then be sold? And I think the question to that, or the answer to that is, yes. I think what we'd like to do is we'd like to make it accessible so that people can do this as long as you have a source of salt water, whether it's recirculating or whether it's flow through. You can do this on your own. You have the instruction manual, so it kind of allows you to do it without having to rely on an aquaculture facility. But either way, yes, trying to get people to go out and collect them, grow them in a captive control setting, and then sell them to the consumers. And then your second question was, do we see a difference in the organic versus a not organic or a pesticide covered vegetable material? And I think that's a really good point. And that was something that I took into account when I got these vegetables. And a lot of them I was able to buy organic, which I was really grateful for. But the ones, I want to say it was the artichokes, I was not able to find organic. And so I was really, really careful about meticulously cutting them up, rinsing them all off, because if our pesticides kill bugs, what are they going to do to marine invertebrates, right? And so that'd be a great study to see, if you don't take the care of rinsing all those pesticides off, how does that affect the quality of the urchin that you have? I think that'd be really interesting to find out. Thank you. Yes, we have one more.
Yeah. Now I understand your theory of change. So let's say you are successful and people are going out and collecting the urchins. Let's just take a healthy system where there's a good balance of urchins.
Yeah.
Kelp. What if you over collect urchins? What's the flip side of that?
Yeah, that's a great question. That's one that I always think in my mind, and I always kind of ask people who have been doing this longer. It's a really great question. It's something that we don't know the answer to in a marine setting, because this is a new problem, and this is a new problem that we're trying to fix. But one thing I like to compare it to is I like to go back to I'm an ag student when I graduated from Cal Poly Slow to get my bachelor's. And so one thing that we see in the terrestrial environment is that we'll see once you take those grazers away, then the plants will start to overgrow and they'll start to get old and they'll lose their quality. And you lose a lot of the productivity of that land, just like you lose it from it being all eaten away. You allow them to grow to a point where you've got lots of weeds and low quality plants and things that you don't want anymore. And so I wonder, right, in my head, if a similar thing would happen in the oceans, because urchins are there for a reason and they serve an important purpose. But what is it, right? What does that mean if they go away, if it flips, if it's way too successful? Hopefully we won't have to see those consequences. We can see them before, but that's a great question though. I have a question here.
A little bit related to that, how much of an impact on all of the sea urchin populations that are out there do you think that ranching could make?
Yeah, that's a great.
There's a lot of sea urchins out there.
There's a ton of sea urchins. There's a lot of area to cover. And I think that this would have, like I said, our prior question about what if it, you know, is too successful and what if it flips everything? I think a more likely question is, are we even going to have enough support from our commercial divers and our fisheries to go out and collect enough to even make a difference? Because there are so many urchins. And for my projects, I'll go out and I'll collect 1,000. And that's not even a drop in the bucket, because there are a lot of urchins out there right now. And so and hopefully we can combine lots of different techniques, like putting back out, like trying to grow sunflower stars and put them back out and try to combine things, grow kelp again, you know, go out and outplant kelp. And those can all come together to help to solve this, to find a solution. You have a question?
I understand that this.
One more question and then we.
Sounds good. Thank you. Go ahead. Yes.
That the starfish are the natural predators of the urchins, right?
Yes.
And keep them in balance.
Yes.
And they're terribly prolific, right?
The urchins?
Urchins.
Yes. Yeah.
And why are you growing them then?
Yeah. So that's a great question. That's kind of one that it can be confusing. So what we'll do is out in the wild, right there, one of the questions before when Peter was talking was, how do they survive if there's no food available? And the urchins in the wild will actually, kind of like how we use our fat when we don't have anything to eat, they'll use their uni, and their uni is gonad tissue, right? And so they're not really, the reproduction doesn't happen as much in the wild, but when we collect them, we bring them into captivity and we don't reproduce them. We just grow the uni back out and we fatten them up so that there's something to sell in the commercial industry. Because if you have an urchin that doesn't have any uni left, like they are when they're starved in the wild, you're not going to be able to sell it for anything. And so to go out and collect them, bring them in and fatten them up or grow them that way, we can then sell them for people to eat. And so we're not making more urchins. We're just fattening up the ones that are there and then pulling them out of the ocean. Does that make sense?
Yeah, but is it creating
another? To stay on schedule.
Absolutely. I will be at the aquaculture booth in the courtyard if you're able to make it out there, and I'll talk to you more about it.
Sorry about that.
Yeah. Thank you, guys.
In this water bottle. Yes, thank you so much.
Okay.
Yeah.
I agree. Going to help.
Okay, so I'm gonna get started. My name is Tom Connolly. I'm on the faculty here at Moss Landing Marine Labs. I'm a physical oceanographer. And I study the ocean currents along the coast and estuaries and how they interact with marine ecosystems. And I'm going to be talking a little bit about tidal currents in Elkhorn Slough, specifically how they've been changing over time. And this is something that we have a long history of studying here at Moss Landing since we're so close to this important estuary. So just an overview of Elkhorn Slough, just to get you situated. We're located right about here at this blue dot. The Highway 1 bridge is right here, and the Moss Landing jetties are right here. And Elkhorn Slough is the estuary that is connected to the ocean through Moss Landing Harbor. So this is an estuary that we have small boats in Moss Landing Harbor. We have a lot of access, and a lot of students and faculty over the years have done research in this estuary. Just to give you a sense of how big it is, it's about 10 kilometers long. In the main channel, it's about 3 meters or 3 yards if you prefer, deep. But then it gets to be about 10 meters deep or about 30 feet deep, right near the Highway 1 bridge, which you drove over if you came from Santa Cruz. And then there are also large intertidal mudflats where it's much shallower and that would be exposed at low tide and very shallow at high tide. So we have tidal currents flowing in and out through the Moss Landing Harbor, through the jetties at Moss Landing. So that's creating currents going in and out of this estuary. But not all those tidal currents are able to flow freely. So there are places where the tides are artificially restricted, like here at Moro Coho Slough, which is right near Phil's Snack Shack, if you've stopped by there, or the Old Salinas River, which is right behind us here. There's an artificial tidal restriction there, and that's primarily in order to reduce the tides so that more land is available for agriculture. And one unique aspect of Elkhorn Slough and other California estuaries is that it's often saltier than the ocean in the summer because there's a lot of evaporation. So there's a lot of issues facing Elkhorn Slough, like many estuaries. There's excessive input of nutrients due to agricultural runoff, all the agriculture that we have around here. Nutrients stimulate algal blooms. And if that happens in an area where the tides can't flow, then all that algal biomass will sit there and decompose, and that can lead to poor water quality. And so this is an image from the Elkhorn Slough report card, and you can see all these red areas are places where the tides have been restricted, and that's where the report card is giving it more of a D or an F, not a passing grade. Another issue facing Elkhorn Slough is that the salt marsh has been eroding, and that's led to loss of habitat. And so Elkhorn Slough is world renowned for bird watching, and so a lot of that habitat for birds has been eroding away. This issue is linked to changes in land use, which I'll talk about, and alteration of the flows and pathways in the tidal currents. And so what we're working on is, as researchers, is trying to do science that we can use to assess restoration strategies to try to support or try to stop the erosion of salt marsh or at least slow it down. And this involves discussions with a lot of stakeholders with different perspectives on marine protected areas, agriculture, recreation, conservation of land, and the endangered species that are in the area. So to give you a perspective on how the Elkhorn Slough has changed over the years, these are some images that were pulled together by Andrea Woolfolk at the Elkhorn Slough Foundation, who's really looked into the history of the estuaries and their how they've been used over the years and how they've changed. This is a map from 1865 on the left here. And this is a map that doesn't show those tidal restrictions. And you can see that Elkhorn Slough was emptied out or connected to the ocean a little further north than where it is now. And there is a connection between Elkhorn Slough, Moro Coho Slough, Tembladero, and the Salinas River. And that's a connection that doesn't really exist today. Salinas River now connects to the ocean. When it's open, in the winter, it connects to the ocean further south, and there isn't that connection there. And this is a painting from the 1870s showing, illustrating that connection. So much more open channels than we see today. One major change that's happened to the estuaries is the construction of the harbor mouth. So the place where Elkhorn Slough is connected to the ocean now between the jetties, it didn't always connect to the ocean there. So it used to, in this aerial photograph, you can see the place where it connected a little further north. So this is the construction of the jetties here during the 1940s by the Army Corps of Engineers. This was the creation of Moss Landing Harbor as the port that we see today. But having this much deeper and wider entrance, and also the fact that it's aligned right with the main channel of Elkhorn Slough, this introduced a lot stronger tidal currents into the slough. So before it was narrower and shallower, the tidal currents coming in and out the slough were weaker. And this introduced a lot of
energy, tidal energy, into the system. And this is one factor that's led to a lot of the erosion here, is that now there's stronger tidal currents. The tidal currents were able to erode away a lot of that sediment, leading to that habitat loss. So it looks much different in the modern day. If we take the same perspective that we saw before, we have Elkhorn Slough, which is now directly connected to the ocean, but then we also have this lack of connections to these areas here. So a lot has changed in the hydrodynamics of this region. As I mentioned, Moss Landing has a rich history of studying changes in Elkhorn Slough. So we've been, we've been here for over 50 years, and researchers from different disciplines have been studying different aspects of Elkhorn Slough. And one thing that has come out of that long history of research is that measurements of the depth of the channel, if you just went to Highway 1 and measured the depth of the channel underneath Highway 1 or right next to it, it's gotten wider and deeper. So this gray line here, this would be if you're looking down the channel from the ocean. This is showing the gray line is 1972. And if you go to the 80s and 90s, that channel has gotten wider and it's also gotten deeper, so indicative of the erosion that's been happening. Another thing that's been observed is an increase in the tidal prism. And what the tidal prism is the amount of water that's stored between low tide and high tide. So as that water rises from low tide to high tide, water rushes in and then comes out on the ebb tides. And if there's more erosion, that creates more space for the tidal currents to flow in. So more water is stored in the estuary between low tide and high tide. And what's been noticed is this increase in the tidal prism, so indicating that there's more space for the water to flow into at high tide, and that's also indicative of erosion. So two ways of looking at the problem, which have been consistent in terms of the trends that have happened over the years. So these last measurements were done in the early 2000s. So what we've been doing in our group is collecting new measurements, and what we were expecting is to see this trend in the tidal prism increase. And spoiler alert, that's not quite what happens. So it's actually really similar to the previous measurements. So how we do this, one way we can do this is put instruments out in the channel. So this is on our boat, the Sheila B, which you can visit if you go out to the parking lot. We're deploying these instrumentation here, and this instrumentation uses sound to measure the speed and direction of the currents. And if you come to the Physical Oceanography Lab, walk all the way down the hall, we have some of this instrumentation and can explain how it works. But it uses sound to measure the currents. And these colors here indicating, the red is indicating water flowing into the slough, and the blue is indicating water flowing out of the slough. So we're able to use, put this instrument on the seafloor and measure the currents, and also has a pressure sensor to measure the water level moving up and down. So we see water rushing in as the water is rising. We can also use the same technology, but put it on a boat. And this is one of our students on one of our vessels. We can go across the channel and measure the currents going into and out of the slough and go across. And this allows us to get a full sweep of the amount of water that's rushing in different locations across the channel. So this is the type of data we can get. This is the over the course of a day. And we can see the tide height rising. So this is the tide gauge in Monterey showing the tide going up. And then by going out on the small boat, we can collect all these measurements of how much water, what's the volume of water going into the slough at each one of these time points. And by adding up all these measurements, we can get an estimate of the tidal prism. And as I mentioned before, we haven't seen that increase in the tidal prism that we were expecting. So we're still looking into explanations for that, but we're looking into how has the methodology changed over time. So different measurements will have different sources of uncertainty and bias. There have been some restoration projects like adding sediment to the slough and also trying to alter the flow pathways in a way that will reduce erosion while not allowing the water quality to deteriorate. And there's also theories that suggest maybe that the system should eventually stabilize, and we're using our data to test those theories to see if we're in that stable state now. Okay, and yeah, just looking at the data another way, as the water level is rising, we see water flowing in. As the water level is lowering, we see water flowing out. I just want to end by thanking all of you for visiting and showing an interest in our science. And I also want to thank my collaborators, and also students who helped with this research, helped analyze the data, and help deploy the equipment. Wouldn't be possible without a great team. So thank you. I'll be happy to take any questions.
Yes.
One back there, if there's one.
All the pesticides and they go into the water, causing the algae bloom. The thing is, I put perch in Elkhorn Slough. Bring them home and eat them. They tasted terrible thrown out because of all the pesticides and everything that are stuck in there. So is there any thought about putting more gates to slow the erosion, slow the tide, so we don't have all this negative input?
It's a complicated topic, so there is talk about that. It's a complicated topic because there's a trade-off between restricting the flow, which it could cause those pesticides to accumulate in some places if you restrict the flow instead of letting the water go in and out. And then, but there could be a benefit to slowing down the flow to reduce the erosion, and that erosion, if there's legacy contaminants in the sediment, that could be an issue, another erosion issue as well. So there is discussion of that, but it's a complicated topic with trade-offs that really need to be discussed by people with diverse perspectives.
Okay, we live close to the entrance of Elkhorn Slough, up there. And how does the flow of the estuary, how does it affect the sea otters? Because they're eating all day long.
Yeah, so another thing that we've had to take into account when discussing these possible solutions to flow restrictions that we weren't expecting to have to think about was the tide gate in Morocojo Slough. The otter, there was an otter that would swim around right near the snack shack at the entrance, and the tide gate opens up during low tides. So that otter would go into Morocojo Slough when the tide gate was open, and then the tide gate would shut at high tide, and then that otter would be trapped there. And so it'd end up having to cross the road, which is really dangerous. So that's why you see otter crossings there. And so that's actually something we've had to account for in the size of the flow restriction. Can an otter fit through there? Yeah.
Okay, one more question.
I think we have to.
Yeah, it's time to
go.
switch, try to stay on schedule. Try to find Tom
Yeah,
down.
I'll be all the way down the hall.
Yeah. Okay, so, thank you, Mom.
Okay. Good morning, everyone. I was your parking guy. I did a terrible job parking, and now I'm going to pretend to be a professor at Mount Landing. My name is Ivan Aiello, and I'm a geologist here. And I have an idea to start the PowerPoint presentation. You know where my presentation is? Is it just click? Click, click. Okay. Also, I don't have glasses. No, I didn't do anything else. Also, I don't have glasses. Okay, so yeah, so today I'd like to talk to you about the Monterey Canyon of California, which is conveniently located next to our lab, and is an amazing feature which has puzzled scientists and everyone alike for many years. And so I'm going to give you some potential interpretation as how the Monterey Canyon formed, and actually some really cool stuff how it actually was first discovered. whenever technology is gonna catch up with us. And if it doesn't, I can also perform different, dance moves. I have my. That's me, when I started my tenure on Moss Landing. That actually is familiar. Looks like a Tuscan countryside, mostly.
Yeah. Yeah, I'll go see if I can find someone that knows where it is.
Oh,
Well, they scheduled him for 25 minutes, so we have a little buffer.
yeah. Yeah, so let's start. How many people know that we are next to a submarine canyon here? Oh,
Yeah.
so the surprise effect is not part of my thing. Okay, so how many people, does anyone know how it formed? How come we have an underwater canyon here? Actually, it would be good to, does anyone know or hasn't?
Rough idea. Rough idea is, the sea level was much, much lower in the geologically recent past, and the Salinas River outflows cut the canyon, much like happened in Colorado River.
Okay. Yeah, that's one of the main hypotheses. However, as you'll see, there's a little problem with that, which is it supports the formation of part of the canyon, the shallow part, but the canyon is very, very deep. It goes down to more than a mile. And so sea level change that occurred during the ice ages is only in meters, only 120 meters. So that will explain how you can have such a huge feature forming offshore in the deep sea. So that's part of the story. So how can you have a canyon that, on a seafloor, which is never exposed to river action? What can make
Seismic activity.
seismic activity? Landslides, seismic activity, underwater landslides. Underwater lake, because everything.
Did it just shut down?
Was that?
Coastal subsidence, like it was at a higher elevation.
Subside, yeah, the whole thing is subsiding, is a continental margin, but earthquake activity in faults.
Right.
Because of the San Andreas fault system, this is a very faulted area, everywhere you look.
So you want to use the clicker, the clicker's here.
And food for phantasm, that's my talk. Wonderful. And, oh yeah, all right, thank you, sir.
Yeah.
Technology, yeah. I do advance here too, I have no.
Yeah.
This one
There you
here.
go, that's the one.
Okay.
Give it a test.
Okay, so, yeah, so we are not, okay, I'm gonna use arrows because I have no idea how to use this thing. So we are here. So this is a Google map. You can go online and you can go online and you're gonna see that the seafloor is characterized by this deep feature that indeed is part of the Monterey Canyon system. And actually the system has different branches. There's a main, shoot. Okay, so there it is. You should be here, it means that if you want to be a marine scientist, that's what you would be, right? But so the Monterey Canyon has different branches. The main one is the Monterey Canyon per se, but then there's also Carrillo Canyon, Ascension Canyon, Soquel, Carmel Canyon, which actually goes the other way, if you notice that, it emerges in Monastery Beach. So really interesting topography. And those are some, and if you have any question, please ask. And those are some of the numbers we were talking about. So it meanders for about 300 miles. And it can be up to, almost 8 miles wide at its widest point. And you can reach a depth of almost 6,000 feet before it ends in the abyssal plain. And it makes this really cool bend that we call the Shepherd's Meander. And we'll talk about that. Okay, so Mbari, our friends next door, made a really cool video. I'm going to show you just part of it. Just have this feeling that you're looking out there and you see the water, and all of a sudden this feature is going to emerge. And it's like being at the edge of the Grand Canyon, really. So you look down, Mbari has a really cool video production, and they also are rich friends. Those are all multi-beam data, so they're actually being collected by AUVs, autonomous operated vehicles and ships. And you can just travel through the canyon, and it gets deeper and deeper up to a mile deep. And then eventually, you see the detail of the seafloor. I mean, it's amazing. It's like really amazing, the work that we can do out there. And then eventually ends up, as I mentioned, in the abyssal plain, it is making this large meander here, which is called Shepherd Meander, down to 4,000 meters. So sea level ranged only, as I said, about 120 meters during ice ages. So this is some of the work that we're doing here at Moss Landing. This is actually my student, Marcel Felix. So we got there with our small boats. You saw one parked up there in the parking lot. That the heart of our operation, marine operation, is essential for us because our students and researchers alike, we can go out there and actually map the canyon after a storm. If there's any event that is significant, we can go out there and produce really high resolution maps of the canyon, see how it changes over time. And indeed, we find that this is highly influenced by what happens on land, especially on the beach. So there is a connection between the head of the canyon and the coastal system. But how did it form? That was our conversation earlier. So this is really cool imagery because it tells you how incredible has been the exploration of the underwater habitats here. So the first map of the Monterey Canyon, the Monterey Submerged Valley, was made by George Davison, who was an incredible pioneer. He actually, they named different features around the country in his honor because it was one of the most successful mappers. He mapped for the first time much of coastal California back in the late 19th century. The Davison Seamount, the first ever identified seamount, was actually called after him. And so using lead lines, so they were dropping long ropes with weight and then measuring the distance, they were able to map for the first time the fact that it was that, deep features out there. And everything using triangulation, no GPS, no Google, where am I? Things like that, right? But look at the map, and it's really good. You can see the Soquel Canyon, you can see the head of the Monterey Canyon. And the father of marine geology, Francis Shepard, who was a professor at Scripps, actually continued the high resolution mapping of the canyon. So you can see here the meander was talked about, which is we call the Shepard meander. And he came with this hypothesis that the submarine canyons were carved by rivers because they look like rivers. They make meanders, they go up and down. And also he compared the size and the extent of the Monterey Canyon with the Grand Canyon of California. Well, not California, Grand Canyon. And they look pretty much alike. Same depth, same slopes. So he thought, well, it's like the Grand Canyon. But here's the problem, as we discussed, is that I forgot what I did. Okay. So if you look at the Monterey Bay today, and you go back, say, during the last glacial maximum, so 80,000 years ago. So here the four main rivers that empty in the bay, the Salinas River, is Alphonse Loop, which used to be a river, Pajaro River, and Sunrise River up there. If you go back on time and you drop sea level using very special effects, there you go. The coastline, 18,000 years ago, that's yesterday. There were people living out here. It was all the way down to the shelf down there. So the head of the canyon was further offshore. And the rivers, as you said, were empty at the head of the canyon. So it does make sense that the head of the canyon was actually carved by a river over and over during glacial interglacial sea level changes. However, the problem is that there's a whole deepest part here, which cannot be explained by sea level change, right? So, and then we go, please.
The same quandary that they had with the Columbia River gorge with the floods from Missoula, Montana, could have been an ice age flood that caused the extreme, a dam across the.
Man, that's a good question. I do not know. No, I do not know. So, yeah.
That fooled all the scientists until this one engineer said, Missoula flood is what carved it. It wasn't the Columbia.
Right. No, I know about that. I don't know what actually triggered it. Yeah, yeah. Let's talk about that. You and I, in my lab, secretly. So, but let me show you something. If you look at the San Andreas Fault system, right? The topography, the shape, the relief around here is highly influenced by it, especially if you go down to the desert. If you have been to the Wallace Creek down near San Luis Obispo, it's fantastic because you can walk in the fault and you can see offset streams. I mean, things are moving there so fast, like an inch a year, and streams get decapitated, really. And so the topography, ridges and streams is highly influenced by the San Andreas Fault system. Then if you project that to the Monterey Bay and you start looking at geometry of some of those I don't know what the plane is doing here. It's not supposed to be here. If you start looking at the geometry of some of the canyons features, they all offset by similar fault system. In fact, you can actually directly plot the faults out there. We know where they are because of seismic activity with some of the features of the canyon. And then if you take a step back, and this is actually my last slide, so I think I'm being efficient here. And if you look at geophysical data from the Monterey Bay, so now we are looking at something that's a little more complicated. This is a gravity anomaly map. So basically the red shows where you have more rocks and the blue shows where you have less rocks. This is because the crust of the planet is kind of bumpy, and it's bumpy when you have a lot of faults. So what you see here with this black line are indeed offshore strands, faults of the San Andreas Fault system. This is the main fault, which is going through the King City. Sorry, that up there is the main fault, which is going through the Santa Cruz Mountains. Then you have another fault system, an offshore fault system, which actually parallels the Carmel Canyon. And then you keep, as you go offshore, you find more and more strands, parts of the San Andreas Fault system, and also cross faults, which are elements that join the different faults. Things are kind of being branched and broken up in pieces. And when you plot the faults and the canyons out there, using again a very fancy graphics, you see how they actually follow those faults and cross faults. So they bend whenever two faults are crossing, and they eventually rectify when you have another fault system. So there's evidence that probably the deepest part of the canyon was, but it's not the fault that's making the canyon. What it's doing is probably grinding the rocks, so they are more susceptible to being eroded by underwater current, by landslides, submarine landslides, and so on. We actually see the submarine landslides, so there's good proof of that. So that's kind of, so in conclusion, I have no idea the time, but
You have lots of time.
oh, let's slow down.
Technical issues that we'll use the time to fix.
Sure. All right. So scuba. So here's what we learned, I think, that first of all, there's an amazing feature out there, which for us, it's really a great opportunity to do marine science because we have deep water right next door. So we can, and not just geology, I'm talking about all the science here. That the Monterey Canyon was first surveyed at the end of the 19th century using lead lines and sextants and very simple but yet powerful topographic tools. That the head of the canyon, so the canyon is, I call it the Frankenstein Canyon, because the head is actually created by sea level change, right? But the deepest parts are probably related to structural features that enhance erosion, underwater erosion. There's actually bioerosion. There are theories out there. It's really cool. So, but essentially the faults create those weakness points that eventually underwater current and gravity flows can carve, and then you get this really deep canyon out there. Okay, thank you. Yeah. And now it's time for questions even.
How significant are the currents out there?
Well, Mbari, again, this is the Monterey Bay Aquarium Research Institute, that has an incredible program of underwater observation, including smart boulders. There are boulders, there are things that show boulders that pretend to be boulders, but actually document where they tumble down the canyon, so they can be retrieved and monitored. So, and they did monitor some very large submarine landslides. So the sediments accumulate at the canyon's head and get essentially, well, today, let's go to today, it's better. 18,000 years ago, there we go. So, just think about all the sediment transfer that occur in the coastline here, each sediment itself. Much of it ends up at the head of the canyon. It kind of collects all this stuff, and then it gets big storms or even seismic events, and then eventually, boom, it gets flushed down the canyon. And it moves rapidly all the way to the deepest part. So that, it's called a gravity-driven current. It's a gravity flow. Because it's not a current like in the river. Actually, the sediments are moving or creating a current. And that's like a very powerful original character.
Isn't freshwater lighter than saltwater though?
Is it?
I don't know, that's what I'm asking. I thought it was, but then I guess that got rocks and sediment in it. I guess that would make it heavier, but.
So the sediments, when sediments mix with water, they make really high density currents.
Right, okay.
In fact, they're called high density fluids. That's what they're called. And actually, because of the density contrast between, so you might imagine, you have a bunch of sediments out there, you just push them down the slope and they will disperse. They don't do that actually. They stay pretty much lumped together because of the density contrast between this body of sediments and water that's moving and the surrounding water. So they can travel hundreds of miles offshore. Those are called turbidity flows.
And it's like a barrier between the two.
Yeah,
Water.
yeah. I mean, the whole east coast of the United States and many other the continental rise is shaped, occurs because of that, because of these flows of sediments and water. And they have a high erosional power. So they can carve, trenches underwater.
Any way to check if it was formed terrestrially and then went submarine because of tectonic activity?
So the subsidence that, yeah. So this is a really good question and it's really possible. And let me tell you one last part of this, because this, it gets complicated. And the reason why is, you can also ask me another question. Can you? Thank you. How old is the canyon? How old is the canyon? How long has it been there? So, and talking about the Frankenstein concept here. So if you, and I'll finish whenever they wait on me. So many of you probably know that the San Andreas fault system is a system where there's two plates, right? The earth is made of hard things that move around, which are called plates. And then the Pacific plate is moving relative to the north, relative to the North American plate. So this stuff here is going to the north and this stuff here is going to the south. And it's going fast. 1 inch a year is fast. You can see it in your lifetime. If you've been to Hollister, you can see sidewalks being offset. I mean, it's not something that you have to wait, a couple million years. It happens really fast. So 1 inch a year multiplied by 1,000 years is already like meters. Million years, it's kilometers. So if you think about that, this part here is moving to the north relative to this part here, right? This is moving relative to the north to this part here. So this is moving way faster relative to the north. And if you go further offshore, this part here is moving even more. So stuff is moving fast. This used to be all the way down the side of California, maybe 20 million years ago. So it's possible that part of those canyons were indeed the head of the Monterey Canyon today. So they were, emerged, carved by river, subsided, and then eventually eroded underwater. So the answer is yes.
Is subduction over?
Subduction is over. Yeah. So subduction is the Pacific plate used to subduct underneath the North American plate. There's still a remnant here. That's this dashed line. Thank you for the question. This is actually Pacific crust. This is North America. We are in between. This is not, we're not in North America, we're not in the Pacific plate. We're actually something in between, all this broken up stuff. Yeah. So be careful.
Then to answer the question about the age, it varies depending upon where you're pulling it from. Not to mention that center part's got to be really interesting because it's mixing a lot of different stuff together.
Yeah, and it's rotating too. If you go to the transverse range in Southern California, there's this mountain range all of a sudden just crosses. That's because it rotated. So probably some of these blocks here actually being also rotating. We know that about Año Nuevo. Año Nuevo is rotated about 60 degrees in the last 3 million years. Used to be, because we have paleo-magnetic data, we can actually see how it moved over time and rotated. It's pretty cool. Yeah, it's, again, if you take your shirt and you do something like this, you'll see the stuff in between rotates. I mean, it's very basic physics, right? So, yeah, I should publish it one day.
All right, yeah,
Okay.
they had made a mistake on the scheduling, so we had a little bit of extra time, but in this, so Roxy is next, and she can do one. Yeah.
No.
But I know you have 4 more.
So I really love parking, so I miss that and then that. It's finally, I found my, talent. I mean, it's such a terrible job. It's like everything's bent and. All right. My name is Ivano Aiello, and I teach here, and I'm the only Italian called Ivano in the whole Monterey, probably the world. So, yeah, but please come to my lab, geological oceanography. We have microscopes, we scan electron microscope. We have a really cool SCM because we look at sediments in the ocean, not just faults. So it's really cool stuff. So come visit us. Okay, thank you so much.
What?
Thanks for fixing it. You just said to just hang it, don't. You just said to don't wander from your.
Well, you can, because again, the live stream's picking up the presentation. Yeah, so yeah, it doesn't really matter, yeah.
How are you? Yeah, I didn't do that. Everything's
I have no idea what.
It's good, yeah, we just don't want to touch it, okay, just kind of run the presentation, the slides, but anything else like exiting the presentation, that's gonna trigger the,
Okay,
yeah.
yeah, that was, we had no idea. What happened. I'm sure Amani will really find that you today. Just don't touch the computer. Okay.
And it's just. Okay, that's the.
And yeah, touch it out, make sure.
So you have to point here, that's the, I wanted to point at the screen. Yeah. Yeah.
Yeah, you can actually.
Well, usually like this is where the dongle. And so it's a very common thing to point there.
Okay.
Oh, really? Yeah, you stand here.
Okay, cool.
Nice, good start. I guess it's fine.
We'll wait one more minute to make sure everyone can get here who wants to come and learn all about scuba. Then Roxy will tell us about, yeah, Scuba 101.
But I don't need the notes singly, so. Okay. Yeah, I'm gonna get started. Before I'll introduce myself first. So my name is Roxanne. I'm a third year here at Moss Landing Marine Lab. I am in the psychology lab. And kind of to give you background on what I do, we'll talk about a little bit more. But I do have a scuba thesis. And so I've been in the field trying to get my project going and collecting most of my data. And then I started diving in 2017, mostly recreationally. And then I worked at the National Aquarium in Baltimore for 3 years. And I dove a lot in the tanks there. Before I keep going, does anyone a diver here? Okay, we got a couple divers. Okay, cool. Thank you. In the beginning of this, I'll move on to the next. Sorry, Gita. What am I doing? This one or this one?
It should be this one and maybe point at the double. Okay, it's not working. He said not to touch this.
He did say not to touch me. Guess it's not. Okay, sorry. How about it? We're just all scared to touch. Yeah, I know. I'm so about to touch it. No, okay. Okay, cool. So I'm going to talk about a little what is scuba for those people that maybe not familiar, how it works for the most part. I think that's one of the most common questions I get when I'm talking to like family or friends or anything like that. And then I'll kind of talk a little bit about like what we do here and kind of aside from student thesis, what other people do as far as like scientific divers. And then there should be a little bit of time for questions at the end, and I have some videos to share. Okay, so scuba stands for self-contained underwater breathing apparatus. And so kind of in that description, self-contained breathing underwater apparatus, you need to be able to breathe while you're underwater. So the thing that I'm wearing right now, there's another picture of that here. And so this back thing here will inflate, and that's kind of what helps you float. And I'll go into a little bit like why that's important in the next couple slides. Other things on here, you can see there are a lot of little danglies. And so as scientific divers, we have to carry everything in, make sure we don't lose it with the swells, with the waves or anything like that. And so I think there's a whistle on here and a place for a knife, a light. This is not mine, but we mostly carry about the same thing. There is a little bit of variation in personal preference as to what this looks like, and it's called a BCD. Sometimes it's kind of also referred to like as a vest.
And this is what helps me. And then this hose right here is the inflator hose. There's a button on here that allows me to add air to the valve or the little bag that's up here that's going to turn into essentially a balloon. You can think about it. And then the top allows me to take out some air from that. And then there's another hose which comes from, let me grab this. So the other thing that you really need is your regulator. And so this has 4 different hoses. This top part will connect to the tank. And then from here you have different, so there are 2 of these. These are where you're going to actually breathe out of. There are 2 because there's one for your buddy and in case of an emergency, which is the yellow one. That's why it's bright. That way they can see it and grab it immediately. And then the other one is what you breathe out of. And there's a mechanism in both of these that essentially as you're inhaling, it can sense that change in pressure and you breathe in. And then as you exhale, that's why you see all the bubbles and you see scuba divers. And then the other hoses here vary a little bit depending on the person. For example, this one here in the photo, which I'm not supposed to wander too much, but there's also, it has a pressure gauge on it as well. So you can see how much air is in your tank. And then the other one is the one that connects to this hose here that allows me to put air into my BC. And then other modifications that some people might have onto where the pressure gauge is, sometimes they'll have a compass attached to it, if not clipped onto one of these D-rings. And then on occasion, there's also a dive computer. So that's actually not listed on here, but the dive computer is really important. It can give you depth and it also gives you how long you've been underwater. And then for people that might be doing a little bit more complicated dive, that's really important to make sure you're not staying under for too long. For me, mine's just like a watch. And so I'll take this with me and dive with it. But some people have it clipped onto like this set. Another modification, which I think is on here as well, is you can have a little reader. And so I think this is my friend Jesse's. And so on his dive computer, this is a transmitter out to his computer and it'll let him know how much air is. And that's really helpful because then you're not looking at 5 different things to figure out, like, should you be down there? Do you need to leave? Or just kind of like figuring that out. And it's just one screen to look at everything. And then of course you need your cylinder that has air in it. And then other things, I have a couple of fins down here that help you swim. As well as your mask so you can see, and then weights that help balance you down, which I'll explain a little bit later, and a wetsuit to keep you warm, which is, there's an asterisk there because it's a little bit optional depending on where you're diving. Let's go to the next slide. Okay, so how does it work? So your BCD, as I mentioned, in place. I think I like to compare this analogy to imagine if you were throwing a rock in the ocean, it's going to sink immediately. You don't want that because you don't want to sink to the bottom. And so your BCD is a little bit like a balloon attached to that. So it's going to help you float. And so depending on what you want to do or what you're working in, you're going to want to have more or less. And then as you go deeper, there's more pressure. And so your balloon is going to compress a little bit more. So you might have to add more air into your BC. So if you're hanging out near the surface, you're probably not going to do that many adjustments. But as you're getting down, you're going to add more. And then if you go up, you might want to let more air out because if you're too buoyant, you're just going to float to the surface. And that's what's dangerous and not what you want to do. So if you're too heavy, you're going to sink, and then too buoyant, you're going to float. And so while you have all of your gear ready, I didn't bring a tank out here, but there is one at the dive booth if you all want to try it on as well. But you have your gear ready, you're checking things that you're checking while you're diving, or as I mentioned, your air, and then looking at the depth and where you are, your surroundings, and also checking on your buddy to make sure they're okay. And we have the other thing that you can see here, this is our diver that has everything ready, so she has a pen and pencil in her hand. And another question that happens really often is like, we're diving, we're taking data, like how are you taking data? And so you can do that through videos, which is really great because you can look back on it and see, like do your survey that way if you're looking at fish or other animals or anything else. But the most common way is to have a data sheet with like waterproof paper, and then you have a dive slate, which I didn't bring with me, but they do have it at the dive table if you want to practice, and then you just write on it with a pencil. And so that's what she has there, and it's really convenient even if you're not taking data because you can write notes to your buddy. If you have a question, like should we go this way? Did you see that? Like anything like that. Do you want to go up? And so that's like a really fun other way of keeping track of that. And let's see. Let's go on a dive. Okay, well, I guess we're gonna skip that. In case you were interested in being a diver, so we have down here. Just kind of like do a little bit of an overview for that. You're first going to want to find an instructor. There are a lot of dive shops in Monterey, but also in other places. You do not have to be by the ocean to find a dive shop, though it is more rare. A lot of places will have a pool and you do some of the training in there.
The next thing that's an optional thing that I included here, because there is an item in the auction to bid on this, I believe. And so Discover Scuba course is a quick, I think it's a half-day thing where you go to a pool and you can try it with an instructor, and if you can try it out, and you don't have to do all the other training. And then every step after that point really varies on how much of a trained diver that you want to be. This first open water certification involves a swim test, different skills tests in the water, first in the pool and then in the ocean, and then different lessons and exams. And I think like another question that I get a lot is like, do I have to be a strong swimmer? And no, you don't, but you do need to pass the swim test. And I think it is just helpful to be a swimmer, especially if you have to kick different places. A lot of people do boat dives where you just kind of get on the boat and then you plop in and then you just get kind of hang out, and those are really easy and fun. And so I would say that those are somewhat less strenuous, but if you're doing a shore dive, you might want to just be a stronger swimmer. But you do not have to be. And then from there on, there are different specialties you can do. I listed a couple down here. So being a scientific diver, commercial diver, or other like technical divers, like if you want to go in caves, requires more training, in part because it's a little more dangerous and you need to be ready to prepare for that. And let me see, everyone here at Moss Landing, which I'll talk about scientific divers in a second, is a master diver. And then from there, we also do a scientific diver course. So everyone's under that, just under an instructor, although we do have some students that are instructors in addition to our dive safety officers. And then I keep mentioning this location dependent. I think depending on what you want to do and where you are, you might want to get trained differently. And I think that just getting practice and being safe is really what the goal is for all of these things, and understanding how your equipment works in case something perhaps doesn't look right, and you can be ready to either perhaps it's not a good day to dive, which is totally fine, or it's fine and you can get an adjustment for that and knowing if you can fix it. So what do we do here? Let's hope this plays. Okay, this is a short little video. And so like I mentioned, we're all scientific divers here at Moss, and most research institutions have them. And you can see there's another diver here that has that dive slate. That's the white square. And then they're also holding a Transect tape. That's really useful when you're doing different surveys. And what you'll do is set down the Transect tape and then kind of go back on it and use that as a reference for if you're doing any like specific surveys or samples. You can kind of look back if you're interested in doing that. But it really just depends on what your study species is. I study kelp, and so I might be doing something different than like any other of the fish scientists, for example. Let me see. Okay, so as far as Moss, so we have two scientific diver courses. The first one takes place every fall, and it's led by Dr. Diana Steller, who's one of our research faculty, and she's also a former Moss student. She got her master's here on Rhodolith, and so it's done in the fall, and it's an introductory course, and so they walk you through everything through master diver, all your scientific diver skills, which I think it's all, I'm going to talk about it now. The course, I think what I got from it is you learn how to task load. And so it's uncomfortable to be underwater as a new diver, and it can be uncomfortable later on, or if you haven't done it in a while. And then you're adding on to that, if it's swelly, if there are a lot of waves, if you're going to be cold, perhaps. And so kind of being accustomed to doing all of that. And then on top of that, you need to get data, and you want to get good data, and you're also limited on time. And so those were all things that I had to learn as I was doing my thesis project, as well as like all of the class learns to do in a preliminary way. And then the Springfield course, which varies in location every year. Jessica Franks is doing a talk right after lunch. So if you're interested in learning more about it, however, it's kind of a way to execute those skills that you learned. So you're a scientific diver now, like how would you handle doing a project? This year I was able to go with them. We just came back from being on an island in Baja California, and it was really great to see the students kind of pick up on the different things that they couldn't get access to, but had to make adjustments to with their projects, and like learning how to dive out there as well. Let's see, and I talked a little bit about this. Let's see. Yeah. And so one way that we have a lot of divers here is doing student theses. Some of the main study species are giant kelp, like myself. We also have some bull kelp studies and Stephanocystis, which is chain bladder kelp as well. And sometimes we'll go out. So most of these pictures, except for the top one in the middle, are at Stillwater Cove and Pebble Beach. And so another component that I didn't include a lot of photos of this is we do take the boat out. It's a little hard. We want to get a little further offshore. And so sometimes we have an inflatable boat. We carry it down the stairs if you've been to Stillwater at all. And it's fun and it's heavy and it's okay. But then you get out in the water and we have the ability to do our diving and collect some information for that. These photos are
I had a couple of photos here, but another project that's funded not through as a thesis project, but also for restoration and through a couple organizations and other through Cal Poly Humboldt as well. The Nature Conservancy is also on this project, and two of our professors are as well, is doing a bull kelp restoration project. They've been going out the last couple of years working on that, and kind of in conjunction, though not through the same project, we also have a student doing drone imagery. And I kind of, with this slide, sorry for the photos, I wanted to highlight that their diving kind of fits into this model as we're getting more technology to do these studies. Is you can have satellite images and you can have drone studies, but it's also really important to kind of see what's in there. And so kind of using both in order to make a good study and to see how these communities are changing is really important. Let me see. Okay, and then let's say you wanted to get involved, and should you, why should you, and perhaps should you? And so research is one way of doing that. There's also Reef Check is a citizen science-led nonprofit organization that does different studies across the coast, but also in coral reef areas. So they are a global organization, although we most likely have them with their West Coast Reef Check here. And they do studies, and it's all volunteer-based. They do really great stuff, like in Monterey, you get access to their boats and help them out, and you learn how to be a scientific diver with them, getting involved. So you do have to be certified to help them out, but that is one way to get involved nearby. Another reason, so this picture here, I think we lovingly call this type of image the Kelp Cathedral. And I think it's why I fell in love with kelp forests, and kind of you can see the light coming through the canopy, and it's just very beautiful. And then when it's a nice calm day, you're at peace. And I think I compare it to kind of going into a hike in the redwoods, because that's what it feels like, except you can fly through them because you're in the water, and you can also kind of look at different crevices, look for other animals, or just kind of hang out. And I think that this feeling is just kind of what really encompasses it for me. And when I talk to some of my peers, it's the same feeling. So it is very beautiful. But kind of carrying on, it can be hobby. A lot of people, this one's gonna work, cool. A lot of people do photography or different art. You can also do videos and share these with family and friends to let them know what's out there. It is hard if you are not a diver to kind of get close up to this, aside from perhaps at an aquarium. And so that's another way to get involved. But should you? I think that a lot of people sometimes talk about wanting to get involved in marine science, but oh, I'm not a diver, or I don't like swimming. And as much, I don't mean this to be an advertisement to diving as much as I love it so much, but I don't think you have to be. And especially when I'm talking for people like undergrads or other students that want to get involved, I don't want them to think that, because it's not true. Oh no, I had one more slide, but I can just chat about it. I really want to emphasize that there are a lot of other ways to get involved in marine science. If you just love the ocean, like there's engineering, programming, you can do art, you can do teaching. There's so many ways that you do not have to dive, and you can still do field work and not dive. And so I kind of want to leave you with that. Yeah, I can take questions for a little while. No questions, I'm so sorry, but I would be happy to talk with you. I saw that, I'm so sorry. Is that okay? Okay, sorry, yes, please go ahead.
All right, you had mentioned that going up to the surface really quickly was not safe. Can you elaborate a little?
Yeah, of course, yeah. So what happens when you're diving is, so everything is compressed and all the gases in you are also compressed. And so if you go up too quickly, they're going to expand quickly. And so that's kind of where you have the bends, which you might have heard of. And that if you have those bubbles, those gas bubbles, which is nitrogen, going into your brain or another part of your body, expanding, that's where you can get a stroke or something else. And so and that's why if you go up slowly, they're going to expand very slowly and be able to dissipate a lot quickly. It's a really good question. Yeah.
Thank you.
Anyone? I'm not getting yelled at. So anyone else?
You have to exhale.
Yes. Yeah. And Connor was making a good point. You also, another thing that you're taught when you're learning how to dive is always to be exhaling or breathing in, because it helps keep that consistent pressure in your body with gases. Yeah. And don't hold your breath. Yeah. Any other, there is also, like I mentioned, the dive booth outside. You can try on a BC if you want to, but thank you so much for hanging out. Yeah.
I'm sorry.
Okay, hi everyone. If you're here for trivia, we're gonna get started in just a moment. If you would like to compete in trivia, come up to this table and grab one of the trivia sheets and a pen. Yeah, we're gonna have 3 different rounds. So you can grab 3 sheets, or you can write on the back of the sheet, whatever you decide. And it's just gonna be a fun little interactive activity. There are prizes for the winning team. So
So it's a team thing?
You can do teams, or if you want to ditch your whole team and be an individual, better chance of getting the single prize for yourself. Feel free to do that also. So yeah, this is the format. You can fill out the sheet with your team or individually.
I don't want to make any students. We all win.
All right, we're gonna wait just a few minutes and then we will get started. I'm gonna. But yeah, we've got 3 rounds going on, 3 opportunities to win.
Did you want to go? Okay.
Over the PA.
Go ahead and put it back on.
All right, everyone, we're gonna get started with trivia. I hope you have your good thinking caps on. Let's get started with our first round. Be ready to write. So our first round is local flora and fauna. So things that you might see in and around the Moss Landing area, maybe things that people here at Moss Landing Marine Lab study. So first of all, this is a multiple choice question, A or B. Which one of these animals is a sea otter? Give you all a few moments to write that one down.
Chris and Bailey.
I don't know what.
Like, I can trick question. Yeah.
Okay, we're gonna move on to the next one, also multiple choice question. How old was the oldest aged rockfish? We have an option of 57 years old. That's still pretty old for a fish. 139 years old, 205 years old, or 261 years old. So you can write the letter or you can write the number of years, whichever you prefer. You're just gonna swap with a friend after this and grade each other's sheets. That is a great question. I don't know who it was, but the way that they find out is rockfishes have otoliths, which are their ear bones, and kind of like rings of trees, like different layers grow for each year that they're alive. So by looking carefully at the otoliths, you can tell how old the rockfish is. And that works for most fish, actually. It's really cool. Okay, this one is a free response question, not multiple choice. What is the general name of the type of plant-like plankton that generates around half of all of Earth's oxygen? So the hint is that we're looking for one word ending in plankton. Hi, come on in. Feel free to grab a sheet and a pen. Okay, so we're looking for the general name of plant-like plankton, which generates around half of all of Earth's oxygen. It's not all trees like we sometimes think. Okay, this is a true or false question. You can write on your sheet, true or false. You can find corals living in Monterey Bay. Okay. The next one is another identification question. So which one of these algae is giant kelp? So just to be clear, this one and this one are the same thing, and then those two are the same. It's just a close-up of the leaves or of those parts of their body. You can write A or B on your sheet. As a hint, one of these is giant kelp and one of these is bull kelp. We just have to know which one of them is giant kelp. But yeah, we got a nice close-up images of their blades to hopefully help you with your ID. Both of these live here in the Monterey Bay area. Okay, question number 6. What do juvenile white sharks eat? So juvenile white sharks can actually be found in this area. There's a talk on white shark digestion a little later on today. So for right now, just think about what you think they might eat: otters, fish, seals, or crustaceans. And it's not a select multiple. One of these is the correct answer. Moving along to the next question. Which 2 of these animals are most closely related? We have a sunflower star, that's a Pycnopodia. B is an apple anemone, and C is me, a human diver. So 2 of these animals are more closely related than the other 2. So you would write your answer on the form either like A and B, B and C, or A and C. Or if you flip the letters, that's okay too. But yeah, so which 2 of these animals do you think are the most closely related? Okay. Okay, next up, how many teeth do we think this animal might have? 0, they have no teeth. They have exactly 5 teeth. They have exactly 12 teeth, or they have between 40 and 60 teeth, depending on their age. This is a purple sea urchin. They also live locally in Monterey. Kirsten gave a great talk this morning on ranching purple urchins and its ecological benefits. Hi, come on in. If you want to grab a sheet, you can follow along. See if you can come up with these answers. And I don't expect everyone to know every answer. We're just going for like highest score is going to get the prize. Okay. This type of bird can be seen locally. You can go to Point Pinos, or you can see it at the Monterey Bay Aquarium. And this bird is kind of famous because depending on the culture, it can either symbolize good luck or bad luck. So see if you know what type of bird this is.
Yeah.
If you're a Taylor Swift fan, you might have a good idea of what this bird is.
No hints.
Okay. And then I'm also looking for the scientific name of this animal. If you only know the common name, you could put that for half points, but ideally I'm looking for the scientific name of this animal. As a reminder, a scientific name has 2 parts. It's a genus and a species. No, maybe, I can't say. All right, how are we thinking about those questions? Does anyone want to see one of those questions again? Yeah, of course. Let's go back and do 1 and 2 for those of our friends who came in late. Question number 1 is which of these animals is a sea otter, A or B? And then question 2 is how old was the oldest aged rockfish? There might be older ones out there, but this is confirmed, scientifically verified, aged using special otolith ear bone science. Oops. Yeah, those are your options there. Okay. All right, everyone feeling good? Okay. It's time for the answers. So if you have your sheet, I encourage you to swap it with someone nearby. You're gonna grade each other's answers. Alrighty. Okay, who thinks they know the answer to this question? Yeah. B. It is B, correct. B is the sea otter. Okay, who thinks they know how old the oldest aged rockfish was? Okay, who's got a guess?
C.
It is C, very good, 205 years old. Yeah, that's really old, for a fish especially. Okay, who thinks they know the general name of the type of plant-like plankton that produces over half of our oxygen? It's phytoplankton, very good. So the phyto meaning plant, because it's a plant-like plankton. Okay. True or false, corals live in Monterey Bay? Yeah? True, very good. Yes, we do have deep sea corals. They don't form coral reefs in the same way that tropical corals do, but they're still very beautiful, mostly soft corals, and often living in the very deep sea, like in the canyon out there. Okay, which one of these algae is giant kelp? A or B? Any thoughts? A guess, maybe? It's A, very good. B is bull kelp. What do we think juvenile white sharks eat? They eat fish. So adult white sharks have a much more diverse diet, but in general, we think that juvenile white sharks eat fish. Which two, this is a really hard one. Which two of these animals are most closely related? That is correct. A and C are most closely related. So echinoderms, which include sea stars and urchins and sea cucumbers, are more closely related to vertebrates than either of those two groups are to cnidarians, which is the group that includes anemones and jellyfish and others. And that actually we know because of some really cool work with how their larval development occurs. Okay, who thinks they know how many teeth sea urchins have? 5, very good. Yep, so sea urchins have 5 teeth. They're oriented in what we call an Aristotle's lantern. So it's kind of like a scraping tooth jaw apparatus that's on the underside of their body. So it would be against the rock in this picture. Very good. Okay, what type of bird is this? It's an albatross. Very good. Yeah. So we do actually have occasionally albatross in this area that you can see, like I said, from Point Pinos, but they also have one that was injured and needed rehabilitation, and it's now at the Monterey Bay Aquarium. Okay, who knows the scientific name of this animal? It's a mola mola. Very good. Mola mola is the scientific name. It's also sometimes called the giant sunfish or the ocean sunfish. You can get half points for that. And that, I believe, concludes our round 1 trivia. Great job, all of you. Don't worry, we still have 3 rounds to go, but who got over 7 points? You got over 7 points. Who got over 8 points? Yeah, 9 points. Who got 9 points? Yeah, do you get all 10? Congratulations. Yeah, so if you want to flip your sheet over to the back, please turn on Okay, we're gonna have a little change of pace for the next round. The theme of the next round is Moss Landing. Maybe some history, maybe some things that are going on in the surrounding area. Are you guys ready for it? Okay, let's bring it on. When looking outside from Moss Landing's patio, if you were to stand right there, or you can just look through the window right here, you can see some of the federally protected area of the ocean. Do you know the name of this specially designated area? I know this is a tricky question, so I'm giving you the hint that the answer is 5 words long, and you'll receive a 1/5 of a point for every correct word of your answer. You can think about words for protected spaces. Think about the location we're in. All of these things might be good things to think about. Hi, do you want to join us for trivia? Oh, no problem. Okay. Do you need some more time to think about this one, or are we good to move on to the next? Give me a thermometer. Good to move on, need more time. Okay, we're moving right along. This is a tricky one. Which former US vice president presented our lab, Moss Landing Marine Labs, with an Environmental Hero Award in 1998? In 1998, we were awarded the Environmental Hero Award from a former US vice president. Who was it?
He was former in 1998?
No, he was currently the vice president in 1998. I was intentionally vague in wording the question, but that is a valid clarification, so I'll give you all the hint that this person was the vice president in 1998. Okay. Moss Landing had to rebuild many of our facilities after a lot of them were destroyed in the earthquake, the Loma Prieta earthquake. It was a 6.9 magnitude. There are some of our faculty who were students here during the earthquake and they have really wild stories. If you know what year the earthquake was in, that would be a good time to write that down. The year of the Loma Prieta earthquake. If you were walking around, you might have seen the earthquake clock. It was a clock from the original building that had stopped at the time of the earthquake. It has the date on it, but don't go looking for it now because that'd be cheating, but it is somewhere out there. So if you're from the area, you might remember it. Okay, our next question. John H. Martin, who is a notable oceanographer and was also the former director of Moss Landing. We've named boats after him, the patio's named after him. He's known for his research on a certain element as a fertilizer in the ocean. Can you think of what element it might be? It is a metal. If you saw Petra's talk this morning, she spoke about it briefly. So it's the blank fertilization hypothesis in oceanography. A group of Moss Landing students recently took a trip to Baja California Sur. If you saw Roxy's talk, you might have seen some pictures from that. If you stay for Jess's talk after trivia, you'll definitely see pictures. There is a very, very famous Monterey County native and acclaimed novelist, and he wrote the book that we use as our textbook. It's called The Log of the Sea of Cortez. He's also written other novels that you have definitely heard about. Maybe read in your high school English class. Okay, we're going to move along to the next one. In Moss Landing, we have our iconic power plant with the two stacks that you can see from miles and miles away, pretty much across the bay often. What decade did construction on this power plant begin? When did it begin? Okay. Moss Landing is home to a society which honors a very famous Elizabethan era author and playwright. If you drove down towards Phil's Coffee Shop, you'll have seen this on your right-hand side. If you can name this very famous Elizabethan era author and playwright. So, again, you also probably have read some of these plays in your high school English class. Okay, Moss Landing is home not only to Moss Landing Marine Labs, but also to another research center. They use the acronym MBARI. MBARI is the sister institution to a landmark Monterey Bay organization. What is that organization? MBARI's facilities are really cool. They're just about less than a mile that way. You can see it from our patio, which is pretty cool. What degree do Moss Landing students hopefully graduate with? So, and I'm looking for the type of degree and the subject, but if you can only think of the type of degree, that's fine, you get half points. A fun Moss Landing tradition is that when you defend your thesis, you get to ring this bell. That symbol is that you are near the end of your journey of graduating with this degree. Okay, question 10. Moss Landing's logo draws inspiration from this famous Japanese woodblock print. Can you name the title of this piece of artwork? You can see our logo right here. It's very similar to that. Alrighty, does anyone need any questions repeated? No? All right, who's ready for some answers? Okay. First of all, does anyone feel like they maybe know the name of the protected area that we are looking out over right now? Okay. It's the Monterey Bay National Marine Sanctuary. Elkhorn Slough is the estuary that we're nearby.
Yeah, that's what, because that was right outside.
Okay. I did say of the ocean, and Elkhorn Slough is actually an estuarine system. These are my parrots, so I can give them a hard time. I also used to work here, so they should know this name. So if you got any one of those words, give yourself a fifth of a point. So if you got like Monterey Bay something, give yourself, 0.4 points. Does anyone?
0.2 for each one.
Point two for each word. Yes, that's a thing. So if you, who thinks they know who was the vice president in 1998?
Al Gore. Yeah.
It was Al Gore. Yes, Al Gore gave Moss Landing our Environmental Hero Award. Okay, anyone think they know what year the earthquake that took out? Very good, 1989. Yes. Yeah, so Di Stellar, who is our DSO and faculty here, she was in the basement of one of the Moss Landing buildings when the earthquake hit. And she has some pretty wacky stories about how they got all of the equipment and specimens out.
Did you sneak that in first?
No, I did not. Okay, who thinks they know what element John Martin studied? Yeah, it's iron. Very good. Yeah, the iron hypothesis, which is a well-known hypothesis in oceanography, is thanks to John H. Martin. Okay. Who wrote the Log of the Sea of Cortez?
Steinbeck.
It was John Steinbeck. Very good. Yes. Okay. What decade did construction begin on Moss Landing's power plant? Any guesses? Yeah.
I'll take a quick guess, that's like around 2010s.
It was much, much earlier than that. Good guess. Pardon?
80s.
Earlier?
50s.
Earlier? 40s.
Oh.
Construction began in 1949.
I knew it. I knew it was right before.
So there you go. 49. Okay. Moss Landing is home to a society honoring this Elizabethan era author and playwright. Who is it? It's William Shakespeare. Moss Landing is actually home to the Shakespeare Society of America right here in our town. If you're a big Shakespeare nerd, you might have known that. I personally did not until I started doing research for this trivia. Okay. MBARI is the sister institution to what Monterey Bay organization? The aquarium. Yes, the Monterey Bay Aquarium. So MBARI actually stands for Monterey Bay Aquarium Research Institute, and they are sister institutions. Okay. What degree do Moss Landing students graduate? Yeah, Masters of Science in Marine Science. If you just said Masters of Science but didn't have Marine Science, you get half points.
Just had marine science.
Half points. What is the title of this piece of artwork? The Great Wave of Kanagawa. If you just wrote the Great Wave, because that is how it is sometimes known. Yeah, very beautiful piece of artwork. And we're super proud to have it as part of our logo. How did y'all do? 9.5? Okay, do we have anyone that can top 9.5? No? Okay, I think you get the sticker. All right, last round. We're going back to a lot of multiple choice options for this round because the topic is oceanography. So a lot of our students and researchers here study local flora and fauna. So that was kind of the first round, but we also have a really robust bunch of oceanography labs. So we're excited to celebrate those, and hopefully these questions will give you some insight into things we study. Does everyone have a sheet, or they can just keep writing on the same sheet? All good? Okay, let's get into it. What is the biggest source of sediment in Monterey Bay? Is it runoff from rivers that flow into the bay? Is it crumbling sand from the sea cliffs that overlook the bay? Or is it sand swept up the canyon from offshore, like deep sea? Is a geological oceanography question. Okay, next question. Which of the following is not a characteristic of hydrothermal vents? So hydrothermal vents are in the very, very deep sea. They are where gases from within the Earth's crust are coming through to the surface. Some of our researchers like Amanda Kahn study hydrothermal vents, but you can't dive there because it's too deep and too dangerous. So they would use tools like ROVs or maybe submarines to get there. Would you find low iron, rich biodiversity, high temperatures, absence of light? You'd find 3 of these things. What is the one you would not find? Okay, next question. If you were here for Katie Duncan's talk this morning, you will know this term. And it's a term that describes when surplus nutrients cause excessive algal growth in a body of water. It's either hypernitrification, oligotrophication, superalgal bloom, or eutrophication. This is an issue that we can see when we have like agricultural runoff and all those nutrients getting into the bay. Yeah.
Are all those terms real terms?
No. Some of those I made up. But they're all derived, like the root means something in oceanography. Okay, this one is a free response, but given that there are only so many colors, it's kind of multiple choice. As light descends through the water column, different colors are gradually filtered out. What color of light is lost first or shallowest in the water column? So your options are the Roy G. Biv colors. If you don't know, you can think about how wavelengths of light might move through water, things like that. This is a physical oceanography question. What is the name of the climate oscillation that occurs roughly every 4 years, and it brings more precipitation than usual to California? This is something you would hear about on the Weather Channel. Meteorology. Okay, something that a lot of our chemical oceanographers spend some time thinking about is ocean acidification. It's a process that negatively affects calcifying organisms and many other organisms because it makes it harder for them to build their shells or skeletons. Ocean acidification refers to a change in ocean pH. Do you think it's an increase in ocean pH or a decrease in ocean pH? Okay. Question 7. This is another physical oceanography question. So due to Coriolis force and friction between water layers, water transport occurs at a right angle to wind direction. This is California's coastline right here. Wind is this giant yellow arrow moving kind of down the coastline. Do you think that it causes water to move onshore like this arrow or offshore like this arrow? It's one of those two. We'll give you all a few minutes to think out this, or a few moments. Okay. Where can you find the oldest water in the ocean? Oldest water meaning it has been the longest length of time since that water has seen the surface of the ocean and been able to do exchange with the air. Is it near Antarctica? Is it in the North Atlantic? Is it in the North Pacific? Or is it anywhere along the equator? And all of these places would be thinking about water towards the bottom of the water column. Question 9, this is another geological oceanography question. What type of rock makes up most of the ocean floor? Is it limestone, basalt, granite, feldspar? Okay. And last but not least, we're rounding out with a good physical oceanography question. Physical oceanographers spend a lot of time thinking about the characteristics of waves, things like wavelength, amplitude. What is the term for the length of time between wave crests or between wave troughs?
It's not wavelength.
It is not wavelength. You could write that, but you would be wrong. Wavelength is a measure of distance. I'm looking for a measure of time. Okay, does anyone need to see one of those questions again? Okay, feeling ready for some answers? Okay, swap your sheets with a friend for grading purposes. And we will cover the answers. Okay, who thinks they know the biggest source of sediment in Monterey Bay? Yeah. It is actually, yes, oh my gosh, sorry. You're right. You answered that correctly. I'm sorry, I shuffled the answers before I made this presentation today, in the order. Yes, it's runoff from rivers. Yeah, so, from the Salinas River specifically is where we get most of the sediment within Monterey Bay. Very good. Okay, which of the following is not a characteristic of hydrothermal vents? Yeah?
A.
Oh, really?
It's A, yeah. So hydrothermal vents are actually very high in iron because of the gas that is seeping out through and all the also magma and material that's kind of seeping out through those vents. There are high temperatures because we've got hot liquid kind of pouring through because it's warmed by the Earth's core. There is pretty rich biodiversity there. And there's an absence of light because it's very, very far beneath the ocean surface. So, yeah, low iron. Which of these terms describes when surplus nutrients cause extensive algal growth? You saw Katie's talk in this. Yes. It's eutrophication. Yes. Eutrophication means too many nutrients. Okay. Which color of light is lost first in the water column?
Red.
It is red. Very good. Yeah. So due to the energy of the wavelength of light, it is filtered out first, whereas blue penetrates much deeper. Okay. What is the name of the climate oscillation that we see around every 4 years? It's El Nino, yeah, sometimes called ENSO, which stands for El Nino Southern Oscillation, but El Nino is, either of these answers are. Okay, ocean acidification refers to an increase or a decrease in ocean pH. Decrease in ocean pH, yeah. So acidic pH is a lower pH, basic pH is a higher pH. So we're seeing a decrease in ocean pH over time. Okay, do we think that wind down the California coastline causes water to move onshore or offshore? It's offshore. So surface water is swept offshore, which is what causes our coastal upwelling because water from deep has to move upwell. The wind goes down coast and surface water moves offshore. Okay, offshore. So in this diagram, we've got the wind moving this way and water transport. Okay. Where is the oldest water in the ocean? It's the North Pacific. Very good. Yeah. So this is due to the meridional overturning current, formerly known as thermohaline circulation, and that's just how different water parcels move all around the world and where they go. Which type of rock makes up most of the ocean floor? Any thoughts? It's basalt. So basalt is an igneous rock and makes up most of the ocean floor. That's a geological oceanography question for you. Okay, and what is the term for the length of time between wave crests? It's not wavelength, it is. Nope.
Interval?
Nope. Wave period. If you wrote just period, that's fine. Yeah, so frequency, it would be how many crests per second. So the distance of time between, the length of time between peaks is a wave period or the period. Okay. Who got 7 points or above? Yeah? Who got 8 points or above? Oh. The first answer, the correct answer is B. I just misspoke. Yeah. So the correct answer for question 1 is B, runoff from rivers. So you got 8 points. Did you guys also get 8 points? 9? 9. Did you get 9? Okay, another sticker for you, my friend. That is all that I have for you all. Thank you so much for participating in our trivia today. I appreciate it. And I will see all of you back here at 1 p.m. for Jess's talk about field studies at Moss Landing.
Thanks for hosting. That was fun. Yeah.
Bye. Thank you. Thank you. Yes, of course.
I know, I was looking at it, I was like,
They're.
oh, nice. Yeah.
But to be fair, it's been a while. Since
Yeah.
And I'm just like, oh, I didn't. Like, what is he on score?
I know, I didn't know what that was, but I did know it was the small. But I don't know how I did that, but it.
Or limestone, like, okay, no, you're gonna have these.
It's at least not. Yeah.
I'm gonna go check in with, it's over.
No, I don't know anything about rocks, I just know that put in a rock amount from control rocks. No, that's what geologists do.
Oh my God, that's so funny. I used to have a Himalayan salt lamp by my bed, and people are like, if you have it by your bed, you lick it every night. What? You lick your finger and put it on. No. It's salty.
No, you don't. Like, with your finger, just.
Or that. It's just like. No shame in the game. It's like, it's my bedroom. No. I think the light will fit in though. I need to. On the bed.
Yep, that's where it is.
Give it to me. No way.
Spat it out, clean it up, and it's outside. And then you just put it back.
Was he right? You're like, I hope he was right. You put him in his mouth.
Like, yeah, I'm interested,
A geologist. Oh,
I don't need this.
me neither. That's so funny. I didn't know it was a thing.
Yeah.
I should just do that.
Let's go to the ground. Hey, you guys want to find out this rock? Hold on. Let me put this rock.
My friends are already like about, my friends are all like, so I go down to the viewers. And I'm like, okay, sorry, like, I'm gonna put a rock in it. I'm gonna tell you. And you wouldn't know what kind of rock it is, but I do. That's really insane. But like, I mean, it makes sense.
Yeah, it's like you're species of salty.
But then like, I don't know like what a very salinity rock is versus a high salinity rock. Unless it's literally a salt crystal. Then it's not salt. I don't know.
I don't know. So you have to go to training?
Yeah, or you do, you put it.
Yeah, so trivia just ended. Our next speed talk is gonna be starting at, we also have some really fun coloring. You guys want to check out our coloring? The bell is so that when we're done with school, we ring the bell and we party. Yeah, because students who study here, we study here for sometimes 40 years, so it takes a long time, and when we're done, you ring the bell and you say, oh, I'm all done.
Kind of like a graduate.
Yes.
The graduation.
It goes kind of like take it off, yeah.
Yeah. Someone might was done. I forgot, like, this is kind of like graduation stuff, right?
I don't think, somebody finished it with more just hey, we want to show it off.
I think somebody said that they did it and they were like, I need to cancel it out because, I can't. I'm like, it's bad luck. I'm like, no. I don't know, it's just saying. Yeah.
Is.
Oh, yeah.
It's very hard.
I was thinking about something to Sal, he presented my freshman year in event side, but then it came my friends and I just laughed and he's kind of his name from what I understand, a lot of his like making tags for sure, so I'm like, that's not what I'm saying.
We built, the signal that everyone.
That is, but it's just like, I'm not super into the text, but I'm trying to figure out the last.
Yeah.
Not so much.
But there's also portions where it's like you can just go around. And set it up. Like today, I asked finding my tag.
Well, because if you find ones and yeah, I think he talked about that. It seems like, yeah, there's sometimes when like, it's like. They're like, I mean, I would look for a tag for money. Yeah. Yeah, and like, anything that would just be like, cool for me, so I just didn't find it. And I get to turn it into.
Got tag.
See, like, that's because.
Yeah, then we got attached.
Where at, this week? Babies or like full grown? Were y'all like in waiter or like? I don't know why, like, you pink and sharp with a fishing rod, like, yeah. If you wanted to, well, there's like some people like in like the south, like they catch catfish by using their real horn. Like, you die, like, you know, just it's like that. Yeah. It's just like, I'm getting there.
The structures
And then release them?
Yeah, against kids, we did a batteries. I mean, that's just the best too.
One thing.
But you have to keep it very simple.
They're not, which class?
It's,
I like, I'm in my second year right now, so I'm kind of like finishing my GED.
I would say you're gonna be very something.
It's not, I mean, that's.
You know Ryan Salter?
She is. Is he a professor?
He's not a freshman.
He does that class?
Yes, I'm good for he just figures out, and then you have to get to 4:37.
Man, I need to figure out what I have, probably side because that's what I'm doing since start, but.
That's. I love it. The last 1 minute.
I love batteries.
I was just walking and like, oh, look at a pancake.
I know, they're so cute, and they're like this range. But like I don't want to, you just quickly burn them, that would say. I know, it's like, oh.
That's all.
Yeah, nothing else needed.
That's all we did.
That's good.
There's like a little disc thing you play with the dog. And you put it on. On it and then.
You know, nothing, yeah, just the normal stuff.
And Duncan. They were there. Yeah, I just tried to make it loud.
Yeah. Are you guys need a break? I mean, like, I kind of want to check out that earring. Okay, because I was sitting here and I was like, no, I'm not. Yeah.
Well, my here.
Yeah, mine too. Go for it. Go to the. How did you know? Because it didn't look like a male, but it looked like a female, it looked like halfway in the key. I was like, slay. Oh. That's a big hat.
He's like, I. Are you ready to go to guitar?
Oh, wait, I'm back in. Yes?
Yes.
What are you gonna talk about?
Babies.
Yeah, snacks? See, adorable.
Snacks.
I like to talk about snacks.
You go there and say strawberry,
Yeah.
orange, green.
And I would have thought.
Oh, yes. It was actually.
It looks like a garage.
Yeah.
Why? I don't know.
Like, I'll be like, park or the garage? She was like, go garage.
New heart or new garage? No, new garage.
Like, we never let them do anything in there, and it was like, there's just so much stuff to look at.
Look at that stuff is pretty. See what, have you seen the courtyard? Right, that's like a garage and stairway. So stupid. Okay. 1.7, we're not. No, it's just like it. Well, yes, there's a block. That's time. Says it's almost 1 p.m. Do you know how many people have asked for the show? I know that too. I did that 2 years ago. Yeah, you need someone who's like passionate about it to run it, you know what I mean? We've never seen one, we've never like been in the process.
Or like shake people up, just, you care. But then, you know.
We don't need to.
Yeah.
Yeah. It was fun, it was a lot of fun. It's such a. We can barely.
The only way here?
Like. Going to one meeting.
You should just follow us. When people say like, you should be like, oh, the puppet, your skills.
How much squid, I don't know. Yeah,
Wow.
there's a bar, keep that one. Wow, that's a good advice. Yeah.
It's like in the lobbies. Like in the middle of the store, there's like a clock. And you're like, where is it? Where? And it's like halfway across the store, like it points out, I'm like, how did you even see that? It's insane.
Good eye.
Yeah, lights.
What else do you see?
Do you see a fishy? I see one right there.
In the physics lab, he said viscosity, he said intertidal, internal wave.
Who's up
Can you say Reynolds number? Can you say Reynolds number? Reynolds number. I'm just gonna shove your hand in your mouth.
I have all this stuff.
Sounds longer now. Wait, do I have to want back? Oh, you chance.
We can't forget my stuff.
Hi there, our next speed talk is gonna start at 1 p.m. So in just a few minutes now. Yeah, it's gonna be a super great talk about fertility.
Yeah, just kind of see what.
Yeah.
Right.
Been here a little bit, I'm here in the placement at one and my time to go question. Stage. You're here now? Perfect. Okay, so the video is.
Yeah.
The owl is set up for speaker, not for audience. So when audience members ask questions, we want their presenter to repeat the question or the answer so people on the live stream can hear it. It is your job to talk with the speaker and the Q&A like the members do the timing. So what I've been doing is at 10 minutes, I believe, pamphlets that you see. So they know that it's 10 minutes, 15. So what people are doing like 12-minute talks, 15 minutes of questions. The front row is reserved for people with mobility or vision or hearing aids. All of the transitions and videos are working very smoothly. That's it.
Everybody knows the question about that.
Oh, yes. One, then Maddie.
So two other people together.
Actually, no, I didn't mind parking bottom floor, it's there.
Yeah, so it's in the back. Before you take it back.
One car is one car is the sun, because just so. They've been bought with the right. Whatever, it makes it soft. Yeah.
Where's everyone? Sit in the second row, guys. The front is for hearing and low mobility. Yeah.
What do you want me to do for introducing them and stuff?
Just say that I'm a second year. In psychology lab, science lead, also in dietary.
Okay, so like your talk, and then you're talking about.
Oh, and then you can say, and they're gonna talk to us about field studies at Mount Sinai.
Okay, cool.
And then go.
All right, and then what do you want for time?
I'm gonna have my phone next to me the whole time, so it's okay.
Okay.
So I'm just gonna go 10 minutes, and then you'll be 12.
You get 15 total questions.
Yeah, with including questions, so.
Yeah.
Thanks. The owl is on my phone. Yeah, so they can hear us.
Okay.
Yeah.
I'll give it an answer.
Food maybe? No.
It's only that big.
It was like right after I stopped praying when it's not that. I'm definitely you were friendly best. Yeah. Oh, yeah, actually can make sense.
Okay. It's 12:59. We're very, very punctual here. That's three rings about.
No, I know.
You didn't jinx yourself or something.
No, it's, no.
I think it's bad.
We do it every time. It's part of our seminar.
Yeah.
I've run it for on Thursday.
Oh, no.
That's really loud.
That is true. Okay. Hi, everybody. We're going to get started with another round of our speed talks. Up first for this session is Jess Franks. Jess is a second year here at Moss in the Bike Lab and working down at DiveOps. And today they will be talking about field studies here at Moss. And I will let you take it away.
Thank you. Hi, everyone. Like Duncan said, I'm Jess. I'm going to be talking to you about field studies at Moss Landing Marine Labs. Thank you for being here. So today we're going to talk about a few main points. We're going to talk about what exactly are field studies, why do we do field studies, how do we make them happen. We're also going to talk about my experience in a couple of classes, so the Tale of the Islands, I'm calling it. And then we'll finish off with some sort of outcomes that happen in field studies. This is a picture on the top
from Isla El Pardito in the Gulf of Mexico from a recent field course I just got back from. And we'll talk a little bit more about that in a few minutes. So what are field studies? Well, here at Moss Landing Marine Labs, they tend to be subtitle or scuba diving classes. At other facilities, they could just be any sort of class where you are immersed in the field. They don't necessarily have to be underwater. Moss Landing Marine Labs has typically offered a field intensive course every single year in the spring, and it alternates between a class out in Monterey Bay, class in Mexico. Last year we went to Catalina Island. Sometimes I go to Chile. So they try to sort of diversify the different places that they go to. Field studies are essentially just that, an opportunity for students to get out in the field and answer scientific research questions about our natural world. Why do people conduct field studies? I mean, look at these views. Like, why would you not? Also, for the food. These are fish tacos from our trip down in Baja. But that is one of the, the great reasons. But for more reasons, not everything can be figured out in a lab. There is no real environment like the actual environment our study organisms exist in, right? So that is the best place to study them. It can be logistically, difficult, but it is, at least in my perspective, a really excellent opportunity to actually answer questions about what we're interested in. Additionally, so we can discover novel things about the field when we're actually in the field, and we're able to make natural history observations about the things around us and really understand what the organisms are doing, how they're interacting in their environment. And we're just able to make observations that you can't really make in a lab. On the top picture, that's me and my buddy in the estuary. And then this is a nudibranch that we saw in the estuary. And I would have never known that nudibranch was in there if I wasn't in there. So, yeah. How do they happen? A lot of moving things. Yeah, when we go in the field, we take a lot of stuff with us, especially scuba divers. So these are a few pictures of some of the bags that we've taken with us. When we went to Isla El Pardito, we had to bring all of our belongings onto these pongas. And we brought a scuba diving compressor with us, which is really heavy. So it's an experience, but if we have to do all that and we do it again and again, it must be worth it. What do they entail? So for field studies, we spend a lot of the first half of the class writing proposals, doing a lot of research, really figuring out what we're interested in, trying to learn as much about our study systems as we can, although we've never been there a lot of the time. So we do a lot of planning, and then we get there and nothing is what we expected it to be. So yeah, basically when you're in the field, everything is about being able to adapt, being able to come up with a new plan when it's not what you expected. So it teaches us a lot of valuable skills about how to, adapt. So yeah, you get a lot of new experiences, a lot of new skills. You experience so many new environments. I slept in a hammock for 9 days, and I had never done that before. There were no bathrooms on the island I was at for 9 days, so that was a new experience. And there's also a lot of new food, if you're traveling to different countries. So yeah, what else do they entail? Research projects. So I know I've been hyping this up as like a lot of fun and food and views, but in reality, every day you're working, you're diving 3 times a day, you're helping other people out on their research projects, you're entering data, you're doing stuff in your makeshift labs, you're learning about the systems around you. So they're definitely intensive, but again, it's worth it. So again, some of those new environments I was talking to you about, this is my hammock setup on the island. That's me brushing my teeth with my friend, something we don't often get to do together, but we got to do together every day on the island. And I'll talk a bit about this a little bit later, but when you are with such a small group of people for 2 weeks, you really do bond. And it's really community building, which is lovely. Like we all just help each other out, and to feel supported is a really beautiful thing. So everything is DIY in the field. You do not have access to the same resources that you would normally when doing science. So this is our makeshift lab. We had everything plugged in the generator there. We're under someone's palapa that's falling apart, but we're still doing really excellent science, which is so cool. This is a makeshift pinata we made because it was our friend's birthday when we were on our trip. That third picture is us filling the scuba cylinders every night with that compressor I told you about. So got to fill your own tanks when you're in the field. And then the one on the end is Noah. That's his makeshift lab where he spawned urchins. So you can do a lot in the field, even without the same resources that we sort of take for granted in the lab every day. What does it entail? Being in the water constantly. So I literally never go out of my swimsuit on these trips. So these actually were from Catalina Island. These are just some more pictures. That's this whole talk, if you haven't realized. But yeah, this was us on the left on the boat going out to do some rhodolith bed surveys, some experiments. Yeah, you were just constantly ready to get in the water and help. And then this was when we were in El Pardito, more photos of us doing science underwater. And yeah, it's a beautiful place to be. Okay, so now we're moving on to the next part of my talk, which is the Tales of Two Islands. So 2 different courses that I took. The first one is Catalina Island down in LA County. It's a picture from a hike we did. It's just stunning there. So this is the drive we took from Moss Landing Marine Labs all the way down to Long Beach, and then we took a boat over to Catalina. The project that I did there, I'm only going to talk about a little bit of science today, but we have to talk a little bit about science. The project I did there looked at invertebrate communities in giant kelp and sargassum. So sargassum is an invasive species of seaweed down there. So I was looking at how the native species versus the invasive species was affecting the invertebrate communities. And that is not what I planned to do when I went down there. I had a completely different project, but I pivoted with the help of my professors and I came out with a really cool project. I can't remember the conclusion. I think it was actually the mixed plots were the most diverse, but that was Catalina. And then the most recent trip that we got back from a couple of weeks ago was this small island, El Pardito, like 2 football fields long, maybe, in the Gulf of California, near La Paz. And this is a picture of the island. That hammock that you saw where I slept was right up there. It's a small island where this family of fishermen have been living there their family's been living there for about 100 years. And some scientists here have had a working relationship with them now for quite some time. So they allow us to come and visit and do work there, which is truly an incredible, we're very lucky for that. But yeah, so this is the drive we did for that, and that was a lot longer. We drove all the way to the border and then all the way down the peninsula. It was beautiful and took us like 3.5 days. And we may or may not have gotten the van stuck in some sand a couple times, but we got it out. So for my project for this one, I looked at 2 species of green seaweed and I disturbed them. So I cut them in little pieces and I put them in these little compartments that you see up there with either different substrates or at different lengths. And I watched how they grew over time. And there's my experiments in the water on the right-hand side, growing, and they grew, which doesn't always happen. So this one actually worked out how I thought it was gonna. So I learned something from the first time, obviously. Okay, what are some of the outcomes for field courses? This is the last part of the presentation. Data, scientists love data, so that's the most obvious outcome. Also, you can do presentations like I'm doing right now. A lot of students will make posters or even publications from the work that they do in field courses. Like I mentioned earlier, community building is a really big part of it. I feel super close to the class I went with, our professors, as well as the people that we met on the island, which is, very unique and really awesome. I gained a lot of new perspectives. I was one of the people helping plan this open house, and I came back from this trip and I was like, everything's gonna work out. That was my new perspective. I wasn't nearly as stressed out. And there's also a lot of emerging research that new scientists or scientists coming from minority backgrounds can become very empowered from field studies, and it makes them feel a lot more capable. which is one of the things I find to be really cool and why we should keep pushing for field courses to happen. I know they're logistically and financially difficult, but they're so, so worth it. Here's some pictures of the community that we built. So these are all the people I was living with for like 9 days. It's our class and the fishermen on the island. It was nice to just know everybody around me. And then that's us at dinner when we were celebrating Haley's birthday. We all ate dinner together every single night. Also very community building. So what's the key point? Field studies are foundational for both scientific research and early career scientists. So if you ever get the chance or you ever want to support something in science, I suggest it's field studies. And with that, I will thank you and take any questions. Yeah.
What was your favorite piece of gear to take?
Favorite piece of gear, like diving gear or?
Like anything that you felt like, wow, I'm so glad I have this.
A sun hat. Oh my God, Audrey's clothesline. All my clothes were constantly wet and there was no washer or dryer, so I would always use her clothesline to dry my clothes with the wind. Yeah, and some earplugs would have been nice because it was really windy. Yeah. Any other questions? Okay, cool. Thank you, guys.
Thank you.
When you get questions, you're going to speak there, that's like, that's what we're going to do, just talking to that. Time now.
I can tell you. Sorry.
Alright. Hi, everybody. For anybody who is joining us, these are our speed talk series. We're doing a quick 10-minute presentations from Moss Landing students and faculty, covering various topics relevant to our research here. Up next is Maddie McKay. Maddie is a second year here, working in the Fike Lab. And Maddie will be presenting today on research in bull kelp and seaweed cyanotyping.
Right. Okay, so who here has heard of cyanotype before? Okay, pretty good. That's about half of the people in here. So I just wanted to start out by explaining what a cyanotype is And basically, it's a really old photographic method. It's one of the first ways that was created to print images. And it was invented by this guy here on the right, Sir John Herschel, in 1840. What exactly it is a combination of two different chemicals, including potassium ferricyanide and ferric ammonium citrate. And they sound kind of scary, like cyanide, but they're different, so they're safe for you to handle. And when you combine these two chemicals together, they are sensitive to UV light. So when you put it on a piece of paper, put an object on top and put it out in the sun, then that creates basically an image of whatever you put on top of it. And his intended use for it was for things like blueprints and mathematical tables, basically just a way to reproduce an image over and over again. But this was adapted by this woman here, Anna Atkins, around the same time. She was a botanist and a photographer. She collected tons of different specimens of both plants and seaweeds. And she was a close friend of Sir John Herschel. So she took this cyanotype method from him, and instead of using it for his intended purposes, she used it to create these prints of all of her different specimens. As we can see here, there are some ferns, but she used other plants and algae as well. And she was known as the first woman to create a photograph, which is awesome. And the photograph was using cyanotype. And also she was known as the first person to publish a book illustrated with photographic images, which again were the cyanotypes. And what's really amazing about her is, you know, it wasn't really common for a woman to be a scientist during this time in the 1800s, but she really was a scientist at heart. She might not have been recognized as one during this time, but she definitely is now, because she provided us with this amazing historical record of all of the different species that she was collecting during that time. As I mentioned, she did publish some books, and one of those books was photographs of British algae cyanotype impressions. I think there were maybe 3 different volumes of this book, and each of them included hundreds of cyanotypes of seaweeds. They were labeled with the species name, and as you can tell, it's really a good image. You can get great detail of what the seaweed looks like. So we can look back on these images that she created now and learn more about the different species that she was seeing, how they've changed over the last, you know, almost couple hundred years, and maybe see how the species names have changed and that sort of thing. So, you know, it was really groundbreaking work that hopefully she realized she was doing during the time. But yeah, it's kind of set the stage for using cyanotypes for this sort of thing instead of just its intended use for things like blueprints and that sort of thing. So why use seaweed? As I just mentioned with Anna Atkins, it's first of all a great way to preserve the natural history in the area during that time. But for me personally, as Duncan mentioned, I study bull kelp. And kelp forests are critical habitats that provide ecosystem services like primary production. They take in CO2 and release oxygen. They help reduce erosion in areas, and they also provide habitat for thousands of animals and other seaweeds and that sort of thing. But despite being one of the most diverse and productive ecosystems, they experience really large changes in space and time. Some of you might be familiar with this issue. Bull kelp in Northern California, which is what I study, has decreased by over 90% in the last decade. And this was over a 350-kilometer stretch of coastline, so a really large area. And this was due to several different factors. First, starting with the marine heat wave, nicknamed the blob, in around 2014 area. During this time was also the onset of sea star wasting syndrome. So if you've heard of the sunflower star, it's a really large species of sea star that can, you know, get this big and has 24 arms. And that population also decreased by over 90%. In some areas, they're considered locally extinct because they just don't exist there anymore. And sunflower stars in particular were the top predator of sea urchins. So without sunflower stars, sea urchin populations exploded, and sea urchins are what ate all of the kelp in this area. And sea urchins are part of what is preventing kelp from coming back in these areas. There is hope though. There is a lot of amazing restoration work being done. Specifically here at Moss Landing, this is kind of one of the hubs for bull kelp restoration in the area. And what we do here and what they do at other places is we can grow bull kelp from the little microscopic stages all the way up to when they're 100 feet long if we so want to. But also by growing it microscopically, we can grow it on whatever we want, like rocks and twine, and then that can be planted in these areas where the kelp has disappeared. But with restoration work, that's only possible if there's effective science communication to, relay to the policymakers and managers that this is something we need to care about because bull kelp and kelp forests are foundation species and they're super important. So the restoration is also very important, not only for, ecosystem services, but also our culture and economy and fisheries. So it's a really important issue. And this kind of science communication aspect is what makes me really interested in using cyanotypes and specifically using seaweed because it's a really great way to take something that's scientific, make it really beautiful and easy to understand. And it can, help portray the importance of these ecosystems and hopefully help relay to policymakers that restoration is something that's important. And then, of course, I'm also interested in using seaweed for cyanotypes, just like Anna Atkins for this natural history aspect, by preserving a piece of bull kelp, for example, maybe in 100 years, hopefully this doesn't happen, but maybe that species doesn't exist anymore. It's not in that same area where we initially found it. And I really love these images on the top right. It's some historical seaweed presses. So that's another really great way to preserve seaweed. Those are probably like 300-year-old presses and they last really well throughout time. And then in the bottom right, this is an image from, I think, like the early 1800s of people in Europe tide pooling and collecting seaweed. And again, as I mentioned during this time, it wasn't typical for women to be scientists. So women were a lot of the times the ones out there collecting seaweed and pressing it and making cyanotypes. So, they were doing science in that way by creating this record of what we were seeing. So now on to the actual process. If this is something that you want to go out and do yourself, you first have to start by seaweed foraging, just like these people here in this image. And there are some rules and regulations you need to keep in mind. Generally, it's a pretty easy thing to do in California if you're just collecting recreationally. But the few rules that I put up on here, first, you can't collect more than 10 pounds of seaweed a day. You also cannot harvest a few specific species for conservation purposes, including sea palm, which is this tiny palm tree looking one in the bottom left. And you can also not harvest eelgrass or surf grass. And finally, this is kind of the hardest rule in Monterey, but you can't collect in marine protected areas. So whenever you're considering collecting seaweed, always check the rules and regulations of the beach beforehand and if it's a protected area. That's one of the great things about Monterey is a lot of it is protected. So I personally like to collect in Pebble Beach, which is just south of the Asilomar Reserve. Lots of cool seaweeds in there. And once you have your seaweed, you can use it to cyanotype when it's fresh, or you can dry it and press it. If you're interested in seaweed pressing, I'm just going to go over that really briefly, but basically it's a big sandwich. I also mentioned that we are teaching people how to cyanotype and press seaweed in the courtyard at the phycology booth, so you can learn more there after this. But what we like to do is put a piece of cardboard, something to soak up any excess water, like a towel, a piece of watercolor paper, which is what you put the seaweed on top of, and then a piece of wax paper so the seaweed doesn't stick to the next piece of, towel, and then another piece of cardboard, and you just press that all down with like a strap or something or a big book on top. And the key with seaweed pressing is to keep it dry. I recommend changing out wet cardboard and towels every couple of days, keeping it in a cool, dry place, and just checking up on it until the seaweed is completely dry and flat. So once you now either have your pressed or fresh seaweed that you collected, you are ready to make a cyanotype. And it's pretty simple. You can first either buy the chemicals that I mentioned in the beginning and mix them together yourself and coat that on a piece of paper. An easier way, if you're just starting out, is they sell pre-prepped paper so that you don't have to worry about combining the chemicals yourself. And then you take your paper that's prepped, put seaweed on top of it. I like to put a piece of glass on top as well so the seaweed doesn't blow away. And then you just put that out in direct sunlight. The time kind of varies depending on how cloudy it is. But I found if it's like really sunny, even just 5 minutes out in the sun will do the trick. If it's a little cloudy, maybe up to 20 minutes or so. And once it turns this kind of brown color that we see here, you can take the seaweed off and you want to rinse the paper underwater, and that basically develops the image and turns it into that beautiful blue color. And I just wanted to end by saying that you can really do this with anything. You don't have to just do it with seaweed. You don't have to do it for scientific purposes. It can just be, you know, it's a beautiful art form and a great way to express yourself in different ways. So I put some examples on here. You can use photo negatives, which is something I've been experimenting with a little bit more lately. You can use household objects. You can use your own shadow. If you're not in the seaweed, you can also use flowers, different specimens, similar to what the first image from Anna Atkins that I showed, you know, tools. It's really a great chance to just create cool images, you know, and if you want to do it for scientific purposes as well, that's awesome. You know, you can write and label your prints about what the species is and where you found it and that sort of thing. But yeah, it's just, in my opinion, a great way to combine art and science and communicate the necessary restoration to policymakers. And that's all I have about cyanotype. Questions? Yeah.
When you use your own chemical to kind of just paint it onto watercolor paper?
Yeah, I like watercolor paper, and the thing with the chemicals, when you mix them together, they don't last very long. They last maybe like 6 hours. So you only want to mix like a small amount, whatever you think you'll need. And then I just use like a sponge brush to brush that on the watercolor paper. Okay.
Stand up like maybe here. So I get 15 minutes. You can make like 12 and breathe.
Cool, yeah, I don't think that'll be 15, so I think we're good.
All right. All right. Once again, if anybody is joining us, this is our seminar, like speed talk series. So we have student faculty members here coming in to give quick talks about their research and relevant topics to Moss Landing research for the open house. Our next speaker is Hannah McGrath. Hannah is a second year. Hannah is a second year here. She studies in the biological oceanography lab, and she's going to give a quick talk about some of her work doing long-term monitoring here in the Bay and working on harmful algal blooms. All right.
Thank you.
Thank you for the introduction. In that case, I'll just dive right into it. Here's a picture of our beautiful bay. If you're local or not local, this is what it looks like on a beautiful morning during sunrise. Okay. So let's dive right into it. What are phytoplankton? They are, so the name actually comes from the Greek word meaning plant or wanderer, and phytoplankton are microorganisms. They're mostly autotrophic, so autotrophic meaning they produce their own energy. And so you may recognize this equation. So carbon dioxide plus water and light produce glucose and oxygen. And so this is what a lot of these species undergo. So these are all phytoplankton species. They come in a lot of different shapes and sizes. So since it produces oxygen as a byproduct, every 5th breath you breathe, you can thank these guys. So they're like the trees of the water. And so that's what I like to study. And so they are the foundation of our marine food web because, you know, lots of organisms eat these and it goes up the trophic level. So we're going to dive into it. But even though phytoplankton are super duper important, there are some harmful algae. There are some harmful ones, and that's what we're going to discuss today. So not all phytoplankton are bad. I want to preface that because that is a stereotype they get. Okay. So when they do grow in big abundances, so there is a broad range of effects of phytoplankton that they can have. And so in this case, this is a harmful algae bloom, the backdrop, and it's when they grow in exponential abundances and they can either make the water hypoxic, so low, very low oxygen conditions, so fish can't breathe, or they produce toxins themselves like domoic acid or neurotoxins. And so here's a fun little cartoon I found. And it's this guy mowing his lawn, you know, a nice Saturday. I'm sure a lot of you can relate because there's been a lot of rain lately and you use fertilizers to grow things in your garden. And he's mowing and excess nutrients goes into the watershed. And it's this algae monster saying algae blooms and it's terrorizing his property. Well, you might be able to relate. And so in Florida, they have red tides and nutrients. There are a lot of controlling factors that go into what causes a harmful algae bloom. So nutrients can be one, light, temperature. And so this is what we're going to explore a little bit. So you may have seen red tides. Raise your hand if you know what a red tide or an algae bloom is. Okay, good. Beautiful. So you may have seen some of these in your local news. So this says dead giant Goliath groupers wash ashore during red tide, fish killed in Florida. So for my research in undergrad, I actually went out in Florida, and this is me. I'm not doing it because of COVID. It was the year of COVID, but I was outdoors. It just smelled so bad. You can see the water. It was pretty rancid, a bright yellow color to it. It's discoloration. There were manatees that were kind of beaching themselves because of the brevetoxin. It was a species called Karenia that we were studying, and it caused massive fish kills. So just swaths of them on the surface of the ocean. So not a good thing. There's also on the other side of the East Coast, dog killing toxic algae discovered in 3 New York City parks. And this is where I grew up. And every Saturday and Sunday, my mom and I would go out and rowboat. And this is what the water would look like. Not very inviting, but we weren't swimming in the water, but I always wondered what it was. So it sparked my interest in studying harmful algae blooms. And so it can get pretty dense. More locally, in the news, why a neurotoxin is closing crab season in California. You may have seen this in 2015 if you were local. It was from a DA event called from Pseudonychia, a type of species that is locally out in Monterey Bay. And then another headline, hundreds of dolphins and sea lions have washed up dead or sick in California amid toxic algae outbreak. So here's a quick video. Okay, maybe not. There was a quick video. It's basically about a woman talking about Monterey Bay and the 2015 bloom, but we can dive into that after. I don't know why that's not playing. Okay, so what are the local species in Monterey Bay? Well, here's some local species. They do look pretty inviting, but some of them are toxic. So this is Ligulodinium, Akashiwa, my lab mates and I like to call it fat pants, Alexandrium, Percentrum, which looks like a leaf, Dinophysis, a teacup, Pseudonychia, a needle. And so Pseudonychia is the big one that people focus on in California, and it resulted in this. So it produces a neurotoxin called domoic acid, and it can result in marine mammal mortalities like this. This was actually taken right off of the beach down that way, right in front of Mbari, our neighboring scientific community. And it's a little terrifying. And so how do we monitor for HABs in California? I'm so glad you all asked. Well, we have a network of stations and it's called Cal HabMap. And this is actually my side job out of being like part-time as a grad student to pay my bills. And so I'm part of the California statewide HAB network and forecasting system. So along the coast, we have 9 stations and the star represents where you're sitting right now. And MW2, Municipal Wharf 2, it's right next to the commercial wharf if you've been down to Monterey. I sample off of that every Wednesday. And other people are doing it on the same day. So we have a time series that dates back a really long time, just a few decades. And so for sampling, I take observations, so water color, temperature, just basic observations, or there's a lot of activity at the wharf, nutrients, chlorophyll, phytoplankton, and different toxins. And then I compile that into a weekly report. And so here is a QR code to look at the stations more in depth. So this is my sampling site. It's a zoomed in version. And this, again, this is where we are. And we have 2 stations in Monterey Bay. This is the one we're focusing on. So we're zooming in again. So every Wednesday, as I mentioned, I go down in my small electric vehicle. I fit all my equipment in there and she gets stuffed. I fit all my equipment in there and this is my sampling site. It's actually very pretty. The Monterey Abalone Company is right over here. This is me taking observations. And the first thing I do is basic observations. So air and water temperature, cloud cover, and ocean color. For ocean color, yes, it is subjective. I use a Munsell color scale. So I compare whatever color that is to the ocean and record it. So here's all the equipment that I put in my car. And I'm gonna go more in depth. So there's just buckets. Anyone can really do it. It has buckets, a net, a sampling device. It's pretty low cost. So this is the first thing that I do after observations. I get my phytoplankton net. So it concentrates all of the water that I put at 5 meters. And so I'm capturing all the phytoplankton in the water in that area. I then put it in these small transparent containers. So you can see there's a lot of things in there. There's some phytoplankton. So then I take it under the microscope and you can see they're beautiful. I think they're beautiful. There's Akashiwa, fat pants. You have Eucampia that looks like a necklace. And then you have a lot of Pseudonychia, so those long chains. And so I examine it using a way called Relative Abundance Index, and it's a way to qualitatively measure what's in the water. Another method that I use is called a Van Doren, and it collects whole water. So I take it down at different depths, so 5, 4, 3, 2, and 1 meters. And all that water is put into a large car buoy. And I basically do a few things. I spin it down to concentrate it, and then I do a cell count. All of this information you can find online. It's readily available and it's accessible to the public. It's called ERDAP. It's on this database and you can play around with the data. You can see real time. So this is updated frequently every week. So here is a snapshot of what's in our water. If you type in Environmental Biotechnology Lab, I take a picture of what's in the water each day, well, every Wednesday. And so you can get an idea of what's in the water if you don't believe me. There's images, and I write a weekly report. So sometimes it's along the lines of, there were a lot of playful sea lions out today, or the seaweed was crazy. Sometimes I make it narrative. Another way to know what's in the water, you can scan that QR code. We have a weekly blog. And so it's both stations, Santa Cruz and Monterey Wharf 2. So the north and south of the bay. So you can see if the species are differing, if you should be fishing at that time, if it's toxic. So it's a good snapshot of what phytoplankton are present and if you should be worried. So stepping back, why is this important? Well, although weekly monitoring, across California helps us predict different HAB species and their frequency we can monitor short and long-term patterns, and it allows us to look at different environmental factors in relation to different types of phytoplankton species that are present. And so, yes, this data is being used, not by just me or this community, but a lot of local stakeholders. For instance, I'm sure you're familiar with the Monterey Bay Aquarium. they use this data because a lot of their tanks have, they take in the water for their filter feeders. And if the water has a lot of toxic species in it, they don't want to feed that to their oysters or their exhibits. Another local stakeholder is Monterey Bay Abalone Company. They grow abalone, and abalone are susceptible to certain toxic species. And so they can move the cages at certain depths in the water column to avoid certain blooms. And so this data set is used by a variety of different people for academic, scientific, citizen science. So you're more than welcome to go on there and check it out. And yeah, that's my talk. If any of you have questions, I'm more than happy to answer. Yes.
Why is there no station at Moss Landing?
Yes. Why is there no station at Moss Landing? Right now we are seeing a budget cut. In general, I think it's because of money at this point and starting a new state. They're trying to add more to other stations before doing a new station. I'm not sure about that answer, but I can get back to you. That'd be a good idea. Yes.
What have you been seeing so far this year for harmful algal blooms? Have you been seeing a lot or less of the dangerous kinds?
Yeah, we haven't had a big Pseudonychia bloom, which is the species that produces domoic acid up here, but there was one in the south. So we haven't seen a lot of activity and sense of toxins up here, which is a good thing. Usually it occurs the spring diatom bloom, which is typically toxic. We see in March and April, but I think we missed it this year. So that's good. Yeah, that's good news. Beautiful. Well, yes.
How do you see like harmful bacteria and whatnot change as rain comes and goes? Is there like a safe amount of days wait, like stay out of the water, that sort of thing?
Yeah, I don't know a lot about bacteria. I will be transparent, but if there's heavy rains and you're near an agricultural field, I probably wouldn't go swimming in that anytime soon. I think you'll know by the discoloration of the water, there will be a lot of sediment, and also potentially phytoplankton because they're stimulated by overload of nutrients like phosphorus and nitrogen, which are in these fertilizers.
Yeah. Do you see a difference between the Northern California sites and the Southern California sites in terms of what's in the water?
Absolutely. Yeah. So we want higher resolution. As I mentioned, there are only 9 stations along the coast of California, because it takes time, it's money, and those are the long-term HAB sites. But yeah, they can be different just within the bay. And that's only, I think, 45-ish miles away from each other, both stations. Yeah. Yeah, well, I welcome you to check out any of the websites, and if you have questions, you're welcome to, contact me. Yeah. Thank you.
Oh yeah, when you get okay, it's like
I went up to go dedicated.
I was gonna apply for the real deal, Yeah. thanks. Yeah.
All right. Okay, if you are just joining us, this is our next talk in our speed talk session. So we are having people come in and give quick talks about their research and projects ongoing here at Moss Landing. So our next speaker is Jake Todd. Jake is one of the senior members of the ichthyology lab here. And he's going to be giving a talk on his ongoing work with the California Collaborative Fisheries program.
All right. Hi, everyone. Thank you all for coming to my talk today. Like Duncan said, my name is Jake Todd. I'm the lead technician here at Moss Landing Marine Labs for the California Collaborative Fisheries Research Program, or CCFRP for short. Today, I'll be talking about 17 years of collaborative fisheries research off Central California and walk us through some of the results and data that we've been seeing over time. So to give you all a little background, the California Collaborative Fisheries Research Program, or CCFRP, was started here in 2007 when the Central California Network of Marine Protected Areas was put into place here off the Central California coast. This program was devised with a variety of inputs from the fishing community, resource managers, agencies, and resource scientists. In 2017, we expanded this program statewide. We now have 5 other partner institutions, including Moss Landing Marine Labs here. So we have full MPA coverage from Cal Poly Humboldt to the north, all the way to UC San Diego in the south. The overall goal of our project is to conduct scientifically sound research with the intent of using these data for MPA monitoring and fisheries management. So why take a collaborative research approach? Collaboration has been fundamental to the success of our program in so many ways and extends to our partner institutions, charter boat captains, the fishing community, resource managers, and overall the general public. Some of the benefits of collaborative research include the ability to include stakeholders in science and co-management of our marine resources to help generate finer scale data on these important fish stocks. We can help create a shared understanding of the status and our marine resources that are so valuable to us and we all use. And we can overall help build trust between resource users and resource managers that are making those management decisions. And in general, we can increase overall ocean stewardship towards our marine resources. So a little history behind why our project was started. In 1999, the California state government passed the Marine Life Protection Act, or MLPA. Essentially, this act mandated that the California Department of Fish and Wildlife reevaluate existing MPAs or marine protected areas, along with design and implement new MPAs, with the whole goal of all of these MPAs working together as one large statewide network. And with the overall goal of that being to increase cohesiveness and effectiveness at protecting and preserving marine life, habitats, and the valuable ecosystems that are right off our coast. Currently, there are around 124 MPAs in California, covering about 16% of our state waters. Right now, there's a big push to increase that number. There's a big act, 30 by 30, which is trying to aim for about 30% protection by 2030. So when the MPAs were put into place, they divided up the California coastlines into 3 different MPA regions. The first group being established here. It doesn't work. Being established here in Central California in 2007, followed by Northern California in between 2010 and 2012, and ending with Southern California with about 50 MPAs in 2012. Like I mentioned earlier, there are 6 collaborating academic institutions as a part of the California Collaborative Fisheries Research Program. Up north in the northern region, we have our partners at Cal Poly Humboldt and UC Davis Bodega Marine Laboratories. In Central California, we have us and Cal Poly San Luis Obispo. And in Southern California, we have UC Santa Barbara and the Scripps Institution of Oceanography. And each of our partner institutions, along with us here at Moss Landing Marine Labs, surveys 2 of their local marine protected areas as a part of these surveys. So in total, we survey about 12 MPAs, and that number is kind of bounced around from year to year, depending on funding and sampling availability. But for the sake of this talk, I'll just be talking about some of the data and trends that we're seeing here on Central California, as that is the longest time series that we have as a part of that project, because we did start in 2007. So as I mentioned earlier, each of our monitoring groups surveys 2 of their local MPAs. Here on the Central Coast, Cal Poly San Luis Obispo to the south surveys Point Buchan and Piedras Blancas. And here at Moss Landing Marine Labs, we survey Point Lobos, just south of Monterey, and Año Nuevo, which is just north of Santa Cruz. And we're here at that Gold Star. So there's Año Nuevo MPA and reference sites with the MPA is that protected area in red, and those reference sites are areas open to fishing in blue. And then here's that Point Lobos site as well. Each of these, like I just said, each of these sites are separated into MPA and reference sites. And these are those areas, again, that are open to fishing that anyone is allowed to fish in. And within each of these sites, we have a fixed set of 500 meter by 500 meter grid cells. These cells are fixed in space, so we can return to the same areas year after year and ensure we are fishing in those same areas year after year. On any given sampling day, we'll randomly select 4 of these grid cells to fish in. So each time we visit a grid cell, we aim to fish for a total of about 45 minutes, which kind of breaks that up into 3 15-minute drifts or 3 15-minute chunks of fishing. Helps us to fish in different areas of the cell as well as kind of break it up, let us regroup after a hectic fishing drift or something like that. Sometimes the weather doesn't cooperate, and these have to be broken up further into smaller drifts. Either we're drifting out of the cell, sometimes we're catching too many fish and can't measure them all that fast. So we'll stop fishing, let us get caught up, and then start again. But overall, we aim for 45 minutes per grid cell, which kind of chalks it up to around 3 hours of fishing time per day. During each fishing drift, we collect a variety of data, such as the start and stop times, GPS coordinates for the start and end of that fishing drift, so we can map our drift. We also take a lot of GPS coordinates throughout the drift, so we can accurately map that drift, as well as assign each fish a coordinate to where it's been caught. We keep track of whoever caught a fish. We identify those fish to the lowest possible taxon, most likely species. We take the total length of that fish, any general condition, if it's in bad condition or anything like that. And lots of times we will tag a subset of these fish as well to gain some more data on fine scale movements. So just to orient us all to some of the graphs I'll be showing on the next few slides, I want to include a couple example graphs of what we look for. All the graphs that I will be showing will look similarly to this. We have year on the X or horizontal axis and our MPA performance metric on that Y or vertical axis. All the data coming from these protected areas, the MPAs, is going to be in red, and all of those data coming from the open areas is going to be in blue. So when we're evaluating and looking at graphs to see how well MPAs are working, we're often looking for a divergence of values similar to these. The trends might be different. They might both be declining. One might be going up, one might be going down, or they might both be increasing. But overall, if there is that divergence in value over time, those MPAs are generally working for that performance metric. And then very quickly, before I jump into the data, I wanted to quickly walk us through how we calculate our abundance metric. We use CPUE, which stands for catch per unit effort. And essentially, this is a calculation of all the fishes we catch divided by the total amount of time we spend fishing. So like I said, we keep track of that time when we're on a drift. We multiply that by the number of people that are fishing, and then we subtract any time someone might not be fishing, if they have to retie their rod, if they're in the bathroom, if they're not feeling that great, or they're just tired and they're taking a break. So this gives us a metric of CPUE in catch per angler hour. And just a little bit more about that CPUE metric and MPA performance. This top example, we have 2 people fishing for the same amount of effort. They should be catching about 4 fish per person, whereas the bottom, there'll be 2 people fishing for the same amount of effort, and they'll probably each catch around 1 fish per person. So that top example would be a value of high CPUE, which could then indicate there are more fish in that area compared to that bottom area. So on average, we see there are more fish inside these MPAs compared to the outside, those reference areas. So here on the left, we have all 4 of those MPAs here on the Central Coast that we've sampled since 2007. You can see they started pretty similarly as we would expect. They're both open to fishing for forever. And then come implementation of that MPA, they start to diverge, and you can really see the MPA values branching off quite greatly. On the right, it's separated out by MPA. Some MPAs are doing better than others, but in general, they're all doing very well. There are more fish inside those MPAs compared to those reference areas. And then we can further take that catch per unit effort, or that abundance metric, and combine it with those length measurements that we are taking on the boat, and calculate something known as biomass per unit effort. So we can take those length measurements, use published length to weight relationships, calculate the approximate weight for each of those fishes, and then we can multiply that by our CPUE value, and that'll give us the biomass per unit effort, which is essentially a weight metric for the whole sample that we are fishing in. Again, similar results to CPUE. There's more biomass inside the MPAs compared to outside. And so this really hammers down that idea that there are more fish inside the MPA, but there are also bigger fish inside the MPAs compared to those reference areas. And this is also very important with these certain species. These species, rockfish species, are very slow growing. They mature very late in life. So oftentimes the bigger fish mean more offspring. So these MPAs are really providing them a good refuge to spawn and help repopulate those populations. So some quick takeaways just to wrap this up. Fish are more abundant, larger in body size, and higher in overall biomass in all of our sampled MPAs. MPAs are working well across the statewide network, particularly here in Central California. I didn't touch on any of the data from our sampling institutions, just because, like I said, we have the longest time series here, but we're seeing very similar trends in all the MPAs across the state. Collaborative research is beneficial to the overall understanding and management of our marine resources. It's helping to bridge that gap between those resource managers and those resource users, and they have direct input into how we are using this data and how we're collecting the data. And it really is beneficial to get them all involved in the data collection process. And long-term monitoring also helps to identify any natural versus human-induced changes in these nearshore ecosystems and help eliminate natural variability. So as you might have noticed in those graphs, there's quite a lot of variability from year to year, but the overall trend is it's increasing. So if we were not doing long-term monitoring and just sampled 2 years, we might see a overall decrease. And that could lead us to make some false assumptions about these overall systems. So thank you all for joining today and listening to this talk. If you'd like to stay up to date with CCFRP, feel free to follow us on any of the social media platforms. You can head to our website, ccfrp.org, and sign up to volunteer with us to go fishing. And we'd love to have you out on the water with us. And lastly, I'd like to extend a huge thank you to our funding agencies, our collaborating partner institutions, the captains and crew members of the various vessels we've worked with over the years, and overall the many, many volunteer anglers that have come out with us year after year to help dedicate their time to help us catch these very valuable data. And with that, I can take any questions. I'll be down the hallway in the ichthyology lab for the rest of the day. If you'd like to chat some more, I'd be more than happy to talk. Yeah, I think we have time for one question.
Is your database held by NOAA? Are they the ones that keep the overall?
So we actually house the database here ourselves. It's actually also been publicly published online. It's hosted by the California Ocean Protection Council. We're our main funding agency throughout some of the years. So all the data is actually publicly available it's called Data One, like it's their database, online database.
Could you access it through trying to get through NOAA's database for?
I don't think it's linked to NOAA's database. NOAA uses quite a bit of our data. Our data has contributed to about 10 different stock assessments so far on groundfish. But they're kind of just taking that from our database and taking any metrics.
Is there any thought that it should be coordinated directly?
Yeah, NOAA doesn't really do a lot of the surveys like this. They have a hook and line survey in Southern California, but that's about it. They do a lot of trawling surveys up here, but these are kind of they do a lot of surveys that are out deeper and with a lot more different methodologies, things like that. So they might not mesh very well. So, yeah, thank you.
The turnover.
They're managed by the state.
So. All right, everyone, thank you for tuning in. We are going to continue with our talks. I'm your new facilitator, Olivia. Next up, we have Dr. Sarah Smith talking about ocean iron fertilization for mitigating the climate crisis.
Thank you. All right, let's make sure I know how to drive up here. Excellent.
Yep.
I will wait just a minute while people kind of funnel in. So yes, I am the biological oceanographer here at Moss Landing Marine Labs, and I'm also visiting scientist at the J. Craig Venter Institute, and on the steering committee of an umbrella organization called Exploring Ocean Iron Solutions. And this umbrella organization is not a legal entity, but is a group of scientists and researchers and governance experts, social scientists who's come together to chart a roadmap forward for tackling what I believe to be one of the biggest issues facing mankind today, which is the CO2 problem and the climate crisis. So what I'm showing you here is a plot of atmospheric CO2 concentration that's generated by Scripps Institution of Oceanography. And you can see that there's a seasonal kind of increase and decrease. This is probably very familiar to many of you. But the general trend is that the atmospheric CO2 levels are rising in addition to other greenhouse gases, which are causing big problems in terms of global warming. So what do we do about that? Well, the number one thing that we need to do, I have a fancy animation that's not going to pop up, but is to stop burning fossil fuels. That's the easy answer to that question, because that's where all that CO2 in the atmosphere is coming from. But it's not so simple, because if we stopped burning fossil fuels completely today, there is still too much carbon dioxide in the atmosphere to prevent the atmosphere from warming beyond what our targets are to prevent kind of ecological damage. So what I'm showing here on the plot is a timeline on the x-axis and the billions of tons of CO2 emissions per year on the y. And that dot shows where we are now and how if we continue business as usual, CO2 in the atmosphere is going to continue to increase. And then the gray curve is showing emissions reductions. If we stop burning fossil fuels, pivot to clean energy, employ technologies for carbon capture and sequestration, and start to think about kind of not belching all this carbon dioxide out into the atmosphere, that's a slow curve that gets us to 2050 to a much lower place on that line. But as you can see here, in order to achieve net zero, we actually do need to take out some of the legacy carbon dioxide from the atmosphere. And that's according to the agreement laid out in the Paris Agreement in 2015 to kind of prevent warming to really dangerous levels. So I want to take a moment to distinguish between carbon capture and sequestration technologies, which are things you might have heard about, like coupling CO2 scrubbers to stack gases. That's still a CO2 emissions industry, but it's reducing the amount of carbon that's being emitted from the burning of fossil fuels versus CDR, or carbon dioxide removal. And this refers to a new set of technologies that are specifically tailored to actually just take CO2 out and store it durably away from the atmosphere and slow down our global warming. So when we think about where carbon is stored on the Earth, the ocean does a disproportionately large amount of the heavy lifting of storing carbon. Relative to the atmosphere, which has about 600 gigatons of carbon, the deep ocean combined stores about 37,000 gigatons of carbon. So a tiny amount of atmospheric carbon can be moved into the deep ocean and have less catastrophic consequences than when it's found in the atmosphere. So the question for us now is, Can the ocean store more of this carbon? Can the ocean remove some of that legacy atmospheric carbon dioxide? And in 2021, the National Academies of Sciences published a research report summarizing 6 of the main technologies or 6 of the main areas where the ocean can be involved in carbon dioxide removal. And that's a suite of technologies referred to together as MCDR, or marine carbon dioxide removal. Among them are iron fertilization, deployment of these ocean tubes for artificial upwelling and downwelling. Seaweed farming is envisioned as a way of removing excess CO2. Ecosystem recovery, you may have heard of blue carbon initiatives like mangrove restoration. Ocean alkalinity enhancement, this has been in the news a lot lately. It's like putting Tums in the ocean and forcing the ocean to take up more CO2 from the atmosphere, and electrochemical approaches. The one I'm going to focus on today is iron fertilization, because this is a real Moss Landing special. And all of what we know really was born out of research that was done at this institution by our former directors, John Martin and Kenneth Cole. And I don't have enough time today to go into details to justify the significance of this contribution from a research point of view, but I am going to talk a little bit about what ocean iron fertilization is and how we're thinking now about how to approach using it as a technology to remove some of this excess CO2. So the short story of ocean iron fertilization is that oceanographers recognize that there are big patches of the ocean that have many nutrients that are capable of supporting phytoplankton, or the base of the food web, the microscopic base of the food web. Yet they were devoid of the kind of life you might anticipate, given how much nutrients were there. And it was through careful investigation and careful analytical techniques determined that these regions were limited for productivity for the availability of the micronutrient iron. And iron is not something that we think of being limiting on Earth at all. It's very abundant in Earth's crust. But the ocean, because it's such an essential nutrient, micronutrient for life, in the ocean it gets scavenged readily, and it's not soluble in seawater. So whatever is delivered to the ocean precipitates out rapidly if it's not taken up by biology. So with this recognition, John Martin put forth the iron hypothesis. And this hypothesis was built on the idea that iron supply to those regions of the ocean controlled phytoplankton productivity. And when iron is delivered, phytoplankton bloom. Phytoplankton, if you remember photosynthesis, they take in CO2, carbon dioxide, convert it to organic carbon. And when that amount, when their biomass dies and sinks out of the surface ocean to the deep sea, that carbon is sequestered in marine sediments or in the deep ocean away from the atmosphere. And so through glacial and interglacial cycles, the supply of iron to the ocean regulates global climate. And very famously, John Martin is remembered for declaring, if you give me a half tanker of iron, I'll give you the next ice age, recognizing that this may be a tool that humankind could use to potentially engineer the climate. So Kenneth Cole, after John Martin's passing, went and took this hypothesis and tested it out in the ocean by taking a ship out to these regions, spreading iron sulfate, and determining that, yes, in fact, it stimulated a phytoplankton bloom. So what you're seeing on the left is what the ocean looked like before, and what you see on the right is the result of just adding iron, nothing else. Massive, massive bloom was stimulated. So how much carbon are you actually capable of sequestering with a technique like this? That all depends on the efficiency of transfer of that carbon from the surface ocean down to depth. In the surface ocean, if it remains there and bacteria respire it back to CO2, it ventilates out back into the atmosphere and it's not sequestered. However, if the biological carbon pump via marine snow production and flocculation actually clumps out and sinks to the deep ocean, that carbon can be sequestered on the order of hundreds of years, thousands of years, or potentially permanently in the sediments until we come along and figure out ways of drilling it up and burning it for our energy needs. So this is a really promising technique, and geoengineers are thinking about ways of deploying what we know to be true now, which is that large regions of the ocean are limited for this micronutrient iron. What are the uncertainties that remain? What is the ecological impact? Are we going to change the way that ocean food webs are structured? Certainly. If you turn something like the oligotrophic Pacific, where there's no nutrients and no life, into the Monterey Bay, you are fundamentally altering ecological trophic food webs. Is that something that we as humankind want to do or need to do? And I'll just remind you that that curve at the very beginning of this talk, which is that we are already kind of beyond what we need to do in terms of removing CO2 just by burning, reducing emissions alone, requires us to seriously investigate some of these approaches and the pros and the cons that might be associated with them. So with that in mind, this technique, which had kind of fallen out of favor due to some kind of rogue engineers and this general distaste with the concept of using the ocean as our dumping ground to store excess carbon, the conversation has come back to the front now with the recognition of the severity and the urgency of the need to solve this issue. To stay involved in this, I'm part of that umbrella organization I mentioned at the beginning, EXOIS, and we have published a couple of papers now. These are not in the peer-reviewed literature, but they're white papers, and they're able to be downloaded on our website, oceaniron.org. I'll skip forward. There's the website here. We have one on ocean iron fertilization. This is really describing the state of the art. It summarizes all of the generations of fieldwork that have been done to allow us to arrive at our current interpretation of events. And then last spring, almost a year ago exactly, we convened a workshop in this very room where a bunch of oceanographers and social scientists and international policy experts and governance experts and economists gathered to discuss the next steps and to really consider what do we need to design if we're going to think about the next generation of field studies to address some of the remaining uncertainties about the durability of carbon sequestration, the ecological impact, and how to address the question of social license. So I would encourage you to come check that out. Here's a picture taken out of the Martin's point of view of the group that convened here. This group is largely driven by the science. And one of the things that we agreed upon very early on is that any of these marine CDR methods is going to be valuable on the carbon market. That's the idea. The economy drives all technologies that end up going to scale. And in order to approach this question in this problem ethically and responsibly, we needed to develop a code of conduct. And so we have a code of conduct that's been published on our website. We're considering all of these kind of ethical standards, the costs, the ecological impacts, and the scaling. And at the end of this month, end of next month, we're gathering in Woods Hole again to start to design the next generation of field studies, hopefully start to be deployed in 2025, with the goal of scaling it up to kind of a full-scale marine CDR operation by 2050. I will say, this is not the only answer. This is not the answer. This is in addition to being able to wean ourselves off of fossil fuels. And this is one technology in a portfolio of technologies that will likely need to be deployed to keep our emissions below what we consider could be a climate crisis. So it's there's reason to be optimistic, and it's really exciting to think about oceanography being used to apply to solve a problem. But it is not the only answer, and it's not the end. It's just the beginning. It's so great to see Moss Landing continue to be involved in it. And with that, I think 2 minutes?
Yeah.
Okay.
We have time for 3 minutes.
Okay, great. Happy to take any questions.
Yeah.
Pretty amazing. How do you how do you distribute the iron into the ocean? Is it gonna hurt our fishes? And with acid rain, how are you gonna keep the ocean alkaline with all the acid rain going into it?
Good question. Yeah, ocean acidification, carbon dioxide from the atmosphere equilibrates in the surface ocean and drops the acidity of the ocean, which damages organisms. This will keep things in balance more or less, because as the CO2 enters, the phytoplankton take it up and sequester it out. So there's a mechanism for it to kind of buffer in that surface ocean to some extent.
Get the iron.
How do you get the iron in? So it's iron sulfate is the form, and it's like iron filings that get they get acidified and actually piped in at the fantail of the ship, right into the propellers, and the propellers, as you move around and lay a patch, stir those iron filings into the surface ocean. It is inefficient, and 90% of the iron that you put in precipitates out immediately. So we call it the bake-off. It's a series of research actions that are considering different forms of iron, different iron delivery methods that might make this a lower CO2 endeavor to invest. You know, ships make a lot of CO2 when they go out. You don't want that if what you're trying to do is remove CO2. So a lot of consideration into how could we do it better? But that is the way that it's been done. I hope not. Yeah.
We have time for one more question.
Okay.
Yeah.
When you said you're using iron sulfate.
Yeah.
You said iron filings. I thought iron filings were iron oxide.
So essentially that's dissolved in acid. So it's basically, it's not a siderophore bound version of iron. It's not a organically complex version of iron. It's just as iron dissolved, you can get it into the surface.
But in what form? And you said iron filings.
Essentially, but it has to be dissolved in order to spread it. So it gets acidified and dissolved.
And it doesn't come out iron oxide.
It does, it precipitates out, much of it does.
Okay.
Yeah.
All right. Thank you very much.
Thank you.
Next up, we have a wonderful first year student, Audrey Sarin. And she is here to talk about some rocket breeds. All right, thanks, Audrey.
Hi, everyone. Thank you for the introduction, Liv. My name is Audrey, and I'm excited to share a little bit about rotolith beds with all of you. So I'm going to be introducing you to my new friends, the rotoliths, but first I'm going to give you a little bit of an intro about myself, a little more than Liv did. My name is Audrey. I use she, her pronouns. I am a first year student at Moss Landing Marine Labs. I am in the phycology lab here, and I am also in the coastal ecology lab at CSUMB. So I spend a lot of time thinking about things that are plant related and things that live on the benthic or on the bottom of the ocean. So all these things kind of led me to study rotoliths on a recent field course at Moss Landing in Baja California Sur. So let me introduce you to rotoliths. Rotoliths are a non-geniculate coralline red algae. If anyone knows the word geniculate, it pertains to the root of the word for knees. So bendy, so a non-geniculate algae knee that does not have any flexible bendy joints. Coralline refers to the fact that this algae creates kind of like a skeleton. So it leaves hard parts behind when it dies. So it's got that nice calcium carbonate base going on. And then red algae is just refers to not only the pigment, but the family that rotoliths are a part of. So we are looking at a non-geniculate coralline red algae with all of those characteristics. So I've got a picture up here. My laser pointer gonna work? That's okay. I've got a picture up here of a rotolith in a petri dish, and then a couple here on trays, and I will show you a little bit more about them in a moment. So rotoliths can come in a diverse array of forms. They can come in this more foliose flowering form, a more warty lumpy form, and I actually have some rotolith skeletons here with me today. I'm going to pass the, please be a little bit gentle with these, but pass them around to each other. So that is the coralline part of the body that's left behind. As I described, they leave that encrusting system behind when the pigment is gone. And so, yeah, you'll be able to see these forms as they come through the crowd. Rotoliths are really special because they're what we call ecosystem engineers. So the rotoliths themselves are living algae. They are a plant essentially, but they are also creating habitat for many other things that are living around them. So imagine if you lived in an apartment building, but the apartment building was also an alive living organism, right? Maybe a different organism than you. So my image of a large person and some small people isn't exactly accurate, but it's kind of a wild thing for us as humans to conceptualize, but it's actually very common in the ocean. If we think about giant kelp, most of us who live in this Monterey Bay area are really familiar with giant kelp. That is another great example of an ecosystem engineer species. Giant kelp serves as a nursery for many other small fish, developing larvae, et cetera, just as rotoliths do. So in this way that these two organisms are comparable as ecosystem engineers. Rhodoliths are also really an interesting system because rhodolith beds, which I'll talk more about in a minute, but are aggregations of rhodoliths, actually move around a lot, as do the individual rhodoliths. So if you'll see in the specimens that come around to you, but as you also may have noticed in the pictures, rhodoliths are in general kind of spherical or have a kind of elliptical shape going on. And the reason for that is they need to be able to roll back and forth, maybe a little bit gently with currents as they come, or with tides or different waves as they come around. And it's really important that these individual rhodoliths be able to move around a little bit because they're free living. They're not dug into the sediment in the way that coral reefs are, but they're still playing host to a lot of organisms living in them. And they're algae, they're plants, right? And how do plants get their how do plants get their food, anyone? Sunlight. Sunlight, yes. So if a rhodolith was to just kind of settle down on a sandy bottom and not move at all, it would eventually become covered by sand and sediment that was kind of washing over it over time. So the fact that they move is really important for their life history. Rhodoliths and rhodolith beds are very widely distributed. You can see that they can be found in Alaska, in Baja California Sur, where I just was, in the Mediterranean, in Oceania, up near some of the more Nordic countries. So they are so widely distributed that we believe that their distribution does not have a lot to do with latitudinal factors, that is factors having to do with where on a map they are, but they're really more worried about local factors. So things like wave movement, because of that motion that I described to you earlier, they need to live in an area that has enough wave motion that they can get that gentle rolling movement, but not so much that those kind of delicate skeletons are going to be breaking apart. They need to live in sort of moderate-ish temperatures, but that's not as important to them as that wave motion is, at least on the distribution, you can likely tell that. They like to live on sandy bottoms that are either flat or very gently sloping. Rhodolith beds can look something like this. This here is a photo that I took in Baja California Sur, and you can see these clumps here are all different rhodoliths. This is an image courtesy of an organization called EcoAqua, and it was taken in the Oceania area, I believe. So the color of the pigment is slightly different, but you can see that we've really got a sandy bottom and then just these free living individual rhodoliths all around there. Now, the structure of rhodolith beds, or many rhodoliths together in one area, is also really important for that ecosystem engineering process I talked about. So having all of these rhodoliths together actually helps buffer a little bit of the water flow going between the individual rhodoliths, which increases larval retention. So if you're a really small, tiny larvae, you're going to get pushed wherever the water is taking you. But if you can sort of nestle in between these two rhodoliths, there's a better chance that you can settle there and maybe be among some of your conspecifics, among some larvae of your same species, which is kind of a beneficial strategy for survival. As I mentioned, there are so many different species that are associated with rhodoliths. We call these species infauna because they are fauna that live in the rhodoliths. A pretty snazzy term in my opinion. There are hundreds of species that are considered rhodolith infauna, though they may also be found in other places. Some of the most common ones include tunicates, like these ones on the left, sponges, like these ones on the right. Still no laser pointer. What a shame. We also see anemones sometimes hiding amongst the branches of the rhodolith, like this small brown anemone here. Ophiuroids or brittle stars are very commonly found. In fact, across different locations, you very commonly will find ophiuroids hiding in rhodoliths. Urchins can be found within rhodoliths. You might be thinking urchins are quite large and rhodoliths are quite small. Well, that's absolutely the case. Rhodoliths can vary in size a little bit, but what we normally see is larval urchins. So we'll see pretty small urchins, maybe the size of a pencil eraser, will be living in and around rhodoliths, hiding between those branches, being all protected until they get big and spiky like this. We also see a lot of different species of bivalves as in fauna and rhodoliths. For example, these scallops, maybe some different species of clams. And once again, since these organisms can be quite large as they grow older, we're seeing them as their larval form living within the rhodoliths. However, also sometimes we will see small adult boring clams in the middle of a rhodolith. So instead of boring into a rock, they will find a rhodolith instead and think, good enough. The most common infauna in rhodolith species in Atlantic and Mediterranean oceans are chitons. We see more ophiuroids over on this coast, but chitons are also very common kind of across different locations, and nudibranchs can be found in most different locations that rhodoliths can be found. This is one species of nudibranch that can be found amongst rhodoliths, but there are quite a few. Okay, so I just gave you a little bit of an introduction to rhodoliths and the rock and reefs, but I also think that it's really important to talk about how we study rhodoliths. Before we do that, I want to take a little bit of a step back and think just about laboratory science and field science. In my mind, at least as a scientist here at Moss Landing and having gone through undergraduate training at another Cal State campus, I sense a little bit of a divide between laboratory and field science in terms of what I think about, but I wanted to kind of get the pulse on what you all are thinking. So I was wondering if this could be a little audience participation moment. Could someone raise their hand and tell me a tool or a technique or a topic that they think about when they think about lab science? Microscope, yes. Data analysis, yes. Pipettes, yes. Sequencing, okay, cool. Lab coats. Clean environment, yeah, all great things, okay. What about when you think about field science? What are some things you think about? Boots. Transects. Transect tape, yes.
Variable environment.
Variable environments, yeah, out of your control. Anything else? Okay, these are all great things. Yeah, so when I think about lab and field science, I have some of these similar ideas. A lab is a clean space, you've got a scope, you've got pipettes. Field is a dirty place, you're wearing waders, you're going to get data and bring it back and look at it later. So something that I learned when I was learning a lot about rhodoliths is how to merge lab and field science, which I thought was a really important lesson that I've learned here at Moss Landing. So as a part-time rhodolith scientist, this is what my lab has looked like for the past couple of weeks when I was in Baja California Sur. So this is by no means a clean or sterile environment. As you can tell, we have, we've got snacks over here. We've got a lot of different research projects going on. And I think that that is sort of a really wonderful perspective on science. So this is me in the field and this is me in the lab, but I want to zoom in to show you that some of the things we've talked about are here. Even though I'm in the field and in a wetsuit, that's something I would associate with field science. I have a microscope. I've got a tall petri dish. There are pipettes over here and over here. This is my friend Haley, who is also studying rhodoliths. She's got a caliper. That can be a lab or a field tool. I've got probes, pipettes, this really cool cell counting dish that we often think of as a laboratory-only tool. And then that's me in the field deploying some artificial rhodolith treatments to look at how infauna might colonize them. So if this experience of studying rhodolith has taught me anything, it's that you really can break down the barriers between laboratory science and field science. And in fact, that is one of the most effective ways to study really cool ecosystem engineering organisms like rhodoliths. In fact, that's how we study kelp forests a lot of times here. And that's how rhodoliths have been studied a lot in the past, especially by students who are now professors at Moss Landing. So basically, the message I want to share with all of you is that the categories that we apply to science are not necessarily as hard and fast as we believe. And so the world can be your oyster or your rhodolith bed. So I encourage you all to go forward and be curious about the world around you with whatever tools that you have. I'd like to thank all of the people who made my project possible. I'm really excited to keep developing it and maybe share some of the results out soon. And with that, I would like to invite your questions. No questions about rhodoliths? You guys know everything there's to know about rhodoliths? We were in Baja California Sur. So we were very, very south of the Baja Peninsula on the Sea of Cortez side, just off of La Paz, on an island called Isla El Pardito. Yes. Yeah.
How would they interact with coral? Because I know algae and coral don't get along.
That's a great question. Rhodoliths tend to live in areas where there's not coral, not necessarily like geographically, but just in terms of substrate. So they live on sandy bottoms. So if there was a coral reef area, the rhodoliths simply wouldn't persist there. Great question. Anything else? All right, whoever has those specimens, I would love to have those back. Oh, thank you so much.
Thank you. Another round of applause for Audrey. Thank you, everyone. It is now 2:30 and we have another speaker coming up, Rachel, who is in the geological oceanography lab. She's going to talk to you about some mud.
That's funny. Yeah, hi, everybody. I'm Rachel. I'm a third-year student in the Geological Oceanography Lab. I like to say I study mud, which is not really an over-exaggeration, but my whole goal with this talk is to convince you guys that the microbes that are living in marine sediments are not only super important, but they're also very cool. And so maybe you guys will fall in love with mud a little bit as well. So yeah, let's get started. Just kind of a table of contents of how this talk is going to go. We're going to start with talking about why marine sediments are so important. Then I'm going to go into a little crash course into microbial metabolism. We're going to get into the weeds a little bit with some chemistry. And if that's not your jam, that's totally okay. You don't need to like fully understand everything I'm talking about to like get the meat and potatoes of what's going on. And then I'm going to roll into some examples of some of my favorite mud living microbes, including Winogradsky columns, AOM consortia, purple sulfur bacteria, and pinkberry consortia, and cable bacteria. So why are marine sediments so important? They're important for two reasons. One major reason is they have a huge role in the carbon cycle. So we have lots of phytoplankton living towards the surface of our oceans, and they're fixing lots and lots of carbon from the atmosphere, drawing CO2 out of the atmosphere and fixing it in their bodies as organic carbon. Approximately 1% of that organic carbon produced by those phytoplankton then settles to the deep ocean. And that might not seem like very much, 1% might seem like a trivial amount, but when you extrapolate that across the entire ocean, that's a tremendous amount of carbon that we're able to remove from the atmosphere and take down into marine sediments. And once it hits those sediments, it can be locked away there for thousands, not millions of years. So that's a major way that we can mitigate the impacts of climate change by sequestering carbon to the deep ocean. The other important role that microbes play in these marine sediments, and this is happening right out the window here in Moss Landing and in the Elkhorn Slough, the microbes that live in these sediments have some really wild metabolisms that are able to remove a lot of the nutrients and toxins that we're putting into the environment. Things like nitrate and phosphate that are running off of the agricultural field and coming from wastewater treatment plants that would otherwise trigger harmful algae blooms like red tides. So they really help to mitigate our impacts on the environment, not only with the carbon cycle, but with the nitrogen and phosphorus cycles as well. So ready for a lot of chemistry. This is our crash course in microbial metabolism. This is something that I really love. It's called the redox cascade. So water doesn't circulate super well in marine sediments. That means that the microbes that breathe oxygen consume that oxygen really, really fast. So as you can see in this graph, we have concentration up on the x-axis and approximate depth on the y-axis. And so oxygen runs out pretty quickly. Oxygen is not available. So what do the microbes do that live in the layers below where oxygen is able to penetrate? They've adapted to be able to breathe something other than oxygen. They've adapted to breathe nitrate in this example. And then below that, once nitrate runs out, they consumed all the nitrate. What do the microbes do? They start breathing manganese, and then they breathe iron, and then they breathe sulfate, and finally they produce methane. And if you look at the chemical reactions on the far right of this, you'll notice that if you breathe oxygen, for every molecule of organic carbon that you breathe, that you respire, you get 854 kilojoules per reaction. That's quite a lot of energy. So that's why it's the best option. That's why most microbes breathe oxygen. And that's why it runs out so quickly. Below that, you'll see that if you breathe nitrate, instead you're getting about 800 kilojoules. And as you continue moving down through the redox cascade, each of those different elements that you're breathing produce less and less energy per reaction, per molecule of organic carbon. And that's what produces this redox cascade. And that's what powers those weird metabolisms that remove those nitrates from the overlying water. So how could we visualize that redox cascade? We can use these Winogradsky columns, which are absolutely wild and also amazing. If you want to, you can waddle out into a mud flat and scoop a bunch of dirt into a tube, and eventually it'll form this layering pattern if you give it a month or so. So in the overlying water, we have lots of photosynthetic cyanobacteria living, and then that bright green layer on top, that's more of those photosynthetic cyanobacteria. Below that, we have our heterotrophs, the ones that are breathing the oxygen that penetrates. And then below that still, we have purple and green sulfur bacteria. If you look at those bands where it's kind of pink and then green down below, those are photosynthetic. They're able to absorb sunlight to produce energy, but they're using a pigment that's different from chlorophyll to absorb that sunlight. which is pretty wild. So they have this bright pink, lovely color. And then finally below that, we have our sulfate reducers. And if you look really carefully on the far right of the tube, there's some little ports sticking out. You can draw water out from inside each of those layers and measure the nutrient concentrations, the microbial activity, and figure out who's living there, what are they doing, what are the microbes up to in there. The other cool thing about Winograsky columns is that they just look super pretty. And so there's all kinds of Winograsky column art. That's actually at the Exploratorium right there on the far right, or on the far left rather. And that's up in New York City, that little bench over there. That's just someone's lab bench who thought these Winograsky columns looked cool. So these microbial communities are forming some beautiful and very valuable functions and things. So pretty rad. The next thing I'm going to talk about are pinkberry bacterial consortia, which are some of my very favorite guys. These guys are two different microbes that are living in symbiosis. So we have purple sulfur bacteria, which I mentioned are present in those Winograsky columns, and sulfate reducers. The purple sulfur bacteria are photosynthetic, so they're getting energy from sunlight, and they're providing sulfate to the sulfate reducers. As a result, the sulfate reducers are breathing that sulfate and producing hydrogen sulfide, which the purple sulfur bacteria then need to use to continue their metabolism. So you can see here on that graphic, they're cycling these different species of sulfur between each other. And the sulfate reducers can't be in the presence of oxygen. So the purple sulfur bacteria completely surround the pinkberries and help insulate them from the oxygen. That's why we have these pinkberries. They're bright pink in color right there. These are a few more photos of them. If I can, this is a tiny little video, but I'm not sure if it'll play of some pinkberries, but if it doesn't, that's fine. On the far right, you can see that's a little dime there. They range from about 1 millimeter to 0.5 centimeter in size. So they're pretty big for bacterial consortia. You can see them with their naked eye. Yeah, big fan. Ooh. Now we're going to talk about another type of consortia. These are AOM consortia. AOM stands for the anaerobic oxidation of methane. And these guys are performing this chemical reaction where they're taking sulfate and reacting it with methane to produce hydrogen sulfide and CO2. And that reaction happens to produce quite a lot of energy. So it's really energetically efficient. But the problem is those two halves of the reaction, the part where sulfate is turning into hydrogen sulfide and the part where methane is turning into CO2, are occurring in two different organisms. So we have the methanogens that are in red right there that are converting methane into CO2, and then the sulfate reducers that are converting sulfate into hydrogen sulfide. And in order for this chemical reaction to proceed, you need the electrons to go from methane to sulfate. So the question is, if these reactions are happening in two cells that are physically separated from each other, how do they proceed? Especially in these bacterial consortia, where if you see this, the red there is the methanogens and the green is the sulfate reducers. They're physically separated in space. And they're actually able to connect themselves using these nanowires. They're literal proteins, teeny tiny little wires that are connecting one bacteria or one archaea to the bacteria. And it's direct interspecies electron transfer. So you have electrons shooting from one organism to another to complete a single reaction to produce energy that helps sustain growth for both organisms, which is absolutely insane. This is what those nanowires look like under an electron microscope. So the A is for the archaea, that's the methanogens, the H is the sulfate reducers, and all those little lines you're seeing are all the nanowires that are connecting them. Yeah. And then the final group I'm going to run through pretty quickly is cable bacteria. They do something very similar to the AOM consortia with the electron transfer, except this is one organism. It is not two different species, but it is connecting two different half reactions of a single whole reaction. So again, we have hydrogen sulfide reacts with oxygen to produce sulfate. The problem is hydrogen sulfide is only available in those very deepest layers of the sediment, and oxygen is only available in the very top layers of the sediment. So how do you get those two to connect to each other so you can produce sulfate and get your energy? The answer is once again, electron transfer, but the way these cable bacteria do it, it's only the very bottom cell is alive. And the rest of the cells tacked on top of it, connected to the oxygen zone, and it transfers electrons along the outside of the cells. And so they're moving up the sides of the cells to go up into the oxygen zone and allow this reaction to proceed. And this is what those look like. If you see these little grooves in this SEM image, those are the grooves where the electrons are transported. Yeah. So these guys are pretty wild. So this is cell-to-cell electron transport. The other cool thing, I briefly mentioned it, is that, again, only the last cell is the one capable of growth and cellular activity. The rest of the cells are just along for the ride. They're not really doing anything related to growth. They're just there to act as a conduit for those electrons. And yeah, so just as a general summary, why do these marine microbes matter? The structure and the function of these microbial communities has a substantial impact on global nutrient cycles, on both the carbon cycle and the nitrogen cycle, and help us mitigate the impacts of anthropogenic climate change. And the other sort of subsidiary importance is that all these unique metabolisms that we're observing, that we're constantly discovering these microbes are capable of doing, allow us to find potential ways that life could manifest on other planets. So there's a lot of astrobiology involved in this, and we can try and figure out what life might look like on another world where oxygen isn't available. How might they be utilizing these metabolisms? So that's my talk. That's the general summary of why microbes are cool and why you should care about mud.
About 3 minutes for questions.
Yeah.
How does a cable bacteria form? Because I'm picturing the only living one is at the bottom, and it can't actually do its cycle without oxygen, which is at the top.
Yeah, so what is weird, you have to look at the two half reactions. It uses water as a subsidiary step. So it takes the hydrogen sulfide and the oxygen that is present in water and uses the oxygen and water to oxidize the sulfide and produce sulfate. And then it has to do something with all the electrons that are produced from that, and that's where it transports the electrons up into the oxygen zone. And the oxygen acts as what we call a terminal electron acceptor that takes that oxygen and is converted into CO2 ultimately.
Thank you. I guess I was asking from a organism level, how
does it like grow?
Yeah, exactly, until it gets to the place where it can dump the extra.
Yeah. So my understanding of how they grow is that they basically grow horizontally. How the first one evolved, I don't know.
Okay, now that makes
But
sense. Thank you.
yeah. So the one that's dividing, it isn't dividing upwards, it's dividing this way. And so it has more of those cable bacteria.
Thank you.
Yeah.
I also have a question about cable bacteria.
Yeah.
Okay, so the one on the bottom is the one that's living, but then are all three of those cable bacteria that you had highlighted in red?
Yeah.
Are those all alive, or is it just the top one?
The top one is also not technically alive. It's not, it's the top three are the only ones in the oxic zone. That's why they're highlighted in red. It's only the bottom one that's doing any sort of cellular growth. They are connected to each other, so they're like little channels between the cells that do connect like the cytoplasm of the cells together, but there's not much activity happening in those upper cells. It's really only in the bottom ones. Yeah.
So where would this fall in the redox? Because it's getting oxygen, but it's down in the sulfate.
Yeah, so this is kind of an exception to the redox cascade. It's connecting two disparate zones of the redox cascade, which is why they're kind of cool. So you have multiple different metabolisms happening in the middle. The only reason that this reaction is actually able to function is because it runs out of oxygen and the oxygen is separated from hydrogen sulfide. Because otherwise sulfide will just react with sulfate or with oxygen naturally, and you won't be able to grab any of that energy.
Great, thank you, Rachel. Right. All right, next up, we have Duncan, who is a first year in the Shark Lab, and he's here to talk to you about white sharks in Monterey Bay. All right.
Hey guys, how's it going? All right. So this is originally a talk that I gave at the Northeast Pacific Shark Symposium, a shark conference up in Seattle. In its original form, this talk was a lot faster and designed for PhD shark scientists who've been studying this exact species for 30 years. So I'm trying. I may accidentally use a couple of jargony words here, and so please feel free to ask for clarifying questions at the end. But so I just want to give a quick rundown of some shark research in the area, and talk about some open questions in shark research. I think people get this impression sometimes that we know a ton about sharks, but we only know a ton about sharks in limited areas. And so it's really fun to get to be able to bring to the public some of the stuff that we talk about only really in academic circles about what we don't know about sharks. And this is all research that we do here locally in Monterey Bay with our shark populations. But this research does have implications for the entire planet because white sharks especially are a globe-spanning species. So starting off, I am primarily talking here about what the public knows as the great white shark, but scientists refer to it just as the white shark because there is no lesser white shark. So white sharks are not only one of the largest shark species, but they are also one of scientifically the most interesting shark species. They have a number of very unique traits to them, and they're very recently evolved species. They're not the most recently evolved species, that's hammerheads, but they are one of the most recently evolved shark species. And so this makes them very interesting because they have a lot of characteristics that we don't see elsewhere, and they have a lot of characteristics that are very unique to them because they aren't related to similar traits that you might see in other organisms. And so we frequently track and tag white sharks in Monterey Bay. There's a very large, healthy population here, especially a large population of juveniles that moved in after 2014 and the blob, and a very large, healthy population of adults that lives up in Año Nuevo and goes after the elephant seals. So we're really good at tagging, and this is my advisor poking a pop-up tag into a, that's a juvenile white shark. He's about 11 feet long there. He's kind of a baby. Honestly, he's only a few years old. He's at most a teenager. But it's very difficult to study a lot of these physiological processes, these internal processes of these organisms. So my thesis focuses on digestion research. And so this is interesting. Digestion and metabolism, the processes revolving around how animals, absorb and utilize energy. There's a lot of processes in that, right? And so we know a lot about things like the shark diet and the shark's behavior because we can track those very easily. We can watch them eat things. We can watch them move around. We can cut apart dead sharks when they wash up. So we know a lot about what's inside of them. And we know a lot about how they move, where they go, things like that. But what we don't know a lot about is what chemically and biologically goes on inside of one of these sharks. It's this big open question. How in the world do you look at the chemistry of a shark's stomach? And how in the world can we study these processes? This is not an animal that you can keep in a laboratory. This is not an animal that you can get to cooperate like a cow or a mouse, where you can keep it in a lab or you can keep it in a field and you can work with it to see what's going on. Especially not something like a person where people are great for studying internal stuff. They're very cooperative. You can pay them money and they will give you data. If I could pay the sharks, my life would be so much easier. So we know things about their diet and we know a lot of general characteristics of digestion from the outside, but the inside of the shark is very much black box. We know things like, we know how fast they digest food and we know temperature. And we've done this in a very interesting way because my advisor, about 10 years ago now, when he was still working for the aquarium before he moved to CSU Monterey Bay, went out with a collection of tags designed to be mounted on the outside of an animal and track the temperature outside of an animal. So eventually like a thermostat and a radio receiver so we can find it again. Designed to be clamped onto the shark's fin. But he said, well, it's waterproof. It's chemically inert. It doesn't, react to anything. So let's just cover it in blubber and throw it at the shark. And when the shark's finished with it, we can see what's going on inside the shark, right? So this graph is actually from that experiment. This is the timeline of temperature. After the shark's fed, you can see that the shark's stomach spikes up in temperature. It goes up to 26 Celsius, which is roughly 75 Fahrenheit. And then it decreases down over time as the animal digests its food. And then when it eats something new, that temperature plummets down and then cranks back up again. It's worth noting, this is in Año Nuevo. This shark is in very cold water. It's maybe 50 degrees in the environment it's in the outside. So this is significantly elevated. And of course, sharks are fish. Fish are cold-blooded, right? White sharks are one of 3 species of warm-blooded sharks. That's one of the interesting characteristics I mentioned. So, a warm-blooded shark, how does that work, right? We don't actually know. How are sharks warm-blooded? We know it's in their temperatures elevated in their stomach, but we don't know where that heat comes from. There's 2 theories. We don't know to what extent each of them is true. They could both be true in different amounts, or it could be one or the other. The first thought is that the skeletal muscle, essentially their red muscle in the shark, the muscles along their spine, and the core of their body, like their heart, the big powerful muscles that help them move, the muscles lining their stomach and their intestines that push food through their intestines. These are dense, very strong muscles, and they produce a lot of heat just through the natural process of movement. If you go for a run, you feel warmer. That's because those skeletal muscles are working. So in the shark, these muscles also produce a lot of heat. And in most fish, they would be dissipated. They would just kind of work their way into the environment and not raise the temperature of the animal. But in the shark, these white sharks, they've adapted to retain that heat and keep themselves warmer because this makes them more efficient. It's like letting your engine warm up before you go drive somewhere. It increases your mileage. Same thing with the shark. But the other theory here is that maybe it's coming from inside the stomach. When you digest food, you produce heat. The chemical reaction of breaking down dense materials into simpler materials that you can digest more easily, as happens in your stomach, produces a lot of heat. In some animals, this heat is very minimal because it's a slow process. That happens in a lot of fish. In some animals, this is a very fast process and it can produce a lot of heat. You could take an entire college course on just this process. So there's a lot of details we won't get into here, but essentially there's these 2 processes that can produce heat inside the animal, and we don't know which one of these creates the heat. We don't know if the shark is putting heat into its body, or into its stomach to warm up its food, or if the shark is producing heat in its stomach and pushing that heat into the rest of its body. It's probably a mixture of the 2, but we don't have a way to measure this right now. So it's no fun to just talk about things we don't know without also telling you how we're going to figure it out. The whole point of science, right? So how do we study digestion in these adult white sharks? The normal processes don't work. With a lot of organisms, you can take it and you can put it in some kind of medical scanner. You can put a fish in a CT scanner, and if you add some contrast, you can watch food move around, you can see things happen, right? A white shark does not fit in a CT scanner. I asked a radiologist, they said I wasn't allowed to. With a lot of terrestrial animals like cows, you can put a cannula in. You can drill a hole in the animal, and you can put a little port in, and essentially just like an ostomy bag, you can take samples out and you can measure them. But this only really works for cooperative animals. This works really well for humans. This works really well for big animals we can restrain, like cows. This works very poorly for animals that will migrate 2,000 miles in a few months and spend the entire time underwater. Also works really poorly for animals that will bite you if you try to do this. So these processes don't work very well. But what does work is, like I said earlier, feeding him some kind of tag. So we can feed a shark some kind of tag that can collect a fluid sample inside of its stomach. And then we can use modern techniques, looking at the DNA present in that fluid. We can look at chemical processes, just like in someone's sediment microbe study, like you just heard about. We can do these sorts of techniques on these fluids, and we can see what's going on. And once we've made the right measurements and we know the right characteristics, we can make these inferences and we can answer some of these questions. And this will tell us things like, how did white sharks evolve? What sorts of environments did they evolve in? And what sort of responses might they have, evolutionarily or behaviorally or physiologically? What sorts of things might they do in response to upcoming climate change? There's a million more questions we could answer, but these are just a few of the things we could do with this. And so we're looking into these emergent techniques. And this is the sort of research that only happens in a place like Moss Landing, where you can bring together the right mix of specialties and the right people willing to encourage out there research, like I want to feed a machine to a shark that will suck up its stomach juice, to get these sorts of answers. And it's just, sorry, this is my project. I'm really excited about it. And just wanted to share a little bit of the kind of exciting things that happen at Moss Landing with you all. All right. A few references. And I'll have time for questions, but thank you very much for listening.
Questions, ask away.
All right.
Are the tags basically collection devices that you pick up after the shark poops it out?
Yeah, so it's actually really interesting. One of the many weird characteristics of white sharks is they don't actually poop out solid matter. Because they eat so many large bony things, like seals, a hard chunk of bone like that traveling through the digestive system would cause them a lot of harm. And they can't chew off flesh like a big terrestrial predator could, like, a big cat or a wolf or a bear or something can pull off the food it wants to eat, the soft tissue, off of a carcass and then leave behind bones. But a shark can't do that. So the bones in its digestive system would hurt it. So instead, they trap all the bones in their stomach. And then when they're finished pulling all the soft materials, the meat and whatnot, into their intestines, they essentially just vomit up all the bones. This makes it really easy to recover a tag, but it also makes it interesting to talk about. I think people don't expect that a lot. It's really interesting. Yeah. Sure.
Well, you may have just answered that question, but I wondered if sharks chew.
So some sharks chew. White sharks, what I've been talking about here, do not chew. They essentially just slice. There are sharks that slice off big chunks of their prey and swallow it. There are sharks that will grab onto prey and essentially just like shake and thrash until a chunk falls off and then they can swallow that. And then there are sharks that have like grinding plates, similar to if you've ever looked inside of the mouth of a stingray, and they can crush their prey. But the white sharks I'm talking about here don't chew. Other questions?
How do you recover the tags?
We have various methods of recovering these tags. The current system we're working with is a system of a satellite pop-up tag so when the tag floats to the surface, it has some foam on it that makes a point. When it floats to the surface, the tag sends us a satellite signal telling us its GPS location. And then when we go out there, it has a little radio beacon on it so we can narrow in and find it on a boat. Sometimes they wash up and we can just pick them up off the beach. A few years ago, maybe 10 years ago now, one of my advisor's tags from the project I mentioned with the graph I showed floated all the way to Hawaii. And a 12-year-old boy there, I believe, you can look up this news story. A 12-year-old boy picked up the tag on the beach and called him and mailed it back to California. And this gave us huge insights into how the sharks actually started migrating. It's a really interesting story. It looks like I'm running out of time. Maybe one more question.
One more question, go ahead.
Yes.
What kind of volume can you collect and can you time when you take the sample or does it just auto?
So we're still in the developmental phase of these tags. We haven't gotten any final designs yet, so we're still working on that. we're hoping for very small samples, a few milliliters of fluid at a time, but with a computer controlling when it collects it and multiple collections at a time. So we can set it as we.
Another round of applause. Okay, well, now we have our final talk. We saved the best for last. I am happy to introduce someone who is near and dear to my heart, arguably one of the coolest people at MLML, Olivia Bedoin. Thank you, Olivia.
Yeah, thank you.
All right, thank you for sticking around, everybody. Appreciate it. Thank you. So I am here to speak to you all today about clams, because why? They rock. Specifically Pismo clams and their geological habitat. A little bit about me. I am Olivia. I'm a first year, so I'm just getting started with my thesis, and it's been a great ride so far. I'm co-advised by Dr. Allison Haupt and Dr. Amanda Kahn. So let's get into it. So you may be thinking to yourself, why clams? Clams are better known on our dinner plates or in our soup, but what do they do for the environment? I could have been studying white sharks. Why did I choose clams, right? But I think they're really cool. What do? So different marine organisms occupy different functional groups. In this case, bivalves are occupying the functional group of biodiffusion. So this is a part of bioturbation where sediments get mixed up, because without the sediments getting mixed up, they would be anoxic. So we need some oxygen, we need some water flow to get clams in there and more species in there. Clams are great for moving sediments around and getting this oxygen in. So as you can imagine, more bioturbators means more sediment mixed and more O2, which means more habitat and more biodiversity. Clams are a huge important driver of biodiversity in soft sediments. So more on their ecological importance. They participate in water filtration. They can filter up to 60 liters of water per day as they are suspension feeders. This is 22,000 liters a year, which is an incredible number. They are also important for higher trophic level groups, such as otters, bat rays, moon snails, and more. So a little bit about their habitat. Has anybody been to a really steep beach and looked at the sediment grain sizes? They're really big. Have you ever looked outside and looked at the water crashing in? There's so much energy happening at these steep beaches. The opposite for flat beaches, right? We have a lot of flat beaches, smaller grain sizes, less wave exposure. So this is kind of the characteristic of how soft sediment habitats can vary, even in just Monterey Bay. So soft sediment habitats occupy 80% of the seafloor with varying degrees of mixture. And what I mean by mixture is the amount of different grain sizes we have. So a sediment that has a lot of pebbles, but then a lot of small silts would mean it's poorly mixed, if you can get my gist. The structure of this habitat can dictate the abundance of fauna within. So these grain sizes are dictating what fauna goes there and how much of them they go. So infaunal invertebrates means that they are within the sediments. They have an optimal grain size that they want to use. So as I mentioned with beach slope, the larger grain sizes we get means the more higher wave exposure is happening and the steeper our beaches will be. This metric relates to how much habitat is available for species who inhabit the swash zone. And you may be like, what is the swash zone? So I'm here to tell you, it is this red box that I have highlighted. It's in between the run up limit and the run down limit, or just in between where the high tide ends and where the low tide ends. This is where our team studies. Yeah, so we study in this area because this is where a lot of these bivalves are going to be. These bioturbators are mixing up the sediments. You can even see here, this is just an example of how it can vary within the swash zone. So you can get a lot of large particles like you see in the sieve right there. And then you can get a lot of small particles as well. So this is an example of a poorly mixed swash zone. So you may be wondering, what's happened to the clams? What's happening now? So here's a commercial fishery timeline. In 1916, we have the opening of the commercial fishery where you can see thousands of pounds of Pismo clams being fished in up and down the California coastline. And you can see now the population has declined all the way until the closure of the commercial fishery and the opening of the recreational fishery, which had to happen because of their overexploitation. So the recreational fishery is still happening in Monterey Bay and up and down the California coastline. Monterey and Santa Cruz, we have a limit of about 5 inches you can take. South of San Luis Obispo County, you can take about 4.5 inches. They're relatively more abundant down there, so you can take smaller sizes, but it's still happening. But illegal take is also still happening. In this picture, you can see an illegal take of hundreds of undersized clams at Santa Cruz County, just down the road. This happened in one day. The largest clam was not even legal size. This was taken from the CDFW, I believe 2 years ago. This is happening all on the California coastline, and it's really important that we keep looking at what's happening. So what are we doing about it? So we need to make population models. We make these for fish. We make these for any invertebrates in the ocean and in terrestrial environments. So this is a way in which we study how a population grows or shrinks over time, and this is controlled by birth, death, and migration. And this has a lot of important impacts for economy and conservation. So what about Pismos in Monterey Bay? Not much is really known. Monterey Bay in particular lacks extensive population, abundance, or size location statistics on a fine scale. Significant number of individuals have been found near Santa Cruz, California. Before I started the project, they found 56 in 2022 and 23 in 2023. So what's most important is for the restoration of these populations to occur, there must be a continued monitoring effort to keep an accurate record of their population sizes. And we must be identifying these physical drivers of their distribution. And I'm interested in the particular physical driver sediments. So we selected some sites. These were sites recommended by Cal Poly SLO's Ben Ruttenberg. So he used grain size as a metric to recommend these sites for us. And a few of these have already been sampled by Cal Poly SLO. In green is where we have already sampled last year. And in yellow is where we want to conduct some future sampling. We want to conduct this sampling on a super fine scale. So that means we want to sample at as many beaches as possible. So this is what our transect looks like. This is called a perpendicular transect. So that means we are starting at the high tide watermark where the water is wet and going as far as we can into the water as 30 meters allows. At times we don't make it to 30 meters because our transect just gets washed out. But we study this in 3-meter increments, and we shovel lots of sand into our gorilla carts to see for clams. And that means lots of shovels, lots of shovels, lots of shovels for lots of sand. So, and that means a lot of volunteers. So if anyone wants to help out, feel free to contact me. We also collect beach slope estimates with what's called a differential global positioning system, or a DGPS. This has an accuracy of 2 centimeters for elevation. So we're trying to get a fine scale metric for beach slopes, as this has been indicative of where pismo clams might have been found in the past. So as you can see, we're out at all times. We're dictated by the tides. So you'll see us at 5:22 a.m. or in a nice afternoon like that. So this is what it looks like, a day in our life. This is us shoveling some sand into the cart and pushing it into the water. And we have to get all that sand sieved out to see if there's clams inside. Yes, lots of fun volunteers and lots of fun days. That's a pismo clam. As you can tell, we have so much fun. Follow us on TikTok. So what have we found so far? Let's see. So last semester, I started my thesis, and I started at MLML, and I took a geological oceanography class with Dr. Ivano Aiello. And he pushed me to study sediments. He was like, you have to study rocks for our class project. And I was like, why not sediments? So that's how I got started with that. We sampled at Sunset State Beach last semester, Del Monte Beach, Island Beach, and Sand City Beach. And all of the morphologies of those different beaches were extremely different from one another. As you can see for Sunset State Beach, we sampled there twice because they're known to be found pismo clams there in the past. And with the asterisks there, that's where we found them again. We didn't find any at the other beaches. Lots of zeros in our data, actually. So as we can see in pink, the smallest mean grain size means it was more well mixed and it had the flattest slopes. So all of those correlated with one another. The largest grain sizes in blue were poorly mixed and had the steepest slopes. And lastly, in red, the flattest slopes were where we found the clams. So we are thinking that clams will be found on these flat beaches like we see in San Luis Obispo County and south of there as well. So this is data that was taken before I joined the lab. Something I want to point out is recruitment. Recruitment is super important because this means that there's a lot of young individuals able to sustain that population in the future. And that's a metric for a growing population. So as you can see, the line in green is where our recruits are. The size classes are on the bottom and the proportion of clams found was on the y-axis. So as you can see, not many recruits are being found. And that's really important because we want to make sure this population is stable and growing. There's some in 2019, some in 2022. You can see those size classes growing with one another as we look further on in further years. But 2023, there's no recruits. So we are really concerned about that and want to keep measuring how much clams are in the area. So all clams we found were not of legal size for harvesting too. So you better not see anybody take clams from here. This is really important because these smaller individuals are again going to be really important to sustain future populations. Okay. Moving forward, we have to make recommendations for CDFW due to the fact that we are not finding these legally sized individuals for harvesting. We would want to enforce stricter protocol as this is where the younger and smaller clams will be located. We are near the northern limits in Monterey Bay. Two scientists who have studied pismos in the past, Coe and Fitch, describe that northern populations of pismo clams take 2 years to sexually mature rather than 1 year in regards to their southern populations. So it's super important that we keep studying here and conserving those smaller guys. We also just want to fill in the gap of knowledge of Monterey Bay. They're here, so let's find them, you know? So moving forward, we will be continuing to use sediment grain size and beach slope as indicators of habitats for clams. We will be collecting and comparing seasonal data as sediments will change compared to wintertime and summertime. We want to continue to sample at the limits of their historical distributions up here past Santa Cruz County and all the way down in Baja California, where I will hopefully be going next week to sample. Let's hope the paperwork goes through. We also want to compare densities along the California coastline. San Luis Obispo County has an immense number of pismo clams, thousands and thousands. So why are we not seeing them up here? What's wrong with our area? So we want to be adding in more physical drivers and more biological drivers of their distributions to study. These include things like harmful algal blooms, salinity, temperature, POC, etc. And with this project, we want to continue to create opportunities for undergraduates. As you can see with this, I have a large number of undergraduates helping me with my work, especially Ilya and her dog Tao over there. We have a great time. I want to thank Dr. Allison Haupt, the PI for this project, Mariana Jimenez, who is presenting some of this work down in Cal Poly SLO today, and Dr. Ivano Ayello for pushing me to go with rocks, because clams rock. In any case, I also want to thank Moss Landing and Cal Poly SLO for starting this project. The Ruttenberg Lab made lots of great recommendations for us in our methods. And I want to thank Coast, CSUMB, and the Myers Trust as well. Thank you. I'd love to answer some questions. Sure.
So your gorilla carp, what stops the little ones from washing out?
Yeah.
And Moss Landing, I see all these little shells that look like that.
Yeah, we were really concerned about that too. So we had to get a really finer scale mesh to put in the carts so that those aren't slipping through, because we want to count those guys. Those guys are the most important. Yeah, I agree.
I find them at Moss Landing, but they're already eaten up.
Oh, no. Yeah, I do see a lot of shells there. We're actually looking at shells as well as indicators of where the clams might be. At Island Beach down here, we find lots of shells, but not lots of clams. So we assume that they're farther out in the subtidal. So
can you take some from Southern California and bring the little babies up here and plant them?
that's something that we'd have to talk to CDFW about, but that's exciting. I hope that's something that we can do. I know there's aquaculture practices going on at Cal Poly SLO too. They're trying to start that up, so that's exciting. Yeah, sure.
It seems like 23 or 56 is a really small number of clams. So if you find one, do you just put it back in the same place where it came, I mean, the same transept?
Yes, we want to make sure we take the GPS point where they are
Yeah.
and put them exactly where we found them. We do take some specimens based on our permits for histological analysis, and we take it back to the lab, analyze the molecular data as well. But we would like to incorporate mark recapture in the future. So you might be seeing clams with little tags on them in the future. Yeah, sure. Say that again? Yes, they can. They're burrowing. So as I said before, they're bioturbators. So they're the ones that are moving the sediments around. They're the ones that are oxygenating the sediments so that more species can come in because they need some oxygen to breathe too. So yeah, sure.
What do you think is the coolest animal besides clams?
What's
your favorite marine animal?
You know what? I think penguins are, yeah, I agree with penguins. Some people get to go to the Antarctic to look at penguins and I think it's really cool. Yeah. Yeah. Any more questions? Yeah, sure.
Besides, like overfishing and
Mhm.
potentially poaching, what do you think are other environmental factors that would affect their population decline?
Yeah, I think it could also have to do with higher trophic levels as well. I know sea otters were a big predator that were exploiting them in the past, and it's really important that we look at those higher trophic levels and incorporate maybe some subtidal data as well, where they're actually feeding on them, where we can't see them. Yeah. Any other questions? Yeah, sure, in the back.
Yeah, is there a historical record of having larger clams in this area? I know there's a lot of Indian midden.
Yes, there's a booming population up here in the past until around the 1990s up here, where they were overexploited. But yes, there was a lot of larger clams that were legally harvested in the past. Yeah, we have a time check over here.
No, you have plenty of time. We can
Okay.
break after some
Awesome.
questions.
Yeah, I would love to answer more.
So if I find a clam up on the beach,
Mhm.
just messing around, I'm always playing in the sand.
Sure.
You want me to put him back in there? I would like to throw him back in the ocean so he can just burrow wherever he wants to
Yeah.
and find a new home.
That would be awesome to think about, but that would also mean that they're out in the open for these other predators to come in and just see, hey, there's a clam right on top of the sediment for me to take up and I'll eat it. But burying it back in where the soil's wet, and they're usually found about 7 to 12 inches down. Yeah, sure.
Who exactly are you finding is doing the illegal clamming? Is that for restaurants or for? I mean, what is it for?
I'm not sure. I know it's just for recreational fisheries, so it might be these single individuals, 1 or 2 people coming and sneaking in. You saw in that picture, it was just a truckload of hundreds of clams. So it might just be these normal citizens coming in, harvesting clams for themselves and their family. Not sure. Yeah, yeah, sure.
So you said you were looking into maybe other biological and other types of factors as well,
Yeah.
physical factors. Is there any precedent for ocean temperatures, like water temperature that have an impact?
Mhm. That's one of the hypotheses right now. One of the reasons why we think that there's more clams being found down south is because it's a little bit warmer. Hopefully with some Baja data, we'll be able to confirm or deny that hypothesis. So we'll let you know. Any other questions? Yes, of course.
Not a question exactly, but are you familiar with Lincoln's program?
I'm not familiar, actually, yeah.
Okay, so you're talking about looking for volunteers. I'm a high school science teacher in Marble.
Yeah.
Limpets is a. Yeah.
Okay.
They get high school students
Mhm.
to come volunteer and monitor the sand crabs.
Wow, okay.
So if that's something, if you're looking for volunteers, high school science students need to do community service and they love it. They understand the transects. They teach them all that through Limpet. So
yeah,
for what that's worth.
Yeah.
It's something to look into.
Let's get in contact.
Did that program.
Oh my gosh.
So I was one of the young women in science at the Monterey Bay area,
Nice.
and that was like one of the projects we did, is we partnered with the Limpet program, and we went out there, and we did exactly what you guys were doing.
That's so cool.
And I had a lot of fun then, and I had a lot of friends who did it too, so.
Yeah, come on out. Come on out. We need more volunteers. Sure.
I'm actually Limpet, so you can.
Okay, I'll be right over here. Come get my info. Yeah, any more questions? Well, in any case, I want to thank you for listening, and now we have a break for our students. I think we're live. Hi, Mom, if you're watching. Hi, Carly. Hi, Florida people. I miss you all. Hi, Bedoin family, if you're watching. Oh my goodness. Okay. See if I can. I don't know. I'm not the tech person for this. All right. Well, I'll go find someone. Thanks for listening, everyone.
So I.
Wait.
That's why you're gonna.
Yes.
Just in case.
Yeah.
Sorry. 12 minutes.
Yeah, it's the best.
Someone commented on this.
Yeah.
I don't know if it's been like.
Can't wait for CC to restart the summer again. I bet you can't wait to.
Just keep looking at this. Like.
Yeah.
Because they can't see, they only see the speaker. So. Yeah. Stay. And they didn't put their questions through. Oh, you can see that. Hey, hey, you can see that? Yeah. 7,100.
That's incredible. Yeah. Yeah.
So I was. And I was like, what? I always thought she told someone to talk to that pizza for.
Yeah.
Last 10 years, you know?
Hi, live stream. We are about to have a student panel in about 10 minutes. So actually, no, it's at 4 p.m. Sorry. So stick around for that. We'll be chilling in here for now. Thanks.
I'm still here. Well, this is all the live stream. We should have, before you adjourn, you should have like come back. So I meant to tell you, come back at 10:30 for the panel. Okay. Oh, I guess it's like 8 minutes. I thought it was. No, everything. It says on here. It says on here 3:30, so it's 7 minutes. Should we go put some chairs up there? Okay. There are 1, 2, 3, there's
It's actually starting in 7 minutes, folks. We're gonna put the chairs up. Stick around.
I would leave those, I would take 5 for not.
It is.
You're like. That man, I just like this associate also.
There's also.
No, he's even interested next year. That's fine. Great, sounds fantastic.
Have a lot of fun.
Oh my God. I was like, what the, go to the next year.
I wonder if my stuff's sold out.
Right now.
I don't know, but I. Oh, I forget that's on.
Hey, Jonah. Look at you. You're a big, strong man just sitting down watching the females put the chairs up.
I didn't wanna. Before you, but you're right.
Fish exactly, is this fish?
That was
The
so
fish picnic station, I went, it's
oh, this is really weird.
How many are there? 5 of us? 5 of you.
Okay.
But Taylor is on the panel, but she also has to be a moderator. because There's no moderator.
I mean, yeah, but do we have a mic that's like handheld mic?
People have been talking loudly. Do you want us to hit the mic?
I don't care.
The mic is in the kitchen.
We don't need
So
it.
if you guys.
We can talk loudly.
Okay.
I mean, it's Taylor, Roxy, me, Bruno, Carol. Carol just has to project, that's it.
Yeah, Carol's good.
Bruno has some pipes on him.
That's it. I think someone just saying. You saying? You got questions? Where are you going now? These are the notes that starting at 5.
Oh, if you talk to any undergrads today, you should tell them like take it away. I think I might go tell some of them that.
There's a few congrats from.
I'm gonna go start working on my undergrads, stay into the break to start listening to the panel, that could be very helpful. Yeah, send them all here. I'll go do that. Yeah. Oh, yeah, perfect.
We have a question. Will the graduates think, what is?
So we're just actually basically sit up there and we're gonna kind of alternate between some predetermined questions and then take questions from the audience. So that's whatever you want. Yeah, whatever you're curious about our research, if you're curious about that. Graduate student life. Any of it, all of the above. Okay. Yeah, there's we'll pass around on like bios on everyone and talk about, you know, if there's a person. Whatever it is, so. Yeah, of course.
Taylor, do you need more students?
No, we're fine. We've too many. We need.
Decided to not go to the Strandings.
Okay.
If you get more calls about the one over by the Tides Hotel, you can go to that one, but the other one I already responded to.
Okay, awesome.
Good luck with the rest of today.
Perfect,
It's like,
Raven.
yeah, I'm watching Sierra's dog. It's been all day. Yeah, blackberry, blueberry, blue.
Whatever its name is.
The anxious dog.
Yeah, I do not envy.
Will be missing me so much. So I gotta go.
No,
Go save her.
Okay.
See you later.
See ya. Oh.
I don't like how it follows you.
I don't like the owl either. Look. So I think we're still live. Custom live streams.
Yeah, we still are.
Okay, great.
Can they not, can they comment?
Come sit over here. If we're not here, I wonder if it will just.
Was there stuff in the chat? Question for you about this, white shark research?
I think it's just showing us.
Not the screen.
But anyway, there's my research on the.
I don't know, the owls still bad.
What's up?
Let's move this onto the other screen and see.
Where's everybody?
What do you mean, where is everybody?
For the panel.
It doesn't start till it starts in 2 minutes, actually.
Yeah.
I mean, we'll
to the
just
undergrads.
wait.
I'm gonna go till the undergrads.
I sent a message.
Yeah. I'm lucky that they're paying taxes. It's a lot of it, it does look like you have to like take money, but it seems like you know, like, I'm very sneaky. Yeah.
Our permitting and our research. Have to be also last year.
Oh, the permitting, yeah.
So like.
Oh, yeah, they are.
Okay, here, podium.
Oh.
Bruno, do you know anything about the owl?
No, I'm actually was just admiring it.
I don't know how to change the main view. Oh, I don't know what I just did. It's on podium. Oh, Sylvester's here. Maybe
it's
You
actually.
see, it's on podium, the owl. Wait. maybe
Oh,
it's-
it's.
Hey, no one's here yet, so there's no point.
Are you sure? So that's just still need to come back.
No, we need people in the audience. Okay, no, we don't need anyone on the stage anymore.
Question, yeah.
Oh, she doesn't mean students, she's for the audience.
Oh, we have 5 people, that's more than enough.
Not now.
This is why.
I know.
I'll ask you questions.
Snag a pic of this pen. No, I'm not. I wish I was. Snag a pic of this.
Oh, that looks more better.
Maybe I'll walk the hallways as I leave and be like.
Yeah, it'd be like, there's a grad student panel happening.
Okay.
I sent a message telling people to come. Hi.
Okay, yeah.
You gotta come learn about grad school?
How long is it?
It's supposed to be a half hour, but I think we probably, if people have questions, we'll go longer than that.
What's after? There's no more talks.
Nothing.
Let's go down.
My water bottle.
What do you think's in there?
I know. We're around here, love guys. Brody from freshman year. He was here. Yeah. He's a firefighter, a paramedic, and he's about to be a firefighter. It's like, that's so random.
Yeah, but you're doing your own pictures like a fish.
He was a police secretary.
Yeah.
Yeah. But he just died, but so this girlfriend, I think.
Yeah, I thought that I was a little bit, still have it wondering.
In terms of what?
Yeah.
I know. You did a great job.
Yeah.
Very good presentation.
Yeah, perfect. No, I can be in the audience and be like, I'll be Carol. I know. I'll play.
Yeah. Sarah, get up here, I'll meet you. Yeah, those are moderated.
Oh, is she?
I am.
Oh, nice. Sorry.
I'm just gonna message on the volunteers. We have. I don't know. Do you know? You guys know if they're.
No, we were out there.
It's just a stream.
No.
Everything in the.
I don't think so. Oh.
Less comments.
Yeah, but it's, I don't know, we can do that right now. I don't want to be in the back. Yeah. Thank you for messaging the. I know. Yeah, okay, as long as there is. Another. They're the ones that. Yeah. Yeah. University's gonna cost all mine. Yeah. Those are all true.
Looks like a red hat.
They're really great. Honestly, like, I don't really. No, you can never be back.
I have Brooks and I'm looking for like a new show.
Yeah.
Or not Brooks, these are.
Yeah, that's okay. I just messaged the, you already talked to the volunteers. Yeah, I just went and took. Yeah. Okay, I sent the message. It's gonna be. So leave the door open. Yeah, everyone close those doors. Okay. Do you hear anything? Or are you okay, Margaret? Yeah. Okay. Should we start, please?
Yeah, let's start.
Okay.
Thank you for coming, everyone.
Coming, I'm gonna moderate and I'm also gonna talk. Sorry, you're hearing a lot from me. So we're just gonna go through, the point of this panel is we're gonna alternate answering some questions that we thought people might wanna know. And then also giving you guys the opportunity to ask anything you want about grad school or our research or anything, basically. So we're gonna start introducing ourselves, and just go through our name, you know, who we are here at Moss, what lab we're in, and what our thesis focuses on, and then, pronouns, we'll do, our hometown, our undergrad. That make sense? Yeah. Okay, I'll start. My name is Taylor Aziza, my pronouns are she, her. I did my undergrad at the University of Idaho, so I'm natural marine scientist. And I'm originally from Maple Valley, Washington, which is near Seattle. I'm a third year student in the vertebrate ecology lab, and so my thesis focuses on emperor penguins. And so I don't know, you guys probably weren't here earlier, but there was a talk at 10 a.m. about penguins. Yeah, should we just pass this down?
So my name is Roxanne, my pronouns are she, her. I am in the phycology lab, which is study G, in case you don't know. I study, or let me go back. I went to my undergrad at UCLA and I majored in biology and minored in Spanish. And I'm looking, my hometown is Greenfield, which is not too far from here, it's in South Monterey County. And I'm studying how giant cell is affected by disturbances and looking at how its growth and reproduction change off.
Hi, everyone. My name is Kelsey. I am a second year here at Moss Landing in the ichthyology lab. That's studying fish. And I'm also co-advisor over at CSUMB. I did my undergrad at Fresno State, so also not necessarily marine-related university with a major in biology. And I have two hometowns, Hermosa Beach, California, and Oakhurst, California. My current research revolves around the abundance and behavior of juvenile white sharks in the Monterey Bay.
Cool, my name is Bruno, I'm he, him. I'm a first year here at Moss Landing. I went to undergrad at UC Santa Barbara, and I grew up in San Diego. I'm in the fish lab as well, and I'm still trying to flesh out my thesis, but I'll probably be looking at nitrogen excretion rates of fish and how that changes with temperature.
Hi, everyone. My name is Carol, and I'm currently a second year here in the Chemical Oceanography Lab. I did my undergrad at CSUMB, so I'm pretty local, and my hometown is San Jose. For my thesis, I'm trying to create a iron detection method on an automatic microfluidic analyzer. So all it really means is we're trying to automate chemistry with a machine in my lab, and I'm trying to measure iron using that machine. Yeah, fancy words for kind of, yeah, something.
So I'm gonna passing around, just a sheet with basically what we've just said. If you consider your period, that should help maybe direct the flow of conversation. But we're just gonna start with very basically, how did we all end up in marine science? Like I said, I'm just in Idaho, so I was never, ever thought I would be a marine biologist or a marine scientist at all. I studied wildlife biology and then I went abroad and I just wanted to do science. And so I got involved in a marine science lab doing we work with right whales, and here I am 4 years later. So I don't really know how to do this, but yeah, I'm doing my master's here, and then in the fall I'm starting at Oregon State to do my PhD in wildlife science with a thesis on the drivers of unusual mortality events in gray whales. So I did not mean to end up here, but I've gotten very deep, so it's no turning back now. Yeah. So no, what, yeah, I think that's much.
So I originally, after I graduated from UCLA, I actually thought I was going into healthcare. Then I decided I didn't really want to do that anymore, and so I kind of switched to conservation, and I have taken some sort of different classes at UCLA that I really enjoyed. One of them being conservation bio, and so kind of switched gears at that point. I also worked at the aquarium industry, I guess, for a little while, so worked at the National Aquarium in Baltimore for a few years, and I wanted to try out research, and so that's kind of why I ended up in this work.
So my biology program at my undergrad was very pre-med heavy. I did not like those classes. The only required classes that I did like were the ecology-based ones. I knew that I liked working with animals and out in the field, but being landlocked didn't really appeal to me. So I turned my focus to more coastal. Plus, there's so much that you can learn from that. So I got involved in some third-party research programs out in Florida to do some hands-on field research on sharks and rays, and I haven't turned back since.
I didn't think I was gonna be the only one, but it seems like I'm the outlier. I always wanted to do marine biology. I feel like if you commonly hear everyone say that at some point in their lives, and I guess I just didn't change my mind. So I went to Santa Barbara with the goal of studying marine biology. I took some time off after my undergrad and I got some professional experience, and I worked up in Alaska on commercial fishing boats, doing fisheries observing. Came back to San Diego, worked at Fish and Wildlife, got to really see the ins and outs of fisheries management. And then always wanted to go to grad school, but after COVID, it was just time. But kept searching for new opportunities, and they're harder and harder to come by as time goes on. So here I am.
Yeah. I was similar to Bruno, where I also knew I wanted to do something in marine science, so hello. But honestly, marine science is such a large and broad field. I didn't know what exactly I wanted to do. I knew I didn't want to do anything biology related or living related. So as a sophomore, I actually interned here in my lab in the Chemical Oceanography Lab, and just never really looked back since, and here I am a few years later. So.
We all only look forward here.
Yeah.
Nobody looks back.
Yeah, there's no regrets for any of us.
So we'll take an audience question, and if no one has any questions, we'll keep going with ours.
Yeah, but I have a question. The jobs, when you graduate, are you hoping to work
in a government agency, in a conservation group, or like fishery management? Where will you be heading on to, you think?
I'll start, a job. Yeah, like I'm going on to do my PhD, so at that point you can kind of go the route of moving back into academia, and being a professor or researcher, or kind of steering into, government, nonprofit research. I don't want to keep going. I've been in a lot of school, so I'm ready to kind of pivot into something that has a salary, and lets me live somewhere on the coast and, not inland, not way long. So yeah, I'm not someone who's like, I have to work for NOAA or I have to work for Fish and Game. I'm open to different things and excited to finally be done with school and try different opportunities. Yeah, so I kind of want to do something a little bit different. Something that I've enjoyed that I think used to be subconscious and now it's very obvious to me is I like the interdisciplinary projects. And so kind of seeing how do people connect to that. And so I want to enter the policy realm after this, probably research management. I'm not quite sure where that's gonna lead to, but that's kind of what I'm looking for. It's not really perhaps doing research, but not necessarily field work as I'm doing.
Well, we said we don't look back, but I look forward only a year at this point in time. So I'm really just trying to finish up here and then after that, hopefully get a job. I've been in school like straight through the entire time, came straight from undergrad, so I need a little bit of a break. I'm hoping to get a job as a lab technician somewhere or just somewhere where I can keep doing hands-on activity. I get to handle animals or potentially teach. I TA right now and I really enjoy teaching people. But doing the hands-on field work, that's my jam. That's what I love to do.
I am very much the same. I kind of had time to explore a little bit the professional world and I realized I really like field work, working at the Department of Fish and Wildlife. Like management has its perks, it has its downside. And I think now that I'm here, this is going to be my test about how I feel about staying in academia. So if I decide that I like it more than I like being at Fish and Wildlife, then I see myself going PhD, maybe becoming a professor. If this ends up being more than I signed up for, maybe I'll go back to Fish and Wildlife and go a more conservation and management route. But I don't know, I'm very open. I'm just so happy to be here.
Yeah, I'm kind of the same as the last two. I'm very grateful for my opportunity to be here and just be able to study marine science, such an awesome place. I work at the USDA, it's like a random side quest, so I have like a lot of lab experience. I'm okay with being in a lab. I'm like kind of a lab rat, but I would say my dream job would be working at MBARI, so the Monterey Bay Aquarium Research Institute, and probably in their analytical sensors lab. I do want to stay somewhere in the analytical chemistry world, so yeah.
Does anyone else have another question or should I read off my sheet? Yeah?
How long does it take you as a student to develop your thesis?
So, yeah, you heard the because it's very different for each individual lab. And it's also kind of a different, unique experience at Moss Landing where we kind of curate our thesis along with our advisors. So just depending on who you are or who your lab is, it just takes various amounts of time.
Like some people come in knowing more or less what their project's gonna be, and that's true of other programs, and some people, like myself, do not have a project coming in. And so I had to develop that with my advisor, and then from there it varies from person to person.
Right, like I applied to mock like a job, so mine was posted of like, you are doing foraging ecology of emperor penguins. And so it wasn't necessarily that I was like, I have to go to Moss Landing, like I said, I'm not a marine scientist. I was like, this project sounds really great. And it had funding, and so that was kind of the clincher for me. So I think it's different, yeah, like Roxy said, some people have a lot of creative freedom and can really make a project their own original ideas, and well, it's kind of like, this is kind of your direction that you have to go in because it's not your money.
Yeah, I feel like also a common misconception is a lot of undergrads think you have to come in knowing what you want to do, but in general, that's not true. Usually you just kind of have to know like a vibe, like what type of lab you want to do, and within your research interest, if it matches up with your advisor, that's kind of the most you have to go.
Yeah. And even if you start with one, it can evolve over time. My thesis has like doubled over and gone back 2 steps forward, 1 step back in many different directions so many times.
Yeah, you when you apply, generally you'll talk to your, to advisors, potential advisors, and part of the process is them kind of feeling out what is it that you're interested in? Is there a direction that you see yourself heading in, or are you more like open to opportunities? And it'll just vary depending on what advisors have going on, if there's projects already going on in the lab, if they're applying for grants to maybe start working on by the time you get there. It's all very different. So I guess the only advice is to, when you're applying, just you gotta be really communicative with whoever you think your advisor is. Cause what I've always heard is that you're not just choosing or like the advisor isn't just picking you, you're also picking the advisor. It's a two-way relationship. So it's just really important to kind of like during the application process that you try to communicate as much as you can and get a sense for like where they're at. Are you gonna mesh well? Are you ready to just like figure out what it is that you want, or are they someone who like has something set up for you already?
How many graduate students are here?
About 80 to 90. Yeah, each individual cohort is about 15 to 20, and then they will stick around, some people stay longer, some people leave quicker. So about 80 to 90. Yeah.
Oh, I was
You.
wondering if, do you guys use any GIS or maps or spatial analysis in your research, and how do you use it?
They do.
Yeah, so it depends on your project, really. I use a bunch of remote sensing in mine, at least like one portion of it. I use open source satellite data for sea surface temperature, chlorophyll, and K490, which is the light scattering. And I pair that with drone data, so another remote sensing technique to look at relationships and modeling. And then
I was just gonna mention, but you can talk about it.
there's another student in the psychology lab who's doing kelp mapping using drones and then looking at kelp bed distributions along the storm coast. Sonoma.
Sorry.
Going back to the advisors, when did you guys start looking into your advisors, and how did you find like one that matched your interests, or did you just like stumble upon them?
Yeah.
I feel like I can start. I feel like there's a lot of pressure. I feel like that process is like the lengthiest part of the whole application process, is figuring out who you want to work with. I started looking at like programs, and then I also cared about location. So it was kind of how I started to narrow it down. And then from there, I was like, okay, for example, like Moss Landing, like what labs am I interested in? I was pretty open. Some people are like, know specifically the type of labs, like whether it's a specific vertebrate or whatever happens to be or techniques they want to learn, depending on what job they want. I think that's something to keep in mind. And then once you know kind of the lab, you can start looking at advisors that are doing something similar. Like the only thing that I would like to add on to that I wish was a little bit more open. I think I was very specific that I wanted to do something CalPE, which I'm still really happy about. But perhaps, like there's something else that I would have been more open to, but I just couldn't pop into that.
Yeah, I just went through the process of finding a PhD advisor, which was unbelievably, I wanted to be very thorough because it's a huge commitment. It's like a 4 to 5 year commitment. I knew, especially after this program, exactly what I wanted. And so the best way I think to know what you're getting yourself into is by talking to other graduate students. And so I spent a lot of time like researching again, kind of by location. I knew there was only really 5 states I was willing to live in. So it's kind of starting there and then going through programs and then finding an advisor and then reaching out to their students. And that took a really long time. I started that probably last, what month is it? April? Okay, I started that in probably like August, September, and submitted formal applications in like October, November. And each school's different too. Like Oregon State had rolling admissions, so I was like, "What's the deadline?" And they were like, "There is none." I was like, "When am I gonna find out?" And they're like, "Very, very soon, tomorrow." And I'm like, okay, so programs are totally different.
I was gonna say your question was about timeline, I guess. So I feel like the summertime, you can start looking, and then in the fall, you can reach out to ask questions, and usually applications are due in the winter to start the following fall. So kind of if that was more what you wanted to say for questions, that's
Kind of.
yeah.
From my experience, well, this was my 3rd time around applying to grad school. So I feel like I went through kind of the whole process. Like I went through all the emotions. The first time I was like, you know, my boss had told me, you know, you want to find a program that you're interested in, look up research, reach out to the people who are doing them that you would want to join the lab to. And that's how you figure it out. Did that, got 0 responses from anybody. Did it another year where I was kind of like, maybe I'll try again. Maybe I'll reach out to those professors. Nothing. And then after 2 years of getting nothing, I spoke again with my supervisor and was Like, do you know of any, like, what's out there in the world? Who do you know of that's studying fish that would respond to anything? And Scott's name was one that popped up, and a couple other professors, so for me, I don't know. It wasn't really a pride thing, but it was more just like, I want to get in based on my own merit.
Yeah.
And not have to like use connections, but everyone knows everybody in marine science, and you'll go so much farther if you talk to people and ask for help, and just like take advantage of the resources that you have, the people around you, because I feel like at the end of the day, everybody here wants to see you succeed. These people are professors because they care about younger people's futures, so just have to like be able to ask for help.
That answer your question?
Yeah, okay.
Yeah. Sheila?
Is graduate school as intimidating as it seems, or do you feel like your undergraduate experience has prepared you for grad school?
That depends.
Yes.
Yeah.
Oh,
Depends what
yeah.
I did in undergrad. So like I did a lot of research. I didn't know what I liked. I went through all these different weird labs. I like worked in an ant lab. I worked in a malaria lab. And I was like, don't like any of that. And then I started working with bats and I was like, oh, okay, this is something I like. And so I had done a lot of research at that point and knew that I wanted to keep going with research. I didn't have a deadline of like, I need to go to grad school by this time. I didn't apply to any other programs besides Moss. This is just a job, like I said, I applied to. But at the same time, I came in, I didn't know a single thing about the ocean. I was flying at home. Our first class, marine ecology, they were talking about limpets. I raised my hand and said, what's a limpet? I don't even know what you're talking about. That was intimidating. I definitely felt imposter syndrome of like, oh, should I even be here? I don't know what these people are talking about. I don't know the people they're talking about. But in this program, I felt like people like welcomed me with open arms and were ready to like build me up and teach me the things I needed to know. And we're really accepting of the fact that not everyone who comes here is a marine scientist. We're all scientists, and then the background of that changes so much between each person. So it is intimidating, but at least here there is such a tight-knit community that you don't ever feel intimidated by people or topics, classes. That's my experience.
Other landlocked undergrad here. Yeah, the content that I was taught in undergrad definitely did not prepare me for here. Like I said, it was very much a pre-med track and they didn't care about the ecology at all. So I knew applying to grad school that I was trying to fill that gap to get to a place that I wanted to be, which is why this program really appealed to me, because it does the practical skills, but also the education portion of it. In terms of like workload, I am the type of person who overloads myself with work, so I'm constantly busy. So the workload has been about the same, if not a little bit more. And I've had to talk to some people in my cohort who like have their blinders on and have gone through undergrad as marine bio majors who expect that everybody else around is the same and been like, you're forgetting, not all of us come in like that and being the advocate for people who don't show up with that knowledge. But yeah, I really appreciate the community here for like, yes, you can be here. And it's like one of the reasons for going into a master's program is to develop those skills for people who don't have them. So in that case, I would say, yes, I felt prepared for in that case, but in terms of actual content, Fresno State does not offer those amenities.
Yeah, but I feel like you shouldn't let that, deter you, because at the end of the day, I feel like they both said the same thing. Even though you're coming in without like a marine science degree, you can still learn the necessary skills to succeed while you're here, because that's what graduate school is, right? You're here to be a student, you're here to learn, and that's all it is at the end of the day.
Yeah, then I will add, I think it does vary from program to program, so maybe if we were in a different program, maybe we wouldn't feel the same way, and so that's like another important thing, that when you're looking for schools, you're able to talk to the grad students or in the lab that you're looking for, like, ask them all the questions, they will be very honest with you and tell you about their advisor, what their mentorship is like, what the environment is like, and if you're able to visit, that's even better, and chat with them.
Yeah. Any other questions?
I have a question. So where did you come from? You didn't know what a Olympic was.
In what sense?
You didn't know what a Olympic was, so I was wondering where did you live before from here?
So I went to school, I grew up near Seattle, but I didn't even like care about the ocean. I didn't think about it. I went to school in Idaho, so I went to school in Moscow, Idaho. It's tiny, obviously. And I studied environmental science and wildlife biology. And so I started studying bats and small mammals. And then I was abroad on a Fulbright grant and I just wanted to do science. I didn't really care who, I didn't care where. And the only lab that wanted me was a marine bioacoustics lab. And so I got kind of thrust into the world of marine science and marine mammals, which is tiny. And I was there as someone who wasn't someone who was like, I love dolphins. Like I was like, I just like science and I like animals and I'm happy to be wherever. And so, yeah, I came in, I don't know if it was.
Ocean in Idaho.
No ocean in Idaho. Yeah. So I have all this information about deer and wildlife management, rangeland management that is useless at this point. Luckily, my degree at Oregon State is gonna be in wildlife science, so I'm hoping to kind of mesh the two of having a dissertation on marine mammals and also being able to reconnect with the terrestrial side of my education. Because I don't wanna leave that behind. I wanna be well-rounded and be able to work in either ecosystem. And like I said, just have a job. That's the end. I think you had a question back there.
I want to know how you manage class research and then what self-care looks like.
They don't wanna take that.
Yeah, I can take it. Yeah, you hear a lot of silence from us because grad school is very hard and time management is very, very hard, but I feel like we're really lucky in that we're a really small community, so we really do rely on each other, I feel like. What's so great about Moss Landing is you can always, I feel like it's not competitive in the sense where you hear about other graduate schools, it's so cutthroat, it's hard to tell someone oh, I'm struggling because of X, Y, and Z. I feel like that alone, because Moss is such an open place, venting to each other and helping each other out when someone else needs thesis help, it's really, really great, which is why we keep stressing on the fact that before you go into a graduate program, really research what the environment if you can, I don't know, handle that environment and if you'll thrive in that setting. Very short, not really to the point answer, but.
I guess I would just add, I think I felt the most successful when I really set clear boundaries on what time blocks for myself, instead of just trying to do everything, because Kelsey, I guess everyone here, so we all like to do extracurricular, we to be in student club.
That's why we're up here.
Yeah. And so kind of really setting boundaries on that, especially I think now it's becoming slightly more accepting to be okay, don't do work or send emails after 5 or things like that. And really being clear to yourself. And so that's sometimes it's hard because oh, but I have this extra time, but also just go have fun. It's okay. The work will be there.
Yeah, I think too, it's hugely dependent on your financial situation.
Yeah.
So, I think all of us here, just knowing, I know that very personally, we all work hard. And so part of that is okay, how do I balance living and being able to afford food and housing with being able to get my work done efficiently and thoroughly and feel proud of the work that I'm doing and manage classes. Luckily here, I finished all my classes in 3 semesters. So 3 semesters, I was completely done with my courses. I am not taking a class. And I was able to focus on my thesis and work part-time. But we do have an intense course load the first year and a half, 2 years. And then after that, you normally have to balance your part-time work with your thesis work. That's kind of the standard here. Yeah.
It's a learning curve. I've been here less than a year now and it's it's a blessing and a curse that this place is so incredible and so amazing and everyone's doing so many cool things. Whatever it is you want to do, someone's doing it and someone is more than willing to take you on and teach you and expose you to something new. And at the same time, there's only 24 hours in a day, right? So at the beginning, you're just wow, this is awesome. I'm gonna help everyone out with everything. And then you kind of learn, like Roxy said, to kind of set some boundaries, prioritize your work or learn to prioritize what you're gonna do and how you're gonna use your time to the most effective way for you and your work. But everyone's been saying, part of it is preparing, talking to people in that program and really understanding what you're getting yourself into. I talked to 5 or 6 students, I believe, in the theology lab before I accepted the decision to come here. And I feel I had a pretty good sense of Moss Landing. I knew it was really rigorous. I knew that don't expect to get out of here in 2 years, expect to do a full research project from start to finish. And I was happy with that and I was ready for it. And I feel like when I got here, it was like not a surprise. So just really trying to put yourself in the best position to not overexert yourself.
And communicating those things, because it can be hard, especially when you're trying to communicate with your advisor. And potentially your advisor may not be here. They may be at a different campus where you have to talk to them about everything else that is going on, because they may only see what you're doing in front of them, whereas we have all of the coursework here, plus your job, plus X, Y, and Z. So making sure that you really communicate that and that you're heard as well is a really, really big part of this topic.
Any other questions before I go back to this?
No, okay, I do. Yeah. So you all work part-time. Have you been able to find work that uses what you're doing?
Yeah, I think so.
Yeah.
I'm not, no.
Serve zone?
Yeah, so like our lab has different projects that are funded and have paid positions. So I help out with the Surf Zone monitoring project where we drop cameras like in shallow water, and then same for fishes, and kind of keep track of what we're seeing at different locations. I currently work part-time down at like the boat yard here where we have research vessels that are used by students and faculty. So once a week I get to go over there and hone my mechanical skills, and that's part of kind of what you're asking about self-care, like finding ways that you are taking care of your needs and your businesses, but also is like fulfilling to you emotionally and spiritually. So like I spend all week grinding it out, going to my classes, and then on Friday, I get to just turn my brain off and like tinker away at an engine or learn how to change an oil filter on a boat engine. So having stuff like that where you can kind of find a way to kill two birds with one stone.
Yeah, I think the follow-up to that, I think it'd be good to talk about how each of us funds our own research. I think that that's something that's kind of like, I don't know, it can be taboo to talk about money, and I think for me, it's really important to talk about money, and I think it's really important to be clear about, you know, how do we live here? How do we work? How do we pay for our tuition for our research? So I think just going off of that, I think it would be helpful to talk about.
Yeah, so I also work for Surf Zone, but I work for the California Collaborative Fisheries Research Program as a member of the science crew as well. So those are two part-time jobs that I work for through the ichthyology lab that are long-term NPM monitoring programs. I also TA at CSUMB, so that covers, I get a tuition fee waiver for TAing there, so that covers my tuition. So all I have to pay for is fees and then cost of living. And there's a little bit of padding from that, not much, from the actual paycheck that you get from month to month on that. And then the part-time jobs add on to that as well. And then in addition to that, I apply to FAFSA every year. I apply to scholarships. I apply to grants. I just got a grant for research funds. So along with that, my advisor has a little bit of money on the science side to pad some things, and he has resources for like the physical care we have. So that portion of the research related things for my thesis is covered. So it's more like I just have to worry about myself.
Yeah, sure. So tuition is covered from SJSU. We get our first 2 years covered. I'm pretty lucky in the sense that my advisor Max has like a side project for me. So whenever I'm working with him on that, I get paid. And like I said, my random side quest, I have a lab job at the USDA. So I'm also part-time working. So I do part-time work in order to survive.
I'm also an SJSU student, so very fortunate for that opportunity. Probably wouldn't be able to make it work without that. So shout out SJSU for that. And then it's, yeah, just finding ways to fill in the gaps. SJSU rocks for that scholarship, but it is a little hampering that student assistants in the whole CSU system. It's not just SJSU, but they're limited to work 20 hours a week, which on one hand, we understand we're supposed to be students first, but at this point, we're not just, undergrads, like we're kind of grown-ups, like trying to live our own lives, and you can't really like live a life with 20 hours a week at part-time student work. So it's just kind of about finding a balance. And again, really being clear with your advisor. Don't be afraid to be like, funding, what does that mean? Because sometimes you're like, is that for the research that you're paying for? Is this for like me to be able to pay rent and food? And I feel like that word gets kind of thrown around sometimes, and it means so many different things.
Yeah.
So yeah, don't be afraid to asked what that is, like, am I gonna get paid for my work? Is my tuition gonna be covered? Am I gonna have to find money to pay for the seawater that I'm gonna use for my fish? So,
I think, like I said, for me, it was a job, like I was like, I can't come here, I can't pay my tuition, I can't, work, I can't live here but not be paid, like I didn't have family support, so like, this is like, I have to be paid, this has to be a job for me. And so like, I wasn't applying to grad school, I was applying to this job that happened to also be part of a master's program, and I applied for grants so that I'm able to get my tuition covered, and that I'm also able to pay a monthly stipend, and so I think there are, everyone is a mix where, even though some of us might have like family support or, this or that, we all do also still work part-time. So there's very few people that I think come here and like pay for everything out of pocket or taking loans for their tuition. I think that's very rare. I think most people come with at least their tuition paid for, and hopefully like a little bit of extra. But also for students, yeah. I mean, we're both 30, so we didn't have scholarships this year.
So most of the students are scholarship students?
So SUSU on the first 2 years basically covers our tuition. So it's just something we don't have to worry about just by being enrolled through SUSU. It's sent to our student account and it's paid for. So it's technically a scholarship, but it is something you enter the program and it's known that you don't have to worry about tuition for 2 years. That being said, Moss Landing is a long program, so no one has graduated in 2 years. And so it's something by the third year, you're thinking, okay, is my advisor gonna be able to pay my tuition? Do I have to find a grant? You're kind of then starting your workshop. How am I gonna cover this? And that's why, like we said, most of us have our tuition covered by our advisors or external grants.
On the other hand, if you're a CSUMB home campus student like me, you don't have that opportunity. So instead, what they do is they incentivize students to teach at CSUMB by getting them a tuition outsider. So it's more beneficial for them to employ students and pay them the part-time wage than to pay lecturers to take those lab sections. So, and it's beneficial for us because we don't have to pay whatever the cost is for out-of-state tuition for the semester.
So you're teaching down in Pismo?
At Monterey Bay.
Mhm.
thank you so much for the panel. Yeah, we signed up to volunteer at other places.
Yeah, so we're not, but the panel will still.
You're in great hands though. Thank you so much for coming.
Thank you. Can we continue with one more question if anyone has something burning in their heart? Yeah.
How much do you like estimate of what tuition is? Tuition for in-state at CSUMB is, with fees, it's like $4,300 a semester. For a full-time student.
It's like three or four.
Yeah, not more than $5,000. That's what I think it's like, I think it's like closer to like low 4,000. Yeah.
So are most from the San Jose states?
Most are.
Because of the scholarship.
It's getting closer though. Yeah.
And then there's also this new state university here in Monterey.
Yeah, that's what Kelsey's talking about. Yeah, that's what you get the choice. You come here. So MLL used to be a consortium, so you could come from Fresno and you could go to school here. You could come from SF and go to school here. Now you basically have two choices. You can go to CSUMB like Kelsey, or you can go through SJSU like we are, San Jose State. Most people choose San Jose State because of the scholarship, but at the same time, I have never been to the San Jose campus. I'm my third year in. I don't know anyone from San Jose. I don't really consider myself a San Jose State student, and it's just.
We like don't need to go there, because all the time.
Yeah, it's like 90 miles from it. It's far, so it's hard to get there. It's expensive. There's no reason for us to go there.
It also depends on who your advisor is, because if you're only advisor, your primary advisor is through Moss Landing, then it's more prudent to go through SJSU, but my main advisor is at CSUMB, so it's easier for me to conduct research over there if I am, my home campus is CSUMB.
Yeah, and you cannot get your Master of Marine Science at CSUMB without being here. Like if you are getting that degree, you're here.
You're here.
Yeah.
But to be here, you have to have a car, don't you?
Yeah. You could not live in this area.
You don't have to have a car.
You do, yeah, and the public transportation is basically non-existent. Yeah, so, yeah. Yeah.
So your graduations are gonna be at SJSU or?
Graduation.
Yeah, you said,
I mean, we don't really have, I don't think anyone You basically have a thesis defense, so, it's an hour-long presentation, everyone gathers in here like you guys are, we present our work, and then we're basically whisked away to a committee meeting where they say like yay or nay, 99% yay, but then that's it. It's kind of anticlimactic, I don't really think anyone walks at San Jose, I don't know anyone that's involved. So, yeah.
I was just curious what you guys do for housing, if it's kind of hard to find some here or.
Yeah, it's very hard.
Yeah, it's a different question for who you are and where you wanna be because of the different price brackets. One of the main reasons that I was like, yes, CSUMB is also subsidized student housing. So I pay a lot cheaper to live in student housing at CSUMB, but that used to be only an option for CSUMB students. Now with the new memorandum of understanding, any student at Moss Landing can get housing at CSUMB, but there's also a bunch of different options and it's kind of a pick your poison of where you want to be and how that affects your commuting and your price range and who you want to live with and all that.
Yeah, I lived in Santa Cruz for 2 years with random people, did not like that. And so I moved, into an Emeritus faculty's basically like, a second apartment and I rent from them and I live like 15 minutes from here. So it just depends. If you can find housing on your own, I think people do, but a lot of people also do live in student housing. So it just depends. We don't have any housing on this campus. Yeah. Any other questions? I want to keep you guys longer.
You guys are very, you do really great job.
Thank you. Yeah, thank you guys so much for.
Hi, it's Joan.
Hi, nice to see you again.
Yeah, so, I'll see you next month, right, in here for the
Yes,
meeting.
for the Angler Appreciation. Yep, I will see you there.
Yeah.
Perfect.